analysis and visualization and processing tools that are really popular in the community. So here are just some that we've worked with so far. Different acquisition systems, different processing, we're converting from different formats. We're working with analysis systems. I just want to point out Suite2P. Carson, our host, is the lead developer of this, and so we've worked with her to integrate it so we can now read and write NWB data with Suite2P, and we've worked with a number of other tools to do the same thing. For publishing, I'm going to go over this in a little bit, but Dandy is a neurophysiology data archive that specifically supports NWB data and leverages the NWB standard to provide additional tools like search and data visualization and data summaries. So the archive really understands neurophysiology data concepts and leverages the NWB standard to provide really useful tools beyond what you would get with a generic archive like Figshare, which is great for storing data, but doesn't really have any neuroscience specific tools. So the idea is to work with these tools instead of competing with them and creating our own tools. We want to work with all of the great analysis tools that already exist and try to get them to read and write NWBs so that they can be used interoperably together. And if any of you are building tools, we'd love to talk with you and figure out how best to save the data into NWB and read NWB data into your tools. So I just want to talk briefly. I don't want to go too in depth about kind of the back-end architecture of NWB, but the idea here is that it's actually quite modularized. And this allows us to have flexibility that can accommodate the different needs in the neuroscience community. So for instance, this orange one in the middle is representing the actual data standard. So we have a specification language, which is blue, which tells you about different kind of data primitive concepts like group and data set and attribute. And they're defined in this in the specification language. And then those concepts are used to define specific data rules. Like if you have an electrical series, then you need to say what the sampling rate of that electrical series is. And you need to say what electrodes were recorded from and those types of rules. That goes here in the orange in the data standard schema. Now those rules can be stored on disk in different ways. We support HDF5 as our primary data storage backend, but since we've kind of abstractly defined all these relationships, we can also support other types of backends, like for instance, a relational database. And we also have modularized out the data APIs. So we support a Python and a MATLAB API, but since we've separated the API from the schema itself, you could implement a new API in, for instance, Julia or JavaScript or some other new programming language. So the idea behind this modularization really is that it kind of future-proofs NWB. If there's a new data storage that we want to leverage, we can do that because we've modularized that out. If there's a new type of data that we want to represent, we can do that because we've separated that from the schema language or from the specification language. And if there's a new programming language that becomes really popular in the field, we can adapt and build an API for that programming language. So in terms of community adoption, there's this energy barrier to data standardization. I mean, I think we can all appreciate that if all of the data was in a standard format, that would be really helpful for the field. But the fact is that it takes a lot of work to convert all of those data and analysis tools to NWB. So how do you move this along? Well, one way is sticks. The NIH could say, well, you have to do it or we'll withdraw funding. That is one approach. It's not my favorite approach, but it can be useful in combination with other techniques. But really, I'd much prefer carrots, right? We want to provide analysis and visualization tools so that people just see that it's so useful to convert to NWB that they're just incentivized to do it on their own. And then the third type of way that we can influence this is guidance, right? Basically what I'm doing right now is giving talks, explain to people the value of NWB and also how to use it. So we've been growing over the last few years, both in terms of our code base, but also in terms of our outreach. And we're really focusing now on reaching out to the global community. We are Neurodata Without Borders, after all, so we are really interested in reaching out. Most of our network happens to be in the US, but we're also starting to reach out more and more into Europe and to countries all over the world. And we're really interested in really building diversity in our community. So NeuroMatch is perfect for this. So we're really grateful to be part of this program. And I just wanted to point out all of the great people working on NWB. I'm a small part of a much larger system of software developers, of early adopters and power users, executive board, sponsors and funders, and now all of you are now part of the NWB community. So thank you for coming to this talk. I really appreciate it. You can go to nwb.org for more information and also check out the links that I put in the chat. Okay, and so without further ado, I think I'm just going to jump into a tutorial to show you how to actually get data into NWB. So we're going to do a tutorial on extracellular electrophysiology. So this is an example, just hypothetical experiment of an animal that's moving around in a maze. There's some trial structure. There's raw electrophysiology data. There's also LFP, and then this is after spike sorting that we have some spike times that we want to write into the file. Now, NWB supports lots of different data types. This is just an example that contains some of the most common data types that we see. But I'll show you afterward how to work with the documentation to figure out if you have a different type of experiment, figure out what types of neuro data types would be useful for you. So you're welcome to just watch, but if you want to follow along, pip install PyNWB. We also have all of this in MATLAB, but I've heard that NeuroMatch is really focusing on Python, so that's what I'm going to focus on here. But if you are bilingual or if you prefer MATLAB, I think you'll learn a lot of useful concepts here. Yeah, could you send us the link to this tutorial? So if you go all the way to the top, Lou, there's a link. That first link there is a link to this tutorial. Okay. So, right, you can also use Conda to install it. Okay, so the first thing you want to do is set up an NWB file object. So NWB files generally hold all of the data for a single experiment session. So you want to package all of the electrophysiology, all the behavior, trial information, all of the data you need to analyze that session goes into the NWB file. But first, we're just going to instantiate it with some generic metadata. So, so first we have the session start time. Now, all of the times that you have in your NWB file are going to be with respect to that session start time. It's going to be in seconds with respect to that session start time. So this is when it started. We've got a time and a time zone. And now there are a few required fields here. You need a session description and an identifier and the session start time. And then there are a lot of other fields. We show a few of them here. Just metadata, mostly metadata that you would want to provide if you're sharing this data. So who ran the experiment, what lab were you in, institution, and any related publications. And there are lots of other fields for kind of notes about this particular session. So, so first I'm just going to show you how to add information about the subject. So we're going to create a subject object. And this holds information such as the age, the species, genotype, and sex and description. So we're going to use diagrams that look like this. This is a diagram style called the UML standard. So NWB uses object-oriented concepts. If you're not familiar with object-oriented programming, don't worry, I'll walk you through it. But the idea here is, well, to start out with, you have a certain class subject, which has attributes. So in this case, subject ID is required, and then you have age, description, genotype, sex, and species that are optional. So when you input data for these different data types. The subject ID can be just whatever your lab uses. The age, we use ISO 8601 duration format. So instead of saying just 90 space days, it would be, the format goes P, which stands for period, 90, and then D, which stands for days. This is really useful because we store data from different species. So if you had said 90 and it's a mouse, okay, well that maybe it's obvious that it's 90 days. But if it was a human, then it would probably be 90 years. So if you wanted to make this 90 years, it would be P90Y. And if you wanted 90 weeks, P90W. It's pretty straightforward, but this format allows us to handle ages across a large diversity of species. And then for the species, we recommend the Latin binomial name just to, again, have standardization across labs. And then for sex, F, M, U for unknown, or O for other. So this is how you would add that information to the file. NWB.subject equals, you create a subject object, and then you input this data. Pretty straightforward. Okay, now getting into slightly more complex territory. We're going to put in the position information, so position over time of the animal. So, position is stored in a data object called spatial series, and spatial series extends time series. So this open arrow represents a relationship where spatial series has all of the attributes of time series and defines additional attributes. So every time series in NWB has a name, a description, the data. The data also has to specify the units of the data. And then the timing can be specified either as a starting time and a sampling rate, or so the starting time and sampling rate is used if you have a regularly sampled time series, or timestamps, which you can use if you have an irregularly sampled time series. So in addition to those fields, spatial series, first of all, it defines that the data must be timed by features and in unit meters. And then you also have the additional attribute of a reference frame, which tells you where 0, 0 is in the data. So, we're just going to create a spatial series with some fake position data. So here we have our position data. We're just going to use linspace to create some fake stuff, and then we'll create timestamps. So, okay, so these timestamps technically are regularly sampled. So in that case, I probably would use a rate and a starting time to represent it. But in this case, we'll just pretend like, oh, they could have been irregularly sampled. So we'll just store the timestamps. So here's how you create the spatial series object: name, description, data, timestamps, reference frame. I guess I should run these, huh? Okay, and then I'll just point out when you print out NWB file, you get a nice summary of what's in there. All right, so I'm gonna create the subject object and create the spatial series object. Now, when I print this out, you can see all the fields within it. There's a comments field, which we didn't fill, conversion, which defaults to 1, data, here's all the data we entered, description, reference frame, timestamps. Okay, now these spatial series objects, they go inside of a position object. And you can have one or more spatial series objects inside of position. So this diamond-shaped arrow represents that relationship, that the position object holds spatial series objects, and the spatial series objects extend time series. So here we can create our position object, and we'll just assign spatial series as the object that we just created up here. And then, okay, so now we need to create a behavior processing module. So, NWB differentiates raw acquired data, which shouldn't change, from process data, which is the result of some processing algorithm and might change. So in this case, let's assume that we use some kind of video tracking algorithm to get the position data, in which case the video would be raw data, but we're not putting that in the NWB file. but the result of the tracking algorithm would be process data. So, so in order to organize this data into the different types, we have processing modules. So we're going to create a processing module called behavior with a little description, and then we're going to add the position object to the behavior module. So, so all told, we now have a behavior module that extends from that is an instance of a processing module that contains a position object. The position object contains a spatial series object, which extends time series. Okay. And now I'll just do a sanity check and write all of this to the file. So we're going to create a new NWB file called ecephys_tutorial.nwb, and we'll write the NWB file that we just built. So now how to read data back from the file. So there are, well, one way to do it is with the PyNWB API. So you can do a read command like this, and then print out different data objects within the file. Here I'm saying, okay, we're calling it read NWB file, and then inside the processing module called behavior, you can print out that processing module or the position object or the spatial series object within it. You can also use other tools. I actually really like to use a tool called HDF View, and that gives you another view into the data where you can navigate through it and understand the structure of the NWB file. Okay, let's go with let's now add trial information. So trials are stored in an object called an interval, a time intervals object, which subclasses a dynamic table object. So dynamic tables are kind of default structure for storing tabular or table-like metadata throughout NWB. They're used for trials, for electrodes, for sorted units. You're going to see them a few times in this tutorial. And the nice thing about dynamic table is they offer flexibility in that you can add columns to the table. So dynamic tables can define required and optional columns, and then you can also just add whatever columns you want, which is really nice specifically for something like a trials table, because often trials have a lot of metadata that's kind of unique to that experiment. So, so here's the dynamic table, and time intervals extends dynamic table, and it requires that you have a start time and stop time. And again, all times are with respect to the session start time in seconds. And then, so this trials table is just an instance of the time intervals type. Okay, so so basically the dynamic table structure looks like this. You have an ID column, and then each column is its own data set. So you have a start time data set and a stop time data set, and then you can add more. So in this case, we're going to add a column called correct, which is going to take Boolean values, and it's going to say whether the animal correctly behaved on this trial. So to do that, we call nwb file.add trial column. We give it this column a name and a description. And then from there on out, we can add trials row by row. You can add a start time and a stop time and a correct value. And then if you're familiar with Pandas data frames, a nice convenience function is you can take any dynamic table like trials that we've just built, and you can say to data frame, and it will give you a data frame that then prints out like you're used to with Pandas. So here you can see we've created this dynamic table with start time, stop time, and a correct value. Okay, now let's get into extracellular electrophysiology data. So, so what we want to do is store voltage data, but before we do that, we need to store information about the electrodes that are used to store this data, to record this data. So there's a number of pieces of metadata that we need to specify in order to set this up. So you need to define a device, which is is the probe, or the a device could also be like a data acquisition system. Electrode groups, which are kind of formations of electrodes, perhaps multiple formations within a probe. And then we have an electrodes table, and this table, each row of the table represents an electrode. So you can put the XYZ position of the electrode, the impedance, the location in the brain. So if you want to mark it belong to a specific brain area, if there's any filtering applied to it on acquisition, what electrode group it belongs to. So this group references an electrode group, which in turn references the device and the group name. So, so electrodes, again, inherits from dynamic table, which means we could add additional columns to the electrodes table, but we're not going to in this case. We're just going to populate this data here. So I'm just going to create some fake electrodes. I'm going to say, okay, we have 4 shanks and we have 3 electrodes per shank. So first I'm going to create a device. And then with each shank, I'm going to create an electrode group. And again, the group has name, description, a link to the device, and then the location. And then I'm going to do a for loop over the number of electrodes per shank and create actual rows of the electrode table, which include a link to the electrode group that we've just created. Okay, so again, we can convert this to a data frame and look at it using pandas. So this is what it looks like. We can see we have, it says 11, but it starts at 0. So we have 12 rows of this, and you see all of the data that we populated here. Okay, so just a point is that in the above loop, we created the electrode group object in NWB file. And then when we added electrodes, we passed the electrode group. But this is it's actually a link to the electrode group table. So we're not creating different electrode instances of electrode group every time. We're actually creating a reference to this single object. So it uses this link feature in HDF5 that allows you to link to specific groups. Okay. Right. So let's talk about storing actual electrical series data. So electrical series is a type that's used to store voltage traces over time, whether it's raw electrophysiology data or LFP. And it, again, inherits from time series. So you can see this here, time series, electrical series extends from it. It specifies that the data must be timed by channel, that the units must be in volts, and that it needs an electrodes object. So this is an important additional attribute. It specifies what electrodes from the electrodes table are being recorded in this electrical series. So, so we're going to use a new type of data called a dynamic table region, which allows you to specify a sub-region of the electrodes table, or you could specify every electrode in the region. So, so that's what we're going to do here. We have a convenience function, NWB file.create electrode table region. You specify the region. In this case, we're going to say all of them. So we just take the range of the number of electrodes, and then we just have a description here, which says all electrodes. Okay, we'll just run this. And now that we have this electrode, this all table region, this electrode table region, we can create our electrical series. So I'm just going to create some example data, some randomly generated raw data. And then I'll use the electrical series object. I'm going to name this electrical series. It doesn't have to be named electrical series, but if you only have one, kind of convenient to name it that. And then data, and then electrodes here, we're using this dynamic table region that we just created. And then this time I'm going to specify the timing, not through timestamps, but I'm going to use starting time and rate. So here's an example of how to do it that way. And this is more often the case when you have electrical series, that it is regularly sampled. Okay, so as I mentioned before, NWB has different places to put different types of data. So, so we have different kind of larger groups. So we have acquisition, which is used for raw acquired data, processing, which is used for the result of processing, and then you have analysis, which is the result of data analysis, and stimuli, which are their own category. So in this case, since we have raw electrical series data, we're going to put it in acquisition. So here's how you do that: NWB file.add acquisition. And then we're also going to store some LFP. So here I'm going to generate a new data set of random data. I'm going to create a new electrical series. I'm going to say it's referencing the exact same channels, but I'm just going to call it LFP electrical elect series. Now LFP there's a data object called LFP which contains electrical series. So this new electrical series object, we want to put it inside an LFP object to kind of label it as being LFP. So here we're creating this new LFP object, and then we're saying electrical series equals LFP elect series, which is this one right here. And then again, this is process data. Since it's, we're going to assume that this LFP has been filtered and downsampled. So we are going to create a new processing module. We'll call it EC-ephys, and we'll add LFP to this processing module. Okay. Now, now the final type of data I'm going to add here is spike times. So this is the standard way to add any kind of firing information, the result usually of spike sorting, into NWB. We have a table called a units table, which is again a subclass of a dynamic table. So so the first thing I'm going to do is and because it's a dynamic table, you can add columns. So here I'm going to add a column to the unit table called quality, and this is just going to be a description of the sorting quality. And so after that, I'm going to randomly generate data. And here I'm going to say, okay, I have 10 units, and then I can, I have Poisson, I'm using a Poisson process to try to get the number of spikes, and then I'm using an exponential distribution to get a reasonable simulated spike train. So this is just simulating data. You don't have to worry too much about it. This is the line that is the most important, where you add the spike times for a given unit, the quality. Here I'm going to say all of them are good, and then there are additional columns like waveform mean, where you can say what the mean waveform of any unit is, and then you're creating this dynamic table, which then of course you can do the to data frame and view it. So here we are with our units table. We have quality, spike times, and the waveform mean. Okay, and then again, I'm going to write this to EC-ephys tutorial. Okay, now it's written. And now I want to go through how to read data from NWB. So, so we've already seen something very similar to this, where we're going to use a context here so that it automatically closes when we're finished, read the file, and then you can go inside of acquisition and look at electrical series, or inside of processing and look at your LFP data. So I'm going to print all of these out. Now, there's kind of an additional feature of NWB, which is that data sets are read lazily, which means that you, like if you just ask for the data, it's gonna give you the name of the data set and the shape and the data type, but it's not gonna actually give you the values of the data. To get all of the values for a given data set, you do this, and that will actually read and print out all of them. But the nice thing about this is that it doesn't read, it only reads the section of the data that you want it to. So it doesn't read the entire data set. So you can have enormous data sets, like gigabytes or terabytes big, that you don't want to read all into memory all at once. So you can access just sections or regions of that data set. So here, if we say, okay, read this electrical series data set, but only read the first 10 rows and 3 columns, that'll just read this section of the data set. And the key here is that it's not just only printing out that section, it's only ever reading from disk this section of the data. So this allows you to really efficiently handle really large data sets. And similarly with the spike times, if you ask for just the 0th unit, you just get the spike times for that unit, and the rest of the spike times aren't read. So this is a really powerful feature, but I guess for newbies, sometimes it's tricky because they want to do this and then like, where's the data? You know, so all you have to do is use this syntax and you get all of the data. Okay, so I've gone through this tutorial, the Python tutorial for extracellular electrophysiology. There's also a calcium imaging tutorial and an intracellular electrophysiology tutorial in Python. There's also advanced tutorials like iterative data write, so being able to write really large data files without having to load all of it in memory, how to build extensions to handle new types of data, advanced HDF5 features like how to use lossless compression to reduce the size of your data set without changing any of the actual data values. And then we also have tutorials in MATLAB if you're interested. We have this YouTube channel that that is a video recordings of a lot of these tutorials, as well as breakout sessions, really nice guided tours for a number of different NWB enabled tools, including Spike Interface and Suite2P and Cayman and a lot of other really great tools. And I guess one thing I forgot to show you before, but I want to make sure that I showed you just before we go into a little break, is the Dandy archive. So you can go to dandyarchive.org, and it'll forward you to this site. So you can just go to this site as well. And I'll just show you all of these public data sets that are currently available in NWB. So this, for instance, is a 2 terabyte data set from the Buzaki lab, and it's hippocampal recordings in NWB with behavioral data. So it has position data of the animal running through a maze. And you can view the data here. And here you see there are NWB files that you can download individually, or you can download, or you can use the Dandy command line interface you can pip install Dandy and then do Dandy download and point it to this data set to get all of the data downloaded in parallel. So all of this is free.
Hey, Ben?
Yeah.
Oh, sorry. Just a question about the data.
Yeah.
Are they going to enable a way to see the metadata, for instance? So if I go on this website and I want to see, or that's always on the first page, and then you assume every single data set has all of those.
Yeah, so what you're looking at right now is the asset summary. So we're basically creating, we're automatically extracting this data from the NWB files and creating a summary. So for instance, there's a position data object in the NWB file. So from that we can say, okay, there's a behavioral technique. And there's an electrical series object in the NWB file. So from that we say multi-electrode extracellular electrophysiology recording. But as you point out, this is just a summary. This is kind of aggregating the data across all of the different NWB files. So the answer is it's not quite ready yet, but we are working on an asset level metadata. Actually, there is something. We have something up. So if you go to an individual NWB file, you click the I, Okay, so we haven't made it pretty yet, but this is basically exposing the result of a REST API call. But it does tell you information specifically about this this NWB file. So it has a decomposition series, LFP, a units table. It's a mouse, right? So it tells you information about specifically this NWB file. So the what we haven't quite, well, I guess step one is to create like a prettier view of this data per file that we're probably going to put like in an info panel here. And then the next step is to enable NWB file level search. So right now there we have pretty good search where you can type, you can type in NWB and see all the NWB data sets, or you can type in like mouse, and you're going to get what from a mouse. But this is on the Dandy set level. So what we're working on is NWB file level searches, where you can hone in specifically on those files. And then beyond that, specific data objects. So you can say like, okay, give me all the electrical series that are like beyond this duration and have at least this many channels, or like are recorded from neuropixel probes. So we're definitely we're just starting to build out more robust search based on this metadata.
That's awesome. That's really useful as a computational person. Thanks. We have a couple questions. One that's related to Dandy, which is, does Dandy have EEG data?
Yeah, you can store EEG data on Dandy. So it currently supports BIDS and NWB. So you could write that EEG data to NWB. I think you could also write it to, I think there are supported BIDS formats for EEG as well. And yeah, you could put EEG data on Dandy.
Cool. And we have another question, which is it possible to store a spatial series without having time data? So for instance, to store a connectivity matrix. And sorry, this question's from Lydia.
Thanks for your question, Lydia. So a connectivity matrix, is that, so that's like of the connection of neurons in the brain?
I guess I'll say the example from Janelia is we have the fly connectome, for instance. And so we have a separate kind of structural information that could be stored.
Yeah. So I think, so for spatial series, it's really, I mean, it's kind of specifically for the movement of an animal in a physical or a virtual space. I would hesitate to use it for something else, because if people are querying on that, they're kind of expecting that type of data. But NWB can provide the structures to store any type of data, including connectome data or structural information about the brain. I think you'd probably be better off creating an extension that specifically satisfies your needs, because I really kind of doubt that spatial series would be sufficient for you in that case. But you can construct a data schema specifically for connectome data that has kind of 3D coordinates and maybe a surface mesh or something like that. We've used the extensions to, I guess, something that's pretty similar. It's too bad I took those slides out, but let me see if I can get them back really quickly. I'm gonna look up slides from another presentation, because it answers this question pretty well, I think. Okay, so we had the task of storing ECOG data, and they have a similar type of spatial need where these dots represent electrodes on the brain, and with ECOG, it's really important that you represent the structure of the brain itself. The kind of the gyri and the sulci, they're really important where the electrodes land on those. So we needed a way to store the information of the surface of the cortical surface. So this is, as you can see, a very intricate formation, but actually it's pretty easy to develop a schema to store this information, because you just develop a triangular mesh. And a triangular mesh just has vertices, points in 3D space, and then faces, which is how to join those vertices into triangles. If you make those triangles small enough, you can basically approximate any surface. So here is basically all of the code that you need to write an extension to handle this type of data. So you have a surface object, you create a data set for faces, you create a data set for vertices, you maybe add some metadata attributes, and then you export it. And that's more or less all of the code that I needed to develop an extension for this. So I think you'd use a similar approach if you were going to store Connectome data.
Cool. Thanks, Ben. I guess then, so there's some other questions, but I guess if you write an extension, you host that, that would be something that then the NWB website would link to, basically?
Yeah, so what you would do, you would share it in two ways. One is the NWB files that you create actually would contain the extension. So anybody who's trying to read that file, they would first read the extension and understand what's in the file, and then you would kind of automatically generate APIs based on the extension that's already in the file that allows you to read it. But if you want to pass code that's kind of beyond what we can automatically generate, so for instance, if you want like validators that maybe if you're using this vertices and faces example, your faces reference indices of the vertices array, so you don't want them to be negative. You can't have negative values for that. So maybe you want a validator that says you can't have negative values. And there are other types of kind of visualizations or other things you might want to package with your extension. And so for that, we have this catalog. Let me pull it up. Let me share my screen again. Yeah, so here are some of the extensions that have been created for the mini scope. This is the ECOG extension that I talked about. We have an extension for NIRS. So we're developing this library of extensions. And the cool thing about sharing this as a library is another group can, if they want to store NIRS data, instead of developing their own extension, they can use yours, and then we can have standardization across the extensions.
That's awesome. So there's another question about fMRI data, and I don't know if that's something that Neurodata Without Borders is going to support.
Yeah, so I think, I mean, we try to think carefully about the scope of NWB. It's already quite broad because we have electrophysiology and optical physiology. I think, so there's the BID standard, which handles most neuroimaging stuff. So I think if you wanted to store like PET or fMRI, I think BIDs would probably be a better standard because they already have a lot of data in that format and a lot of tools that can leverage that format. I mean, you could in theory develop an extension within NWB to handle that, but I think you're probably better off using BIDs. Now, there are overlaps, right, between, I would call BIDs neuroimaging and NWB neurophysiology, but it's a Venn diagram that has overlap. So like with ECOG, like what is ECOG? Is it neuroimaging? Is it neurophysiology? There's often MRI and CT scans that go along with the ECOG. So I think, in that case, we've worked out a way with BIDs to be mutually compliant. So you can use the BIDs folder structure, directory structure, and then the data itself can be an NWB. And that allows you have a little bit of duplication of metadata, but that allows you to be actually mutually compliant and use both BIDs tools and NWB tools to analyze it. In the case of fMRI, I think you're probably better off just going with like the DICOM or NIFTY, whatever BIDs recommends within their format.
Cool, thanks. There's another question that's interesting. Is it possible to add behavior from a second mouse to the same data set, like from a resident intruder experiment?
Yeah, that's an interesting question. So you certainly can enter information from, you can enter a new data stream. The question is whether it's linked, like if you kind of want an explicit link to say this information is from this mouse and this information is from this mouse. And that wouldn't be I guess the default spatial series object or the default position object don't have a link to a specific subject. I think that would require an extension, basically like a multi-subject extension. It would be a pretty light extension where you basically say, I'm going to inherit from position and just add a single attribute, which is a link to a specific subject. But yeah, so if that's something that the question asker is interested in, they should reach out to me. And if they really want to put in data in NWB, I'm happy to help them do that, because I think a lot of labs would probably need to do that. But it's kind of an example of how you can make a few adjustments with the extensions library to handle lots of different types of experiments.
Cool. Thanks, Ben. I guess I'll ask this too, because I think there was some GitHub training yesterday. And so would you want people to ask these questions in GitHub issues? Would that be?
You can ask them in GitHub issues. We also have a Slack channel. I'll put that here. So we
Awesome.
have a growing community. We have a newsletter you can sign up for. Yeah, GitHub is a great place to ask these questions. Yeah, but we're monitoring all of these channels. Happy to help.
Great, thanks. I think we got through all of the questions. Does anyone else have questions before? Did you want to have like a 5-minute break, Ben?
Yeah, let's do a 5-minute break. And then when we come back, I can go through optical physiology.
Awesome. All right, so we'll see everyone 5 minutes past the hour in Eastern Standard Time. That's 11:05 a.m. Hey, Ben, you ready to get started again? You are muted, by the way. Ben is going to get started in a second. I think he's coming back on with optical imaging data. And so for those of you who use like Sweet2P or Cayman or these other tools, a lot of them export the data automatically to NWB for you. So you might already have your data processed in NWB format, which is great. Oh, I need to add Ben again, sorry, to the screen.
Oh, no, wait, I'm back.
I can hear you.
Okay.
Right.
I don't know what happened there. It wouldn't let me unmute myself. I was trapped in mute, but I made it out. Okay. 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 So I'm just going to go over. I don't know. I don't know how to answer your question, Jindu. Maybe Carson can help. Okay, so this optical physiology tutorial, it starts out the same way with NWB file, subject, position information. So I'm going to scroll through that, and then we get to optical physiology. Okay, so here we're going to store information about the imaging plane, an acquired 2-photon series, image segmentation, and the fluorescence DF over F traces. Okay, so just like with electrophysiology, before we stored the electrical series information, we need to store information about the electrodes. Analogously, for optical physiology, before we store any actual data, we need to store metadata about how that data was collected. So we have an optical channel object and a device object and an imaging plane that contains optical channels and references device. And the imaging plane contains name, description, excitation lambda, indicator, location, optical channel, device, and grid spacing. So information about how, about the spacing of the pixels of the image. And then, okay, so we're going to create an example device called microscope in the NWB file. So we do that like this, NWB file.create device. And then an optical channel, which has a certain emission lambda and a name and a description. And then an imaging plane, which uses the optical channel and the device, and also has a name, description, excitation lambda, indicator, location, etc. Has grid spacing, which is in meters, and it has origin coordinates also in meters. Okay, so now that we have the imaging plane, we can create a 2-photon series. So again, 2-photon series, like electrical series, extends from time series. It has all of the attributes of time series, name, description, data, and then some way of representing timing information. And in addition to that, it requires a reference to an imaging plane object. Okay. So there are 2 ways of storing the data here. So one is just storing the data normally. So here I'm going to call this 2-photon series 1, and I'm just creating a data set of 1s, referencing the imaging plane, and I'm giving it a rate. If you don't provide a starting time, the assumption is that the starting time is 0, and then the unit is just normalized amplitude. Now, a second way, because often people are storing data with a TIFF stack, and they don't want to duplicate this data because it's quite big. So we have a way of referencing an external data set. So here you can create a 2-photon series. And instead of putting data, you put an external file. And this points to the relative path of the TIFF data. And again, imaging plane. Here you have to say the starting frame, and you have to say that the format is external. But then you can put a starting time and a sampling rate. And then here's how you would add those to the NWB file. Okay. Now that we have, now that NumPy is not, did I not run some of these? Oh, I didn't run the whole beginning. All right, let me just run through it. Okay. And this too, and this. All right. Now we want to store information about the ROIs, so plane segmentation. So we have this sequence of images, and we want to markate sections of that image as putative neurons or some other anatomical region of interest. Oh, the link to this notebook I can give you is right there. Okay, so we need a plane segmentation, which subclasses dynamic table. So similar to how units represented sorted units, and they had a dynamic table, plane segmentation represents the analogous piece in optical physiology, which is segmentations of an image, also at the dynamic table. So I guess I'm missing this figure, but it's reproduced down here. It's grayed out, so it's a little harder to see. But we have plane segmentation table, which inherits from the dynamic table. And you can specify these ROIs either using an image mask or a pixel mask. So an image mask has the same shape as the image, so width by height, and then you can have like ones be the region and zeros be outside the region, or you can use a float. You can have it be like a soft assignment, so like anything non-zero is part of the ROI. Another way you could do it is with a pixel mask, where you just label which pixels in the image are part of the ROI. And then again, you need a reference to the imaging plane, which we already have. Now, once we have that plane segmentation, we want to put that in an image segmentation object. And the image segmentation can take one or more plane segmentations. And then we want to put that in an OPHYS processing module, which contains this image segmentation object. And OPHYS is an instance of a processing module, much like the behavior and the EC-EPHYS processing modules that we saw in the last tutorial. All right, so we can do that. Now, here I'm creating the image segmentation object. We have a convenience function create plane segmentation, and the name, description, you have to link to the imaging plane, and then this is an optional argument for the reference images. And then we're going to create a processing module called OPHYS. We're going to give it a description, and then we're going to add the image segmentation object to the OPHYS module. So the NWB file now contains an OPHYS module, which contains image segmentation, which contains this plane segmentation that we've created, which has the ROIs in it. Well, not yet. It doesn't have the ROIs yet, but it will, just in the next step. So we're going to create some image mask objects So again, image masks they have the same shape as the image. So and they just use ones or some non-zero value to indicate what part of that image is part of the ROI. So I'm just going to randomly generate. Run this again. Okay, so this is my very square example ROI. Another way to do it is with pixel masks. So here I'm going to generate 30 of them. And basically you just store the X pixel, the Y pixel, and then the weight of that pixel, which could again be 1 or could not be 1. Okay, and now, so I'm storing these in, I'm actually gonna, yeah, okay, put these in the plane segmentation by add ROI, which I have here. So I've created 2 different plane segmentation objects. One of them that I'm just using the previous one that I created here for the image masks. And then I created a second one so that I could demonstrate how to use this other way of specifying ROIs. You shouldn't really mix these 2 methods. So I put them in 2 different plane segmentation objects. So here I'm showing the pixel masks for this second plane segmentation object. And again, this is a dynamic table, so you could add a column about perhaps like the quality of this ROI, or maybe a putative cell type assignment or something like that. Okay, so now we have the ROI response series. So this is basically where you take the image sequence and you kind of overlay it on the ROIs, and you pull out the response over time of each of the ROIs. So our ROI response series, it should be no surprise at this point that ROI response series extends time series, and it's time by ROIs. And then it requires a different, it requires a new attribute, which is a dynamic table region pointing to the plane segmentation table. So this is perfectly analogous to the electrical series requiring a dynamic table region pointing to the electrodes table. So basically what this allows you to do is create an ROI response series that records activation for a region of all of the defined ROIs, and which could potentially be all of the defined ROIs. So here we're going to create a dynamic table region by this convenience function off of plane segmentation. So create ROI table region. Here I'm just going to say the region is the first and second. We're just going to store the activation of the first 2 ROIs. And then here I'm going to create this ROI response series object with a name, the data. So here the one feature of NWB is that time series data also always has time in the first dimension. So here we have our 2 in the second dimension, which is the number of ROIs, and 50 is our number of time points. Now we are going to provide this this dynamic table region that we just created, a region referencing the plane segmentation table. Say it's units, which could be just not applicable if that's the case, and then the sampling rate. And again, if you don't provide a starting time, then it's assumed that the starting time is 0. So I'll go ahead and run this. Okay, so this is a lot, but we've already covered most of it. So we've already created our imaging plane. We created our plane segmentation table that held the ROIs. We've put that in an image segmentation object, and we've just created our ROI response series that extends from the time series. Now, the final piece is that ROI response series can go inside a fluorescence object. So just like LFP, the LFP object could handle more than one electrical series objects, which actually hold the data. Here we have the fluorescence object, which can hold 1 or more ROI response series objects. And then the fluorescence object goes inside of this OPHYS processing module, which is an instance of processing module. So we're going to use the fluorescence object. We're going to import that from the OPHYS module of PyNWB. And we're going to say our RR response series is this object that we just created up here. And then we're going to add the fluorescence object to the OPHYS processing module, which you've already created. Okay, there we go. Just as a note, there is also a DF over F object, which works the exact same way as fluorescence. So DF over F also holds one or more ROI response series objects. And then finally, you can just write it like this. And the reading here is pretty similar. So you can use this context, NWB HDF5 IO. So this is read as NWB HDF5 IO. Here's the file name. You want to open it in read mode. Call that IO, and then you do IO.read to actually read all of the metadata, all the attributes and the group structures, but it doesn't read the data sets. So here's how you would open that and print out the data. So these small pieces of data, like single data values, it does read automatically, but those larger data sets, like the actual data associated with the ROI response series, is not read. So you can see data, what's output here is not the actual values of that data set, it's just the summary of the data set. So if you did want to access the values of that data set, again, you would use this square bracket colon syntax. That will read all of the data like this. Now, it's not a big deal right now. I mean, this is really not a very big data set, but you do sometimes have very large data sets, so it's useful for real experiments. And then if you wanted to just access a sub-region of that, you could do it like this. Putting the zeros in here is a little funny. Okay, so this just reads a sub-region of that data set. Again, now we've covered extracellular electrophysiology and calcium imaging. And we even have more data types even for those. It's just kind of the most common data types that we see across those modalities. We also have intracellular electrophysiology, advanced writing, advanced features, and we have a bunch of tutorials in MATLAB. But let me just show you what to do if you're trying to convert your own data and maybe you have some needs that are outside of what has been shown in this tutorial. So let's say you want to store like eye position data. So you can type eye here in the search bar and, okay, pupil tracking or eye tracking. So, okay, I can go to this object and say, okay, eye tracking contains spatial series. It needs a name and it's one or more spatial series objects. And the spatial series objects tell you about the movement of the actual eye. So you can go into PyNWB and search around. I have to say, if you're trying on your own to if you have an experiment, you're trying to figure out what neurodata types to use, it can be difficult to do that. So I encourage you to reach out to me. It's like my job to help you use NWB. So don't feel bad at all about reaching out to me or anyone else on the team for that matter. And you put a question up on the Slack, GitHub, and then we can point you to specifically the parts of the documentation that would be useful for you. Feel free to navigate on your own, but I don't want you just kind of wandering in these docs forever because there's quite a bit of material to handle lots of different diverse types of data. And we've spent years kind of figuring out how to map experiments onto data types, and we'd be happy to just help you with yours. I think that's more or less what I wanted to go over. Are there any more questions?
I think you just got one actually about INCF, and so I don't know if NWB has a partnership with INCF. I have a question for
you. So we've been working with INCF for years. So we are a supported best practice of INCF, and we are working. We've worked with them to create a training space, or I think it's called training space, which is like a collection of NWB courses. And we are the upcoming INCF, I forget what they're calling it, but like the professional training or something that INCF is hosting. We have a 3-day workshop with them. And we also have working groups that are working on extending NWB to specific new types of needs that are kind of organized by INCF. So we, for instance, we have a working group right now that's working on developing an ontology for representing intracellular electrophysiology stimulation patterns. So, you know, when you're doing intracellular electrophysiology experiments like patch clamping, you have signals that you send in, like, yes, INCF training week, thank you. You have signals that you send in like a rectangular pulse or like triangular pulses or sine waves or chirps. So there are kind of these standard prototypical inputs that you put into patch clamping experiments. And so we're working on a working group that's organized by INCF to handle that. And we're kind of building a structure for formally representing that. So we work with INCF in a number of different ways. But we're really excited about this training week coming up because it's a full 3-day workshop that we're doing with them. So this is just kind of a taste, really, of NWB. There's only so much we can cover in 2 hours. If you're really interested in digging in and joining the community, the training days in late August would be a great opportunity to do that. It's a 3-day workshop that's hosted by INCF. Great.
Cool. Are there, if you have other questions, you can put them in the chat or in the ask a question box. I'll ask you another question again about Dandy, but I guess if I'm a person looking to analyze data, it would be great if it is possible to link to the code repo that they're using in their paper so that you have the way that they're loading their NWB data.
Absolutely. So we have, kind of brings me to this feature of Dandy, which is they have this Dandy hub, which is am I sharing my screen? I'm not. Okay, let me do that. Okay, so here I'm showing Dandy Hub. You can get to this by hub.dandyarchive.org. So in order to access this, you first need to go to Dandy Archive and log in here, and then your GitHub username will be registered with us. And then once you do that, you can access the hub. And this will give you actually access to free compute resources that have really efficient access to NWB data. And it also gives you access to a number of example notebooks. So here are notebooks from for data set 55. And they've been provided by the lab that submitted this data. And it shows you how they can read the NWB data. I'll just run through this. And this is just reproducing one panel from their paper, but put some requirements up here and then import stuff from Dandy and stuff from their own. Oh no. Let's see if this works. Live debugging. We can do it. Let's just try. That. Seems to have worked. Okay, so this actually shows up a really cool feature of Dandy. So what I'm doing right here is I'm using the Dandy client to get the path of a specific Dandy set. So I'm saying look inside of Dandy set 55 and get.
Can you zoom in a little bit? Sorry, it's kind of it's kind of small. Thanks.
Sure.
Cool.
No problem. All right, so I'm saying look inside of Dandy set 55 and get this path. And I'm going to say, give me the S3 path of that. And then I can input that S3 path into this NWB HDF5 IO. So this is the function I've been using up until now. I've been giving it the file path on a computer on your local disk of where the file is. But now I'm actually, instead of giving it the local path, I'm giving it an S3 path. So if I just show you, it's actually like a location online. And I'm using this driver called ROS3, which stands for Read Only S3, which is a new driver that has been developed by the HDF5 group to stream data directly from an S3 location. Dandy stores all of its data on S3. So, so what we can do is give the location of this. Oh, go ahead. Sorry.
Sorry. So this won't just work in the browser though. If I download this notebook, I could run the same command and get the data from. Cool.
You can. Well, you can. If you download this notebook, you can run the same command. It's a bit tricky to set up the environment right now, to be honest with you. We're working on releasing it, so it's not quite there. We have this environment that we've set up called NWB Stream, where we've done it for you. And the Dandy archive it sets up its compute nodes in the same location as the S3 bucket. So it's actually very fast to stream data this way. You can run it locally on your computer. You can set up the environment locally and you can run it and it's free and it works. But it's a little bit hard to set up the environment and it'll probably be slower. But so I recommend doing it this way.
I mean, this is really cool, but I guess like you don't mind if everyone is accessing the data this way. There's
no
compute
go ahead.
That's donated for this.
Just go for it.
That's awesome.
Yeah.
Okay.
Yeah. Right. So what's happening here is the NWB file, just how I talked about how it loads data lazily. Well, that's going on here. So the actual data sets. So some of the lighter metadata is being read, but those large data sets are not. So you can have a super large data set. I mean, this file contains ECOG data for 24 hours. And it's read right away because we're not actually reading the data in that data set. But what we can do is read just sections of it through that indexing trick that I showed you earlier, and efficiently pull out only the data we need. So here, this is a diagram of the different kind of states that the subject was in during this 24 hours. So sometimes they're sleeping and sometimes they're awake and active, or inactive, or block list would be if they're like disconnected from the system for some reason or something like that. So I guess, oh yeah, that's much better. Right. So this is just an example of how you can submit notebooks that replicate the figures of your experiment. And not only that, but efficiently stream data directly from the Dandy archive, use the Dandy API to identify which data sets that you want to stream data from, use PyNWB to stream that data directly, and then from there on out, you can just use your own analysis tools. And the data can be arbitrarily big. I mean, we have like NeuroPixel, Spike GLX raw data sets that can be tens of gigabytes at a time, and it can be efficiently read using this system because we're actually lazily reading that data. We're only reading it when we need to. Another really cool, let me just show NWB widgets. Another really cool feature of this, it works on its own again, but it works even better when you use it in Dandy Hub because it's streaming data. So I'm going to pull, I'm going to say, okay, give me data set 53, which is a NeuroPixel data set from the Giacomo lab. And this data set is an example. So this is just showing off that lazy reading. So again, I'm pulling the S3 path of this data set from this, or rather this NWB file from this Dandy set in Dandy set 53. And then there's this library that I'm actually leading the development of which is called NWB widgets, where the idea here is that NWB widgets can look at an NWB file, can parse what neurodata types are in it. Say, okay, oh, this is an electrical series, this is a trials table, this is a units table, and it can offer reasonable visualizations for those data types, reading the data and the metadata from them so that it automatically renders a nice view of the data. So here we're kind of navigating through the NWB file, and here I'm looking at voltage data from this NeuroPixel probe. Now I'm only reading 5 seconds at a time. And that's how I'm able to do this. I haven't downloaded the entire, I put how many gigabytes it is, 46 gigabyte file that contains all of the raw data. I'm only looking at 5 seconds at a time. And so the data streaming for that is quite fast. But you can look at processing, you can look at behavior. Okay, so we have eye tracking data. All right, so you can see the eye position over time. You can change the window. Let's see, I want to look at 30 seconds of data. Okay, and this is all streamed live. So you can see it's quite fast. If it's not very much data, it's quite fast and responsive. Now, of course, if you ask for a large window of raw electrophysiology data, it's going to take some time to download. But it works quite well for exploring data sets. And the nice thing about this is you don't have to install any software and you don't locally, and you don't have to download any data locally. So you can navigate through data sets using this mechanism without having to really have a lot of investment on your own end, and kind of interactively explore the data sets that are posted and see what might be useful for you.
This is really great. I mean, as someone who analyzes lots of different data, it's several hours downloading the data, making sure you can load it correctly, thinking about what visualization to do for this data specifically. But if you have all of this embedded, then it's really great to quickly just take a look at the data and see if you can like test a computational hypothesis or just see what's going on.
Right. So I'm using NWB widgets here, but we have the NWB object that you can access programmatically as well. So I can go in here and say NWB.acquisition. I mean, this shows you, you can access it just like I was showing before. We'll pull this and then .data. Okay, again, I'm not going to load all of this because we would be here forever, but you get the idea. So here I'm displaying the raster of spike times that have been sorted. You can get different units. You can navigate through units this way, time this way. And there are actually some a little bit more sophisticated things that we've, so you can do a PSTH. There's not any trial information that we could group on. So here we can look at activity with respect to the start time of the trial. Yeah, I don't know. You can navigate through here and find a good unit.
Definitely looks like there's some structure in some of those units.
Yeah, there's something going on. At any rate, so the nice thing about NWB is it's really extendable. We have tabs here for different visualizations, but you can put your own custom visualization in here. And I'd be happy to work with anyone who's who's interested in taking a visualization they're working on and reading NWB files, and we can help you incorporate that into NWB widgets so it could just become another tab here. Yep. So yeah, I mean, we're really interested also in when people share data to publish notebooks that reproduce their specific figures as well. So we kind of have hitting, I think, both sides of the data visualization spectrum. We've got like exploratory analysis with NWB widgets, which is kind of meant to be generic and all-encompassing and a good start, but it certainly won't get you all the way there. And then we encourage people to submit notebooks that have specifically reproduced the figures from their papers by streaming data directly from Dandy.
That's great. I think it's a big step forward in reproducibility. So are there any other questions from people? Has anyone else ever downloaded any NWB data sets? I guess I'll say as someone adding visualizations to Suite 2P, it's nice to have, like, for instance, if we've had requests for PSTH analyses inside Suite 2P, but if the data comes out of Suite 2P in NWB format, they could do those analyses on Dandy Hub, basically, or with the NWB widgets on their local computer, which would be great.
Yeah, and you can import NWB widgets. There's another project called NWB Explorer, which kind of provides a similar type of portal for you to navigate through NWB data sets. And they import NWB widgets and use some of the NWB widgets for their own visualization. So it's open source, open permissive. So if you wanted to use it within Suite 2P, we could also do that.
Oh, that's awesome. Cool. All right. Are there any other questions from people? If not, we can sign. All right. So is there a way to write here in the chat? Oh, someone's asking about the notebooks again. So I'll scroll up. I'll get the OFIS one that just happened. Oh, actually, I think you're referring to the link to the Dandy notebooks. Could you put a link there?
So I can't link directly to those notebooks, but you can go to hub.dandyarchive.org, and I can put that in the chat.
Oh yeah, that would be great. Yeah.
Yeah, so once you create an account, so you're going to have to go to dandyarchive.org to create an account, and that's all you do is sign in on the upper right, and then you'll be ready to go. And then you can sign into hub.dandyarchive.org, and the example notebooks that I just showed you are already there.
Very cool. So we can all be analyzing hippocampal data in the next 5 minutes. Even.
In the cloud. Yeah.
Yeah, that's so cool. All right. Are there any other questions?
Also, I didn't show this, but when you start Dandy Archive, the hub, it asks you what kind of compute node you want, and so there's like a base node, but there's a pretty large. You can request substantial compute resources that allow you to like parallelize your work pretty well. So it's actually like, we don't yet have GPU support, but we do have like multi-core support. So you really can start analyzing in parallel analyses in the cloud, streaming data directly from Dandy right now. We would really love for people to do that, because we want to build a whole analysis ecosystem around the data that people publish.
Yeah, no, that's really cool. So I guess if you want to close out, what kind of advice would you give to all these young computational neuroscientists?
Yeah, I mean, I think it's worth thinking about. There's this kind of drive to go into like publish or perish mode, where you're just like focused specifically on getting those publications out. And definitely that is important for your career advancement and for the scientific process to publish those papers. But I think it can be, what can kind of get neglected in that process is like building good software and longitudinal support, and making sure that the data that you collect is packaged in a way that others can use it. So, I mean, I think that largely in the past, there just weren't the tools available to do that. And so you could hardly blame anyone before now for like not packaging their code or their data in a good way. But I think now there's a kind of critical mass of support and of users that allow you to really do this. And I think I think over the next 5 to 10 years, we're really going to start to see a really good shift in credit attribution and in prioritization, where people are really going to start to appreciate data sets that have been really pivotal in the field. I mean, I know in machine learning, there have been some data sets that have really, that are really considered to be great contributions to the field, because so many machine learning algorithms kind of use it as a training set and as a way to evaluate. And I think we'll likely go in a similar direction, where there are going to be really key data sets or key software that leverage these data standards. And I think the key to really being influential in that context is going to be packaging your data or your software, whichever side you happen to be on, in a way that others can use it, providing user support and documentation, and make sure it integrates well with the standards that that are being developed in the community. I guess that's one piece of advice if I had to say something. And I'm happy to help with any of that. Really, please reach out. I don't how many people are here? How many people am I telling to reach out to me?
Now we're at 40. We were at 80 before the break.
So. Okay, yeah. Yeah, please, if you're interested, happy to follow up with you, and we can even schedule like a one-on-one, and I can learn about your needs. We're always interested in getting new users. So yeah, it was a pleasure to talk to all of you. I hope you learned something.
Thanks, Ben. That was great. And that was wonderful advice. And as someone who does a lot of software development and tries to be user-friendly, I've had a lot of valuable experiences with people reaching out to me. And so you're playing the long game because it's not immediately rewarding, but it is a lot of fun.
Yeah.
All right. Thanks, everybody. Have a good rest of your
MacArthur. We're going to have Emma Ganley, Nikki Pfeiffer, and Lucia Savella. I'm sorry if I mispronounced your name. And then we're going to have lots of time at the end for some Q&A. So as your questions come up, please use the Ask the Question button at the bottom of your screen, and we will either type an answer or we will address it on screen when we hit the Q&A time. We'll also potentially be running some polls throughout, and so this is a reminder to our speakers that if you want to ask the audience any questions, we can easily set up some polls. Okay, so without any further ado, I will pass the mic to Dawid. Thank you so much for being here.
Thanks, Megan, and hi everyone. It's really great to speak to you. I'm Dawid Potgieter. I'm the director for programs and discovery science at Templeton World Charity Foundation, and I wouldn't call myself an expert in open science, so I'm sort of more here to tell you why it's important. But yeah, after that we're going to get some real sort of practical expertise on this topic. So my own background is, obviously I'm currently doing research at work for a foundation. My own background is looking at monoamine neurotransmission in the basal ganglia. So I did my DPhil at Oxford a long time ago, and I actually left straight after that to work for a foundation to sort of work on the funding side. And something that's been really interesting is, as a funder who looks for the best kinds of projects, or at least the most like cost-effective way to support scientific research. There's been a really amazing trend of more and more people recognizing the value of best practices in open science. It's certainly something that's caught on at the foundation, and that's why we have decided to sponsor Neuromatch Academy, and why we're taking out the time to talk about this here, because as I'm sure pretty much everybody on this call is super smart and could be a really great scientist, and you've got excellent technical skills. And that really is the important stuff, right? Being able to do the experiment correctly, having the right technical skills. Like, you've got to do that, otherwise it's not going to work. But we think of open science practices as sort of that extra little thing that often sort of doesn't get highlighted as much as it should be. And with just a reasonable amount of effort or inspiration, or even knowing where to look and who to work with, that it could really boost the quality of work quite a lot. So I wanted to say a few things about why open science is important. I'm sure if you're here, you kind of think it's important anyway, and so I'm not gonna tell you that it's sort of I'm not gonna kind of stick to the basics, but I'll kind of share a few things that there are trends that I've seen in my last 7 years or so of working for the foundation. And the first one to mention really is that, you know, when I started, I originally studied biochemistry, and it was with this sort of vision of being a great genius scientist who's going to discover amazing things one day. And you watch movies, and there's always like somewhere there's this genius scientist like that figures out there's one thing that saves the world. It seems so cool. But over time, that like impression of what science is has just been replaced with something much more realistic, which is not just that science is a bit slower and less fun than it is in the movies, but also the idea of the lone genius who's super smart and solves all the problems. It doesn't really work well anymore. Great scientific innovation now comes from teams. It comes from people who work well together. I'm not saying you'll never find like a genius who like solves something big. Like maybe you can, but I think for any individual, if they decide for themselves, how should they work as a scientist? By far the most effective way to get things done is to work as a team. You know, it's that way you utilize not just your own skills, but the skills of other people as well, and you benefit from their expertise. So, you know, doing that is not just about sort of being good at the technical aspects of scientific research, but there are other elements like how to work well with others, which, you know, part of it's being nice. I'm sure everyone here is nice, but part of it's also like taking a slightly different perspective in terms of realizing that the work you do and the results that you present don't have to just be right. They have to be done in a way that gives people confidence that you're right, and that's a pretty big difference. You know, yes, if it's right and you can prove to yourself it's right, great. But if somebody else has to work really, really hard to have confidence that you're right, it's just not going to go as far as it should. And so thinking about any piece of scientific research as, you know, how easily can I give people confidence that this is the case or that's the case? It's just a very, very useful approach. It might not require any more effort, but it's a very useful thing to have when you want to work with a team, when you want to make the most of working with others. There are other skills, of course, skills like learning how to pick the right questions, learning how to work with sort of conceptual hygiene or conceptual clarity, and not confuse concepts. And I'm not going to talk about that because there are other speakers who later this week will be talking about that, and their sessions are going to be amazing. So definitely go to those. Yeah, this idea of being good at working with teams is just really, really, it's getting more and more important in science. And there are two approaches of thinking about this. And I'm going to kind of go through those very quickly, and then I'll hand over to the next person. So the one approach is to think about the problems that we face in science. I'm sure pretty much everyone here has heard of sort of outcries about a replication crisis in certain disciplines, or problems of people you know, p-hacking their results to statistical significance, or you know, coming up with a hypothesis after they know the results, or hacking, or simply just, you know, doing a bunch of tests and then publishing the ones that are that say what they want, or you know, putting in a file drawer the ones they don't. That kind of thing gets like a lot of press, and it seems, you know, just very newsworthy. But open science is much more than trying to just solve those problems. In fact, you know, depending on who you speak to, like, those problems aren't as much as of like an immediate crisis as you know, some headlines would like to think. But you know, they are still, you know, they can be problems at some time. So the idea of thinking about open science as solving problems in science is useful, but there's actually a much bigger side to this that I'd like to sort of finish off with, which is that open science can speed up the pace of progress. So best practices in open science could speed up the pace of progress. So it's not just about making discoveries, but how many we make, how fast over a long period of time, how much confidence people have in those discoveries. Those things are really, really important. And coming back to this idea that, sort of modern science is going to increasingly be about teams of people working together, people not just trying to get data, but trying to make sure that others can have confidence in the data. If you sort of zoom out and think of this not just between the time period between now and when you publish your next paper, but maybe over your whole career, 1, 2, 3 decades, maybe more, that some of you will be in this sector, the skills that you would learn to not just make new discoveries. I'm sure many of you are going to make amazing discoveries, but not just to make discoveries, but to go through a process that by the time you get to the discovery, everybody can have confidence in your work. Those skills are actually going to save you a huge amount of time. It could make the discoveries far more efficient, because when you make your amazing discovery and you want to publish it, and somebody asks questions about, oh, well, did you do this? Did you do that? Did you check this thing? Did you p-hack there? If you have followed these practices in open science, and many of them we're going to talk about today or this evening, depending on where in the world you are, if you follow these, it's much, much easier to explain with a lot of transparency what you've done and give people that confidence in your work. So just to briefly go over some of these practices, because I'm sure talking to a wide range of people here, most of you might know it, but just in case, there are sort of a few groups of best practices in open science. There's the idea of just making your findings available free of charge. So that's the open access side of open science. And I'm not sure if we have Joe on the line just yet, but if we do, he'll be speaking soon about people sharing. Oh, great. Okay, welcome, Joe. He'll be speaking about the open access side of open science. And then there's also a side, an aspect of it that's about transparency. Open access as well, but it's about transparency throughout the process of scientific research. And so we're excited to have Emma here, who's going to speak about that transparency at the point of writing your experimental protocol. And Nikki's going to talk about having that transparency at a sort of slightly broader level as you plan your experiment and plan the steps around the experiment. And then we're also really excited to have Lucia talk about taking that to a slightly next level, and having something that's sort of pre-registered, go through a peer review process, and essentially have a journal approve it, so that it could speed up your publication steps as well. So that's my brief overview. If I haven't convinced you that open science is the way forward, I'm sure they will. So I will just, thanks again for joining us, and I'll pass it over to Joe.
Thanks so much, Dawid. That's great. So we will bring Joe up here onto the screen. And I think what's going to happen is, because there are now 6 of us, it may not let you share your screen without me kicking someone off. So I will have to probably kick off Dawid, because he is done with talking. But if you have questions, Dawid, then we can bring you back up. So thanks so much for being with us, Joe. And we will go ahead and if you click the little button, you can share your screen and we can try and make sure the permissions are working.
Hopefully you can hear me.
Yep.
It's saying 6 simultaneous video sources, so you might need to kick another person. 2 people I will be caused to sort of keep you through.
I'm sorry, Dawid. We will bring you back later for the panel discussion. Okay, why don't you try now?
Okay, got it. I got it. And I'm just going to do my entire screen, so you'll all see each other for a moment.
Inception.
Okay, all right. And now you should see full screen slides. Is
Looks
that right?
beautiful. Okay, so I'll get out of your way and let you do your thing. Thank you so much for being here. We're really excited to hear about what you have to say.
Thank you for the invitation, and I'm super excited to be using a new video casting thing. You know, Zoom was getting a little dull, so it's good to mix it up. So start my timer. Awesome. Well, thank you so much for inviting me. I'm certainly really excited to be here for a number of reasons, some of which will be revealed as we go through, I guess. But I am here to talk a little bit about how to share your paper, and that has a sort of little bit of a double meaning. I'm Joe MacArthur, and a little bit more on me later. So I'm told you've had sort of a week of this, so I'm going to skip sort of right to the end, right to the takeaway of what I all need to hear from this, essentially, and sort of spoil that ending. So I'm going to talk about a tool called Share Your Paper. And the idea behind that tool is in just a few clicks, you can make your paper open access and start getting more citations for your work and more visibility. It's a free tool. You can do this with almost any paper at pretty much any time. And you can still do all of that while publishing in your favorite journals as you normally would. The system looks a little bit like what you see on screen at the moment. A very simple interface where you just sort of go to our website, tell us the paper that you're looking to share. We'll guide you through what versions to share. You upload that, and bam, it's shared and made available to everyone forever. And you know, this is if all you hear from the rest of this talk is that, then that's fantastic. But in the next 30 minutes, I'm going to tell you a little bit more about how the tool does that, what the benefits are, how it works behind the scenes, answer some questions that you probably have, and guide you on your journey in open science. So it's a tool order for 30 minutes, but I'm sure that we can do it. I'm going to start with a little bit about us, and most specifically about myself. I once was a neuroscientist. I studied pharmacology at University College of London, or UCL, and I was sort of a keen neuroscientist. I researched neuroscience at Eli Lilly in the UK as a part of placement years and things like that, and did some cool research that I'm not allowed to tell anyone about. It's always a shame. And did some work at UCL as well, which you know, I dug through my photo archives and dug out some of my old posters and things like that. But partly for your amusement, but also mostly for my own amusement, to be honest with you all. So I sort of have a little bit of an idea of where many of your brains might be at. I actually sort of dropped out of academia before going on to do a PhD. I was so that was my dream from being a kid, but in the end, having discovered sort of open access and some of the challenges around the scientific process, p-hacking, and the reproducibility crisis and things like that, I sort of, for myself, no longer felt like science was the best place for me, and started to do this essentially, and some of what I'll talk about during this talk. But this was a bit of a jumping off point for me. But one of the things I always go back to about from this time in my life was my time at Eli Lilly, where, you know, I'm not sure if they thought about it this way, but internally in the organization, they did what I would now call open science. Of course, it stopped at the you know, at the doors, but I've seen sort of that world, and it is you know, a pretty exciting one. But of course, you know, we would want to do this for everyone. So what I left science to work on an organization that is now known as OA Works. You can tell what we think just from that, but we're a nonprofit that builds tools that tries to make open access easy and equitable. Is our way of summarizing it. It's of course an oversimplification, but a good one, I hope. And I'll talk about one of those tools today. One of the most important things that we like to know about all of our tools is they're free. They're open source, which means you can interrogate the code, reuse them, build upon them in somewhat like we would advocate for research articles and data. And they're co-designed with the community, researchers, librarians, and research funders alike. You don't really need to know very much about us, but just a little bit of context. We're obviously a pretty small team. One of the things we make is called Share Your Paper. And that's what I've been invited to talk to you a little bit about today. So what does Share Your Paper do? Always a good question. So Share Your Paper does does one main thing. Its main job is to help you with what's called self-archiving or green open access, basically making your paper open access. We can go into the definitions of those things, but I often find that unhelpful. One of the ways to think about it is just self-archiving allows you to publish as you normally do in most journals, but make a free copy available somewhere else. And we'll sort of get into the nitty-gritty of what that somewhere else is and things like that soon. The job of Share Your Paper is simply to make those things really easy. These are powerful processes, but they're a little bit tricky to learn. And we try and take sort of some of the barriers out of doing that. Both for you as individual researchers, but also for your libraries or funders and the folks who are trying to support you to get your research out there and to have it have the most impact that it can. So, what does Share Your Paper do? It helps you do self-archiving, and self-archiving helps you make your work open in a pretty powerful way. For those, it's a website at shareyourpaper.org and the service. So I like to keep that pretty light because I think most of the time it's best spent on sort of why should you actually care about these things. These are probably ideas that you're not super familiar with and seem a bit jargony, and so why is it worth paying attention? So I've got 5 reasons that I think are reasonably good. So the first is that the outcome of these processes is that your research is freely available. That means that first off, you can read it. Often, you will have access at some point, but maybe lose it in future, and it means that your future self can read it. It can mean that employers outside of academia can read it. I always like to say that it means your mum can read it. It means that doctors and physicians might be able to read it, and healthcare workers in general do not often have the access to the research that we wish they did or that we think they do. And so it means that they can read it. It means the policymakers can read it. It means that everyone that you would like to have reading your research can read it, and that includes sort of patient populations that you might be working with, or the participants of your study. If it's behind a paywall, more often than not, they won't be able to read it. And the most powerful part of this process is that it is free. With that higher visibility and ability for people to read more of this, tends to come more citations. So there've been sort of dozens of studies of the impact of making your work open access, including specifically via some of the routes we'll talk about today. And the great news is that you get to get more citations by sharing your work openly. And I don't need to tell you how important that is as a measure of scientific impact and for careers and things like that, even if sometimes that's probably not that healthy. The other reason why you should care about it is, I like to think it's the right thing to do. Often we're being funded by public money, and certainly building upon that in a heavy way. And so it's just sort of the right thing to do for the public to be able to read that research and use it in powerful ways. The other argument that I think is sort of similarly compelling is that lots of other people are doing it. About 50% of papers that are published every year now are open access, not all via this route, but via this route and others. A lot of folks are getting the benefits of open access and sort of, but I know that sometimes it's difficult because you don't have money for APCs, or the right journals aren't available. to get some of those benefits. And I think one of the great things about self-archiving is it lets a pretty broad group of people get access to open access, or sort of make their work open without necessarily having some of those hurdles attached. And then 5 is that this is something that is free to do, and it can at least be easy. Can be is in brackets here because I think sometimes it can be a bit tricky, but obviously I'm going to try and talk to you about how Share Your Paper can help with some of those hurdles. So we've gone over what Share Your Paper is as a tool to help you make your work open, especially via self-archiving or green open access. We've gone over some of the reasons why you might care. Let's talk a little bit about how Share Your Paper itself actually works. And I'm going to do this in 2 ways. I've already shown you what it might look like to use Share Your Paper for your paper. And you can see this again now, where you can go to the tool, put in a DOI so we know what paper you're looking to share, and then we'll walk you through the steps of doing that. But of course, many of you might not want to use Share Your Paper for whatever reason, or might have other places where you might want to share your work. And so I want to talk about some of the more sort of meta steps, like what are we doing behind the scenes that you can do yourself in other ways, just with a little bit more time and effort. So I'm gonna, so I'll sort of talk you through the tool and also how you can do things without using the tool. So on the left is the Share Your Paper tool, which hopefully you're getting a good look at. On the right-hand side, I've broken down like seven major steps that you will often sort of go through, from choosing which paper you want to try and share, to checking your rights to share it, finding the right file to share it, choosing a place to share it, choosing a repository, creating your record in that repository, sometimes waiting for approval, and then finally getting a link and actually being able to share it with people. Those are sort of 7 major steps. And so we'll walk through those now and show you how they work. So step number 1 on the journey to making your paper open access is of course choosing which paper. So you might have some papers that are already open access, in which case you can certainly choose to self-archive them again, but I might not put them on the top of my list personally to sort of spend some time on. But if papers are, you can self-archive a paper basically before, during, and after you've published it. You might do that in different ways at different points, but basically, if you've got a bunch of papers published already, it's not too late. You can still go back and make those papers open access. It's not too late. It's just a case of starting somewhere. With shareyourpaper.org, starting this process, you just need a DOI, and we'll take it from there. So here's the first sort of major step with self-archiving. So, some of you might have heard, you can make your paper open access for free in all these journals in all these ways, and thought, oh, maybe this is like a bit too good to be true. And it's not, but you do need to follow a couple of rules while you think about doing this. Most, but not all, journals will allow you to self-archive your work. The rough number is about 70%, but depending on sort of exactly the sorts of journals you're interested in, you might find that all of the journals that you're interested in will allow self-archiving. It's much more common in the sciences and in neuroscience, for instance, than it is in sort of the humanities and things like that. So your mileage may vary, but it's very likely that you'll find a journal that will allow you to self-archive your work. The reason you need to check is because when you're publishing with the journal, you sort of have to sign these agreements, the copyright transfer agreements with the journal. And sometimes in those agreements, they'll say that you can or can't make your work available through this route, and often will give you some sort of terms about how you can do those things. So for instance, a good example is what's known as an embargo period. So the journal, when you publish there, will say you're absolutely allowed to share your paper for free through a tool like Share Your Paper or some other route. You can't do it for 6 months or a year after you've published it here. And so when you're thinking about publishing, self-archiving your paper, you just need to keep some of these things in mind. So the good thing is that you don't have to figure out all of these yourself from scratch. There are tools available to help you figure this out. So you can go to a cool tool called Sherpa Romeo. The URL is on screen now. And you can just type in your journal, and it takes a little while to figure out the interface, but if you stick with it, it'll tell you both what versions you're allowed to share, when you're allowed to share them, and where you're allowed to share them. And so that way you can figure out for your paper where you can share it. The neat thing about Share Your Paper is that you don't need to do any of that. We will figure it out in the background for you and just sort of guide you from there. We know all the answers and have done that work. And basically what we'll just tell you is what your next step is. And you don't have to sort of worry about all of those things. We'll just manage them in the background without you needing to think about it. But if you don't want to use our tool, that's totally fine. You can still do it another way. And that's grand. So once you've figured out what you're allowed to share, often what you'll need to do is go and find the accepted version of your paper. This is just the version, usually after peer review, that the publisher said, yes, we're totally happy to publish this. We accept this to the journal, and then there is often a small process where you move through, maybe correcting a few typos or formatting it into a sort of final PDF or something like that. That is more often than not the version that you're allowed to self-archive. So you have to sort of check with your co-authors, or sometimes go into the publisher submission system, or check your hard drive or Google Drive or wherever you've got these things stored, and find that version of your work. The Share Your Paper tool provides some, I think, helpful instructions for and things not to do as well when finding these versions. Depending on how good your filing system is, it will be easier or harder, but it's usually pretty straightforward to find these. You just got to know what you're looking for. And of course, you can check in with the journal about what version this is if you're unsure. On this page of Share Your Paper, which you've already seen a bit of, you can just see we're guiding you through that sort of step by step to make sure that it's easy and get it right. And also, if there are some small things to that accepted version that you want to clean up, that's normally completely fine. So, for instance, if you've got figures in another file, it's fine to move them in. Or if you've got some comments on that version, you're fine to remove them. Small tweaks like that are totally fine within this process to just neaten things up. Then what you'll need to do is choose a place to put the research. I said at the start that self-archiving was putting a free version of the paper sort of somewhere else, not on the journal website, but on another website. Generally, the places that we recommend to do this are called repositories. Those are special sites that house, preserve, and make your paper discoverable. So for instance, they're like harvested really well by Web of Science or Google Scholar. Like, they know to look there and find your paper. And also they have special features which make sure that sort of, the website will never go down, and you'll never sort of, go to look for your paper and see that it's a 404 or gone missing for some reason. So repositories are really, really important places where this content goes. And because of that, lots of institutions will often run their own repositories. So for instance, the institution that you're at might have a repository, or your discipline might, for instance, or your subdiscipline. Often finding these takes just a little bit of work. I usually recommend just asking your librarian, but Google can also help, or other tools like Open Door, which I'll give a link to when these slides get sent around. Often you will already know about these repositories. You will have stumbled upon them during your journey, but maybe just sort of not quite recognize them as places that you can put your work as well. For Share Your Paper, we've already, again, chosen a repository for you, so it's another thing you don't need to do. We deposit into a repository called Zenodo. It's sort of a general purpose, very large repository for content. It's really well managed. It's run by the team at CERN and funded by the EU. It's just sort of a good sort of general purpose place to put papers. So we put yours there when you deposit through our tool, and it's there for safekeeping and made discoverable. So another way that we try and make your life easy. Then getting towards the end now, I promise, is finally you'll need to actually create your record. So, if you're on a separate site, your mileage may vary. I haven't looked at all of them to figure out what the process is, but it will look something like this. You'll usually need to make an account with that service and then enter the paper's metadata. Metadata is just a really fancy word for the title, the authors, when it was published, what the original DOI was, and things like that. That can sometimes be a little bit of a laborious exercise of copying things from from one place to another, but it's important to pay a little bit of attention because this is how your paper will be discoverable and linked to your published version. Then, of course, you'll need to upload the file that you found earlier and insert some of those statements and adjust settings that I spoke about earlier to make sure you're compliant with that policy. What that often means is there's a set statement that you need to put in that says, this paper was originally published by X journal on this day, and if you want to find it, you should go here. It's normally quite simple and available, but again, it's just a bit fiddly and takes a little bit of time to get your head around. And for instance, I was talking earlier about those embargo dates where if you publish a paper really recently, the journal might want to have you not publish this for a little while. You can normally have a setting which will publish your paper at a certain date in the future. And so that means you can upload it whenever you get the time and just sort that, have it be actually published a little bit later. Again, you might be finding a theme here, but with Share Your Paper, you just upload your paper, which is really the only thing that you definitely have to do. We can't get that file. And then we do the rest for you. We'll get the metadata. We'll figure out all those statements and settings. That's all done automatically in the background, and you don't need to worry about it. But again, if you want to do it yourself, you totally can. And it's straightforward. It just takes a little bit of time and effort. Finally, it's some repositories you might need to sort of wait for approval for your work, because getting all of those little steps before can be tricky. And sometimes if you get things wrong, publishers can get a little frustrated, shall we say. Many repositories will want to check the record before it's sort of finally published and give you a link. That's just for basically for your protection and theirs, and to make sure that the record can be improved before it's published as well. So if you're lucky, that someone will be doing that, and you don't need to worry. And sometimes they'll get back to you with small changes that need to be made or things like that. Again, continuing the theme, Share Your Paper sort of does this instantly and in the background for you, and we'll let you know as gently as we can if you got something wrong on the way, and sort of help you out with getting that fixed. So often the problem is that you sort of got the wrong version of the file, and we've built a system that will sort of figure that out and help you resolve that problem. So the nice thing about that is it happens instantly, and you have to wait around for someone else to look at things, and then probably email you sometime when you're not thinking about this so much anymore. So finally, if everything goes as planned, then you'll get a link to the free version of your work, and that will get indexed in all the places that you would hope it would be. Landing in Google Scholar or Web of Science, or your ORCID profile if you have one of those, and it's certainly good if you do. Sometimes getting indexed by all of these places can take a couple of days, or at worst a few weeks, because everything sort of has to sync up. But give it a little while, and you'll find that your work is now freely available in all of those places. And you will have sort of done yourself and the world a good service. That's pretty exciting. So yeah, that's sort of the whole process end to end. And then the next thing to do is just get the next paper and do the next one. Obviously, I think I make it sound a little bit boring walking through all of these steps, but with some of our tools, I promise you, these are all really straightforward things to do and really quick. So again, this is a real-time graphic showing how quickly you can do things if you're prepared. It can just take literally a few clicks and a few seconds to get all these things done if you've got the right tools and support. And your library may even have a mediated service where you can send someone your final article and they'll do some of these things for you as well. So a couple of frequently asked questions before I finish up. So one great question is like, do the journals support this? And I can tell you the answer is yes. Journals often actually publish policies in support of this and guiding you through how to do it. The journals benefit from the higher citations and the higher visibility of this work just as much as you do. So the vast majority of journals support this, and it's something that universities and funders are also really supportive of and often mandate as well to make sure that your work is open. Another good question is, is it okay to share the accepted version? And again, to that, I say yes. I know that it can sometimes feel a little unusual when you've spent all this time creating a carefully crafted publisher PDF to share. But as most folks are just interested in sort of being able to read the words, and if they can't get to that pretty version, the accepted version is far better than nothing. And it's very probable that you will start to see these accepted versions turn up in more and more places because they're starting to be linked and indexed from most major places where you can discover research. The third question is a really good one. Like, doesn't ResearchGate and academia.edu allow you to do things like this already? And my answer to this is like sort of. ResearchGate and academia.edu certainly allow you to host papers, and sometimes they'll make those sort of more broadly available. The catch there is that they don't do some of the checks that help make sure that what you're uploading is sort of within the contracts and policies that you've signed when you publish the work. So sometimes work can get taken down from there and things like that, and that's something that ideally you'd probably want to avoid. The other thing is that often to get onto those sites, you need a login, and to have that login, you often need to be affiliated with an academic institution. And so obviously that sort of nullifies some of the benefits of doing that sharing. But of course, there's no reason why you can't put the accepted versions and sort of do your upload there. It's not quite as good as a repository, but it's certainly better than doing nothing at all. And then 4, how do I get my supervisor on board? I know that when you're starting out early in your career, a lot of things run through your supervisor, and that this is always a sort of conversation that needs to be had. I would point to some of the points I've made here about the benefits to you and therefore the lab of doing these things, and the sort of very low risk of taking these actions. These are things that are allowed by the vast majority of journals and encouraged by large universities and foundations. This is something that is becoming sort of increasingly common and a useful skill to have as a young academic, even if it wasn't so common when your supervisor was being trained. So as I close up the talk, what's next? Luckily, I have just one link. Heading to shareyourpaper.org is a great starting point for all of these things. And if you have any questions, you can email me, joe@oa.works. And again, just visit our site to get started on your self-archiving journey. So that is, and again, thank you so much for the invitation and for listening. I will stop sharing. And
thanks very much. This was great. I'm sure the audience, if you want to drop some stuff in the chat, we might see a few hands kind of joining in there. But we actually have a few questions also that were not covered in the FAQ, so we have a little bit of time to do that. And so if you can also see these at the bottom of your screen if you hit the Ask a Question box. But just so they're in the recording, it's good to read them out. So the first question that we have for you, which has 15 upvotes, everybody wants to know the answer to this question, is how is Share Your Paper different from a preprint archive or from sharing the accepted version on a preprint archive, like BioArchive or PsycArchive or Archive?
So the main difference between Share Your Paper and a preprint archive, love this question and answers thing, by the way, is that you would generally use a preprint server before you've been published and accepted. So there is absolutely no competition between doing the two things. And in fact, in the law of open access, those two things are actually both called self-archiving. They're both sort of pretty intricately connected. So I probably should have made a distinction between these during the talk, but that's what the Q&A is for. So you can share accepted versions on preprint servers. In some servers, they let you do it. In some servers, they don't. I have a personal opinion one way or another about sort of what's best, but it's not really that important. You go by what the server will allow and accept, and that's probably going to be a pretty great outcome. So this tool I've spoken about here has a bias towards things that are accepted and about to be published, and it's not necessarily about speeding up the publishing process or getting more sort of comments and sort of like peer reviews onto your work before you go to submit that to a journal. But absolutely, sort of, this is not in competition with preprint servers. Some people will do both. They'll get the preprint out there, and then they'll publish the accepted one because that's a more mature manuscript that they want to share, and that will then end up taking precedent in the systems that then say, how do I find a free version of this work? If you have the preprint and the accepted available somewhere, most systems will show you then the accepted one. So hopefully that's clear. I will, the next time I give that talk, I'll make sure that's a question. Yeah. Thanks.
Can I ask a follow-up just to make sure that I understand? So it seems like the advantage of something like Share Your Papers then is that you know that the version that you're getting is like the final version with all the edits and all the revisions and everything. Whereas with a preprint, like good practice is that you upload the final accepted version as a preprint before you kind of do the copy editing and the formatting, but not everybody does that. And so maybe when you're doing a preprint, you might get an older version that's before some of the revisions have been made. And then it's not also necessarily linked. I know BioArchive tries to link to the, now this paper has now been published in, but maybe it doesn't always get that right.
Yeah, no, so I think the other thing that I would say is sort of not everyone makes preprints, and they're not always sort of, you don't always have to do them, and you generally probably wouldn't do them after you've been accepted and the final version's been published. So it's a route where if you didn't quite get to it at the preprint stage, you can circle back and still make your paper available and get that content out there. Even if it's already published and you sort of miss the window for a true preprint. I know that actual usage of these systems varies a little, and some folks circle back and do it anyway, but in general, that's sort of the emphasis of the approaches. Yeah.
Great, thanks. So we've got time for just a couple more questions here. So we'll go to the next one. You did cover why you should use Share Your Paper over, say, academia.edu or research, but this seems like a good opportunity to add anything else that you might want to expand on there.
Yeah, to a certain extent, it's you can do both, and that's okay. It's probably doing better than doing. It's certainly better than doing none. It's probably better than doing just one. If it was me, I would generally have a bias towards, making sure that it's in a repository. ResearchGate and academia.eu aren't repositories. They don't promise to make your work discoverable in Web of Science. They don't promise to preserve your work into the long term, and I mean like hundreds of years, not sort of until the VC funding runs out. I've lost my train of thought. And there's no competition between putting your work onto ResearchGate and academia.eu in a way that is sort of more in line with putting the accepted version out there and things like that. You can still sort of do that, but in a slightly more legal way. That's great too. That's a great nudge if that's one thing you take away from here. I know there's lots of benefits to academia.edu and ResearchGate just in terms of networking and visibility within academia. I'm not here to deny those or say that you shouldn't do those things or that they're really bad and terrible. Just sort of these things have different emphases and sort of usages and yeah. So is that useful?
Yeah, that's helpful. Thanks. Yeah.
Cool.
And I think the point about the kind of permanent repository or the checks to make sure that you're not running afoul of the copyright agreement that you signed, I think those are really important because it's pretty easy to mess that up, actually.
Yeah, it can. Yeah, right. And it's just sort of something you don't want to. There's a lot of things to worry about as a scientist. You don't really want this to be one of them. That's one of the ways I tend to think about it as well. Sure. And sort of the other big emphasis I'll make, right, is that there's a reason why universities, research funders, and societies run repositories to store and sort of help preserve and make discoverable this work. It's not because they didn't find out about ResearchGate and academia.edu. It's because there's sort of a distinct added value to having it in places that are owned by scientists and not sort of there to sort of mimic Facebook, but for academics, right? There's sort of trade-offs that you get there in the long term that can sometimes not go the way that you might hope.
Sure. Yeah, that's really helpful. Thanks. And then before we move on to our next speaker, we have one more question that was for me, actually, which is, if anybody here has ever had a case where they want to reproduce a figure from a paper in a new publication, sometimes it's as easy as just emailing the journal, or maybe you can just like cite the paper, the original paper, and that's fine. Sometimes, though, you have to go through this whole very complicated process of getting and maybe even paying for the right to read the figure. So I'm wondering if you could speak to how, if the figure shows up in the accepted version that then is placed in the repository via shared, how does that interact? This is a very specific question, but at NMA, I also think a lot about figure copyright because we put up all of our content CC BY, and sometimes people read figures from paper. So I'm curious, not just for myself, but for like as a whole, how.
It's a specific question. I'm going to use a get out, which is, I'd need to know more of the specifics. Is this a license for like one time only for an explicit number of views in one location? In which case, I'm not sure that would be okay, and you might need to go back. Is the uncomfortable answer I don't want to give, but might be the case. But it's honestly not a question I've been asked before, and something that I've like looked really deeply into. So my follow-up would be, I'd love to follow up with you over email and see if we can provide some guidance on this that is better than not sure, let me look at the contract sort of things, because I think it's a super good question.
That'd be great. That'd be great. Thanks. And I think that we also move towards, we all put up our talks online and they're all on YouTube. And we kind of start to all inhabit these murky spaces where it's something that you created and you produced, but you published it in this other journal, and now you're wanting to put the talk up on YouTube or make a recording. It's not really clear who or what you're allowed to do. So thanks.
And that's the power of open in the end, right? Is that you don't have to wonder so much. It's clear, and usually the answer is you can do it. Just don't say it was you. Attribute the person who did it, and that's a much nicer system. And the way that I think most of us want to work, no one wants arcane licensing agreements to do sensible things that we all want and need to do day to day for the benefit of our science and for our students.
Sure. And there, yeah, it's kind of a murky space now that everything is digital. It's a lot harder to control things than like a physical print.
Right.
Well, thanks very much, Joe. This was really informative, and we really enjoyed having you. So if there are other further questions for Joe that you guys think of later, and you guys all in the audience, please stick them in that Ask a Question box, and we'll have time for a panel discussion at the end. So thanks again. And our next speaker is Emma Ganley, who is going to talk to us about protocols.io. And before we get started, as Emma sets up her screen, I also have a poll that Emma has asked me to run. And so please click on the polls button at the bottom of your screen. It should be orange and highlighted. And this is just trying to get a sense of who in the audience has heard of protocols.io and whether you've used it before. So while Emma's getting set up, we will please go click that, and then we'll know who we're talking to. Great. Okay, Emma. So Emma is here to talk to us about lots of interesting stuff.
Thank you.
Yes, wonderful. All right, looks great. Go ahead.
Hopefully you see the first slide. So I will not be able to see the answer to the poll, but maybe in a minute you can read it out for me, because I'll have to toggle between windows. But it's helpful to get just a gauge of that before I really start dipping into the details, because it might just let me know how many people have actually come across protocols.io and or used it, or know nothing about it as a tool before I start wittering on. But anyway, thank you so much for the invitation. It's just always really great to be invited to participate in sessions around open science and trying to help people learn more about the ways that you can improve your science. Kind of improving how open it is increases the amount of confidence that people will have and the trust they have. It improves the overall quality and increases the reproducibility of all of the research that you're publishing. So thanks to David and Megan for a great session. Just to give you a little bit of my background, so I'm Emma Ganley. I've been at protocols.io for a little under 2 years now. Prior to that, I worked in scientific publishing for many years. I started out career as a PhD and did a postdoc very much in molecular biology topics. And then I moved into publishing initially at PLOS Biology as an associate editor. So, and then for I think about 18 years. I always used to say over 15, and I realize I'm creeping up towards 20. I've worked in science publishing and with a very common theme around open access to publications, open access to data, sharing information about how the research has been done and the different outputs that are produced and data, and around publishing ethics as well. I don't know if people have done the poll. Is it possible to get the results of that quickly?
Sure. So there's a couple polls that might be relevant here, but the one that you specifically set up is mostly nope, I have not come across it before, and just one yes, but I haven't used it.
Okay,
so,
that's helpful, because it means I know that I will go right back to basics when I present the tool. Before I get on to that though, I just wanted to place the value of method sharing into the bigger context of openness and research data as a whole, and why I think, or we generally think, I think that this is an important piece of achieving truly open and reproducible science. So if we first step back and think about the various outputs that researchers generate, there are actually several different elements that I'm showing here that we can think about as modular outputs that a researcher produces. So there's code and data and citations. And in the middle there, I think, the biggest golden coin that we still have is the actual research article. But method is there as well. So the experimental method. And I want to really talk about capturing that as an important, distinct research data output in and of itself, which I think is often slightly overlooked, but critical part of the research workflow. So it's worth considering that like a bigger question is what the ongoing value of data and results are if we don't know exactly how they were generated. So results and data can only really be meaningful in the context of what was done to collect them, what that scientific method was. You need to know that to ensure that interpretation is accurate and that what you're looking at is reflective of something meaningful. So we also want to reproduce those results. We also need full details of the methods, ideally including equipment, reagents, all of those details in very comprehensive and granular fashion. And this is before you even start to think about pre-registration of methods to reduce all of the risks of various biases that might be introduced through the research process. But, you know, I'm not going to talk about pre-registration, but bear in mind that protocols.io is a tool that can be used for that, and in its equivalent way of really amplifying the importance and value of sharing the methods. And what I was going to say, ideally for open science in this way to really fulfill its promise, we want to know that all of these modular outputs can be captured as fair outputs. So I don't know if you've come across the term fair before, but it's findable, accessible, interoperable, and reusable. And we have really good structures in place to capture a lot of these elements. And I'll talk to you a bit about why protocols.io can help in that way for methods. But first, let's consider why I think this is an important issue right now. Unfortunately, the status quo in publishing these days is that the typical methods written into the materials and method sections of research articles are often not fair in that context of the acronym FAIR, as shown by these tweets. On the left here is a tweet from a biologist, and on the right is a tweet from a physicist. So we see this issue arising across many different research disciplines. It's a pretty universal problem that you go in search of the information in a paper about how exactly experiments were carried out or devices were fabricated or whatever the case may be, and end up on a citation wild goose chase, going from one paper to another earlier paper to another earlier paper, and maybe you hit a paywall, or maybe you just come to a final paper which tells you that devices were fabricated with conventional methods. None of these things are helpful for researchers and results in a lot of frustration. So the GIF on that tweet on the left kind of exemplifies the frustration that researchers feel when they hit against this brick wall, because they actually cannot find the details of the experiments and methods and can't reproduce a lot of the published research. So, you know, Joe's right. It's fantastic to share the paper, even if you've published it in a paywall journal, to make sure that the paper itself is open access and available somewhere is a brilliant thing to do. And I hope everyone is doing that or will do that now they know how. But you also need to make sure that the paper itself actually has all the relevant information in it, or that the relevant information can be found somewhere else. And reliance on in-house notebooks can be patchy. They might have been lost or destroyed. And I know a lot of people are now using electronic lab notebooks, and that's definitely a step up, but this still may not capture all of the details and nuance of a method and how it was optimized. So another great example of this being an issue is the quite well documented cancer biology reproducibility project that was carried out by OSF and partners. The goal of this initiative was to try and reproduce the results and experiments from 50 published cancer research related articles. Unfortunately, when they started this effort, it became very quickly clear that getting the details of the experiments from these papers, either directly from the papers or by approaching the researchers directly, was not trivial. And I'm showing you a quote from a really great write-up of this study from The Atlantic by Ed Yong, recent Pulitzer Prize winner, actually, Ed Yong, about this initiative. He interviewed the main investigators involved, and the consensus overall was that this was an arduous slog, which is a pretty damning statement. And I will just read this quote out. So the hardest part by far was figuring out exactly what the original labs did. Scientific papers come with method sections that theoretically ought to provide recipes for doing the same experiments. But often these recipes are incomplete, missing out important steps, details, or ingredients. In some cases, the recipes aren't described at all. Researchers simply cite an earlier study that used a similar technique. So, the take home here is that repeating others' researchers' work is extremely hard because the details aren't present in the papers. And in actual fact, it proved to be incredibly expensive in terms of time and money, and often the efforts that the researchers trying to replicate the studies put in were not fruitful, and the details of the experimental methods just couldn't be found, couldn't be obtained. So the initiative was halted after they attempted to replicate just 18 of those 50 studies. And this is a pretty damning evidence of the fact that what's going into research papers for what should be very important scientific discoveries is completely insufficient for people to replicate and build upon the research, which is kind of a problem. And so, why else would we want to care about the methods? We definitely want the research to be reproducible. If it can't be replicated and continued and built upon, that use of research funding becomes very questionable. There is a really great quote from an essay written by an evolutionary biologist, Dobzhansky, that nothing in biology makes sense except in the light of evolution. I'd propose an edit to this quote for consideration, which is that nothing in research data makes sense except in the light of how it was generated. So when I first used this in a talk, I noticed in the chat afterwards, somebody had written a comment about how we were drowning in data, but that often there was a drought of information about how it was generated. And I think that captures this perfectly. Ideally, we will see sharing of all research data, and we've seen a lot of effort and movement there and more people sharing the data that go with their research articles. But as we accumulate more and more data, any one data set or method or piece of code is literally one drop in this research data ocean. So unless we understand how the data have been generated, that critical research output has to be captured alongside the data. If we don't know how it was generated, it doesn't become very useful in the long term. So I think, again, if we step back and think about those modules of research data that a typical researcher is generating, and that will ideally be captured as standard practice for open science, it's really important that we try and think of a way for researchers to be able to capture some of this information while you're performing the research, rather than having to be requested to do another additional task after the fact. So we're looking for processes that can be embedded in your daily work research cycle, basically. And it does make sense to look at tools and platforms that already exist that can be easily accessed and implemented. And they are maybe easy to use and free, hopefully. And if we can get this happening as kind of standard research practice, that would be a huge win for reproducible open science. So protocols.io, this is where this comes in. It's a way of sharing your methods. It's a platform online, freely available to use for method development, optimization, sharing, and publication. And it does already broadly adhere to those FAIR principles that I mentioned before. Our mission as a company is very simple: to make it easy to share method details before, during, and after publication. So it's an open access repository. It's free to use and it's free to publish your methods. We do have a business model which functions very much like GitHub, so users can pay for additional private space, but I'm not going to go into detail on that here. The main thing you need to know is that without paying, absolutely anyone can add and publish their methods on protocols.io. It is purpose-built, I'll show you in a minute, to capture all details of a method in a step-by-step recipe-like format. So you can add tips and tricks, details of the ingredients, materials and equipment, everything that's being used. You can add and make the methods easier to use by including images or videos for individual steps. So you can really demonstrate how to do something. Basically, you can add a ton more information on protocols.io with context and explanation than you would ever be allowed to include in a materials and methods section of a research article. And we do all the kind of archiving and mirroring, so the content's all very safe and secure in the same way it would be if you went to a journal and published an article. And when you publish, you get a DOI, a digital object identifier, and everything published is under CC BY, so it's all fully open access. So the DOI is I'm not sure if people are familiar, it's a permanent identifier. Every research paper you publish also receives a DOI, and it's essentially a permanent identifier for that static published version of the paper or the data set, or in this instance, the method as you put it on. And what's really brilliant on protocols.io is you can create a new version of a protocol after you've published it. And each additional version will receive a new DOI, but they will always be linked to the earlier versions. And if you happen to land on an earlier version, you will be told that there's a newer version. So you get this dynamic permanence and a linking through. So they're versionable, they're all indexed and searched, they're archived and mirrored, and it's a really dynamic tool. I'm going to play around and show you if my internet permits in a minute, so you can have a play around. But if you want to look offline later, www.protocols.io/welcome is a great place to start. Please feel free to have a look and contact us if you have more questions. So here's one example of a protocol, MRI processing on the left, and you probably can't see much here, but it just gives you a quick hint. I will try and show you some in more detail in a minute. And protocols can be arranged within workspaces, and they can be either private or public. You can control who joins that workspace and essentially use it as a location for collaboration. Before you have published a protocol, or when you're editing any protocol, you can collaboratively share that with colleagues, peers, and even collaboratively edit it, but you have full control of permissions that you would give to other people. And then the workspaces can either be private or public. So this is a public one for the Allen Institute for Brain Sciences. And I won't go into those details there. What I'm going to do is try and jump to a bit of a demo so you can see a few features live. I've preloaded some pages to kind of hopefully make that a bit easier. So hopefully now you see our file manager. So I am in the file manager space for a workspace that we set up early 2020 for fairly obvious reasons. So this was the Coronavirus Methods Development Community. This is open for absolutely anyone to join and share content within and discuss content with colleagues and other peers who are in there. On the left-hand side, this is protocols side. Each icon is which workspace you have. So the top one here is this coronavirus one. And I can toggle through from this folder with the files to the profile, which is a slightly cleaner kind of public view that you would see of the community. And you can see that very organically, this group grew. It has 547 members, so we can have a look who's in there if we wanted to scroll through all the pages. There's a location to have discussions, and that they can pertain to specific protocols in there, or you can have larger discussions. And so as a big kind of win for open science, discussions we had in here included things like how to undertake testing or sequencing in far-flung locations like Antigua, where they did not have the same resources as better equipped locations. So it was fantastic to see this really taking off. And then there are 253 protocol publications in here. So if we go in, they're all in one folder, but there are categories. So you can kind of help organize content by adding categories. So if I click through to the sequencing detection, and then we can sort as well on things like views and other things. So if I've sorted on views, the top viewed one here is a sequencing protocol. So it also turns out that this was the first coronavirus protocol that was shared on protocols.io. So I've also preloaded that just to save a bit of time. And there are 3 versions of this protocol now on protocols.io. If we click on the left, you can see the dates when they were added. So the first version was actually added, I think, before, general global awareness had really kicked in that this was as big of a problem as it has turned out to be. So right in January 2020. What's really amazing about this, because this is a very long and complex protocol, it's saying it's loaded, it loads on the fly. It's, hundreds of steps long, is how quickly they were able to get it up. And if we look at the top, you can see that it tells you version 1 is forked from Ebola virus sequencing protocol. So this author, Josh Quick, belongs to an effort that was funded by Wellcome, Arctic, for responses, fast responses for kind of epidemic, pandemic global disaster scenarios, essentially. And it happened that they had published a protocol on Ebola virus sequencing in 2019. And they were able to quickly make a new fork of that and change that kind of as a template so that it then worked for SARS-CoV-2. We can see between versions or between previous instances like that Ebola one, we can do a comparison to see how the authors made those changes. Hopefully this will load. So on the right is that original Ebola protocol. On the left is the coronavirus one. And the changes are highlighted. So you can see there were different quantities of template RNA that go in depending on the virus. Marston-Mix issues specific details about the primers for each of these things. But in large part, there is a lot of similarity between these protocols. So it's a huge win for open science to be able to so quickly put up a new sequencing protocol. And that's why this has been seen and used as much as it has over the last year and a half already. I will just highlight a few other features on here. So on the protocol itself, we can see this button is, I love this button, works for me. If we really want to know that people are able to replicate and reproduce an experiment, this is probably the best indicator you might ever find. 34 other researchers have clicked this button because they've managed to replicate and reproduce this protocol. And you can track who those people are. If you think they're all just authors or Josh's colleagues at the University of Birmingham, you can actually go in and see. It's also really easy to add comments on a protocol. So this one has 99 comments on the protocol itself. And you can go in and people are asking questions of the authors. And they've been amazing at responding to these questions about how to run the protocol, what to do, whether you can do variations, et cetera, et cetera. And so you get comments on the protocol level at the high level, but you also get them on individual steps. So you can kind of go through and see, and what you end up with, I have access to private ones, which you probably don't. So when you add a question, you can make it be public or just go to the authors. You essentially end up with a public FAQ. And what's brilliant is if multiple researchers have the same question, the authors don't end up answering it multiple times by email because it's all here and it's all publicly available. So those are some of the features I was going to show. Finally, before I move off this bit, is the ability to run. So when there are protocols on the system, anyone can come in and run and make a record of that run. So if you're following someone else's protocol, or if you're using it in your own lab, you can go in and step through the entire protocol and track your progress as you go through. I'm not sure why that's not working on here. And I'm trying to show you, but because I've not clicked on it. You can step through every step and it will show you your progress at the top. If I went back to this one, when there are timers in a step, you can do the timer in the actual system dynamically and really make best use of this. If you change something or something strange happens, you can add a note to that step. And then at the very end, you can save the entire run and you'll have a record of everything that happened, or if something went wrong, or you dropped a test tube and you had to kind of tweak what you were doing or whatever might have happened during that protocol. You can actually capture all of the information and track the whole thing in the system. Okay, and then just to illustrate how easy it is to go in and actually make a new protocol, this is just my kind of test scratch space that I have. When you're in the file manager here, you can just literally click new protocol. You can choose where you're going to pop that. And you will get some options, hopefully, that will allow you to just choose kind of basic template. And nothing here is irreversible. So it doesn't, you're not shoehorning yourself into one thing that can't be changed. What you can do though is pick collection of methods. If you know that you have 3 key methods for a paper that you want to present, you can use collection of methods. But so say we're going to go here with biology protocol. The only thing that really changes are the options of these components that you see on the right hand side. But you can switch between them. So if you're going to want some of these computational ones too. you can switch to this, but you can also favorite specific things and then view your favorited ones as well. And those components allow you to add things very easily. So heat to 100 C, say. And if we turn that to the component, it will give you the temperature, for 10 minutes. And we can add a duration. And if we add a duration, it will start to add an overall duration for the entire protocol. So you can kind of play around with all of these things. You can bed in citations, links to other protocols, and so on. You can add sections and they become color coded, add another new section. And so that might be 2, and sub-steps and so on. And it's pretty intuitive if you come in and you play around with this as a tool. As you move through, you can switch between editing and viewing. So you can really easily kind of see how this would look if you published it by looking at the view. And you can get to the same options from the edit mode, but you can also see here how easy it is to get to the publish flow for a protocol. So you can click publish, which will take you through, and it will fully publish and assign a DOI. You have to go through a few steps. You can use get DOI if you want to obtain a DOI for the protocol to put into a manuscript, but not have it fully otherwise be indexed. So you can use that for reviewers and editors and so on, which is a great way to kind of make it available if you want to share, but you're not ready to fully share it as you're waiting to publish a paper, for example. Kind of do open science drafts, or you can just do sharing ahead of time, which works very much like sharing a Google Doc. You can control all the permissions of who you want to share with and so on. So there are tons of options of things you can do. I could spend ages showing you other things. I'm going to jump back to the slides. I think I've got about 5 more minutes, so I'll try and whistle through a few final things before wrapping up. So it's all great. You can add your methods to protocols.io and share them publicly and get a DOI. What next? What's the way? How does this fit in with everything else? That kind of interoperable thing and having a digital object identify a link means you can literally drop the DOI into the materials and methods of your manuscript when you're publishing it. And if people clicked that DOI link, they will be taken to protocols.io. I mentioned it's possible to add new versions. So this protocol collection has been updated since the authors published that paper. So hopefully you can read that, but you're told straight away that there's a newer version of that collection available and you can view the newer version. And with that, the comparison tool, you can always check to see what's changed and being updated. But I like to call this dynamic permanence because it gives you as researchers control over updating the methods without necessarily needing to go back to the journal, the publication, if you need to correct something. And it's fully transparent what things get changed along the way. That can be a really valuable thing for just adding some extra clarifications to the method, or correcting something if there is just a typo in there somewhere, a numerical error, etc. We have recently, I'm not going to run through that video actually, we've recently launched a new partnership with PLOS ONE. They've created a new article type, Lab Protocols, and that is the paper and the protocol on protocols.io. So I can quickly jump. This is the first one that was published, and there's a link at the top, very, very clear and easy to see, that will link you straight to the protocol on protocols.io. And if I jump down to the methods, you can just see the materials and methods notes that it's a protocol published on protocols.io. And they include it as a supporting, a PDF of the protocols added as an SI file. So it's there in a Figshare widget as well on PLOS's website, but you also can just jump straight to the protocol on protocols.io, and there's lovely bidirectional linking. If this one loads, you can straight away see that the paper was published in PLOS ONE and that it's a peer-reviewed method as opposed to just something that's being posted, self-posted essentially on protocols.io. So that's a pretty recent thing that we're really keen for people to explore and play around with. But that passive, just putting a DOI into your materials and methods, is actually supported by over 500 journals and publishers who are very happy for researchers to include methods details via linking to an external tool like protocols.io. Oh, that's going to keep going, and I don't want it to. There we go. One of the really cool things about that first lab protocol, I will try and tell you very briefly, is that the first author, actually sole author of that paper, was an early career researcher, and he tweeted how thrilled he was with that publication, as he's now able to get credit for the method in a way that he may not have got just from the research article of the application of the method. So we start being able to properly credit the method, creation of the method, as an important research output on its own, which I think is really valuable, because it was his first author paper, it was his first solo author paper, and it really does give him that recognition. Another very cool thing about this first example is that the method used a piece of proprietary software for imaging, and immediately it was published. I'm not sure if you can see this whole tweet thread, but I'll share the slides afterwards. Another protocols.io user forked a new version, and he is adapting it to illustrate how you can do the same method but using an openly available piece of equivalent software. And because of the whole CC BY and everything, what he's nicely done in the privately shared version of this new version is give that attributed reuse to the original protocol that was there. And very quick example of another way that sharing the methods independently can enhance open science is this series of tweets. So on the left-hand side, this was a researcher from Chile who was looking for someone experienced doing RNA extraction from primary cortical neuron cultures. After a few tweets, the postdoc, Elena, on the left-hand side, postdoc from UCSF, pointed to a protocol that was on protocols.io as a method for this. And when we looked more closely, it turned out that that protocol was linked with a paper that had been published in GigaScience from researchers who were performing investigations into stickleback parasites, so a fish parasite. So I love this example because I think it's really, really unlikely that the researchers working on primary cortical neuron cultures would have come across the paper on fish parasites as a way to find a method that might work for them. But that is exactly what happened here. So by, I think, separating method from application is a really powerful thing to do to enhance open science. I realize I'm running out. I've probably gone over slightly. We have lots of other organizations and funders and institutions who support protocols.io. So if people are worried that using a tool like this might not be approved, actually most people are very aware and very supportive and encourage researchers to share this way. And I will skip over that one, but by using it, there are a ton of benefits and you can be a part of a really vibrant open research community. The team is quite small. We really are a startup, but we're very responsive. You can book demos really easy, sign up for an account for free. So very much encourage people to play around and contact us if you have any questions down the line. And I think that was it from me. So thank you very much.
Thank you. Thank you very, very much. There was a lot of activity in the chat, some of it also from me. So very, very cool stuff. And I think that it's very clear to everybody here that how powerful this framework, this platform is. And I think especially what you said at the very end, this idea of separating method from application and discovery of methods that are applicable to your own science that you never would have seen in your own little corner of the field. This is so important with the proliferation of data and the proliferation of methods. Just how do you separate the signal from the noise for what is relevant to you? And it seems like this is a really powerful tool to do that. So thank you so much for taking us through and sharing this with us.
Pleasure.
We've got some questions for you, and I think we'll take just a few of them here and then save maybe some of the rest of them for the panel discussion. We've already answered the one about journals accepting protocols.io as a place to put the details of the method. So that's awesome. And so I think the most upvoted question here is how or does protocols.io collaborate or interface with other initiatives such as Neurodata Without Borders, brain imaging data structures, and so on?
Right. So for the specific examples that you've listed there, I have to confess, I'm not 100% sure if we have any specific ties, but there are so many groups that kind of come to us, and also they don't necessarily need to talk to us to let us know if they're setting up a workspace and just adding people. We don't necessarily see all of that activity because it is just kind of come and use it if you want to. But there is also an open API. It's got really great developer documentation. Not that I'm a developer, but this is kind of the feedback that we have generally, is that it's really good. So if we do have people who want to integrate much more comprehensively rather than just having a group who's working on the system, then our developers are always really responsive and happy to work with people and give feedback. I will check for those two specific groups and let you know if there's anything more specific, because off the top of my head, I can't answer it specifically.
Great, thank you. And I think that after this session, you'll probably have a flood of people who are interested in neurodata coming to
Absolutely.
IO. So that'll be really informative. Thanks.
Great.
And maybe just very quickly, I'm going to skip my own question because I think we can deal with that later. Could you quickly speak a little bit to the utility of this tool for not just biology and chemistry experiments where you need those super nitty-gritty little details, and neuroimaging experiments? I think everyone understands why you would need something like this, but
yeah,
maybe just for behavioral experiments where the code for generating the paradigm and the data itself are already up on GitHub. Where do you see the utility of protocols.io fitting into that sphere?
Yeah, that's a great question. And obviously, you know, the idea isn't to kind of be a replacement for GitHub, but I mean, people who are familiar will see that it works in a very similar way with the whole forking and everything. If you have a really complicated pipeline that's kind of, you know, run this piece of code and then transform the data and then run this other piece of code, then the protocol in protocols.io can actually help you set out the pipeline for the full kind of flow that you need a user to follow, or that you have followed if you want to track that for your own purposes internally. You can bed pieces of code in there, but you know, if there's big code, it's probably better on GitHub. Absolutely agreed. But if you want to kind of put key morsels or snippets, you can put them in. There's a component for code, so kind of pull it out, you can see, but you can't run it on there. So, you know, CodeOcean is another location where you might want to play around with kind of executable code. So there are a bunch of different tools. They don't all serve the same purpose, but I think there's times when protocols.io would be useful in that context, but it's not to supersede GitHub, obviously.
Sounds like this is kind of a super README file then.
Yeah.
Where it's, you know, you would have a README on GitHub that's like, run this, then click here. This is what this function does. But this is like, how do you put it together? How do you fork the actual like experimental paradigm and analysis pipelines? Okay.
Absolutely.
Great.
And if you want like best practice for as you run this, put the data there and make sure you name the file that way. Like you can put all of that into your protocol too. So you can kind of try and make a conformed, proper process to ensure that you're all naming the same way, for example, as something trivial, but that isn't always easy to achieve. So
very cool. Well, thank you so much for this really informative presentation. I think everyone really enjoyed this. And so if we have any more questions for Emma about protocols.io, please put them in the Q&A and we'll get to them in the panel discussion at the end. But I think in the interest of time, we will just scoot right on to our next speaker. I know this is a long session, so please feel free to wander off and use the bathroom if you need to very quickly. But we'll go right to our next speaker then, which is Nikki. And Nikki, are you here? Yeah, great, cool. So Nikki is going to tell us about Open Science Framework.
Yes, can you hear me okay?
Yeah.
Okay, great. Yeah, thank you so much for having me. I really enjoyed the speakers that have spoken already, just learning so much about the tools in the space. We're all aimed at the same goals, but take different parts of the workflow or different aspects as our focus, which I think is really fantastic. So I'm going to get my screen shared. So bear with me one second.
Yeah, looks great.
Okay, I'm going to put it in presentation mode, and hopefully that doesn't break all the things.
Beautiful.
All right. So first, just thanks again for letting me join this discussion. I'm Nikki Pfeiffer. I'm the Chief Product Officer at the Center for Open Science. I've actually been there for going on 7 years, so been there since the very beginning. We're still sort of a startup, but kind of moving into maturity. And as an organization, we're a nonprofit with a mission, and our mission is to increase the openness, integrity, and reproducibility of research. But that's not something we can do alone. This is something that we are looking and constantly engaging in partnerships with stakeholders across the research community. And we advance that mission through several different sort of mechanisms, through infrastructure, methods, community norms, sort of advancing our incentives and policies to support sort of that accelerated discovery of research and the transparency that we're all looking for with open science. So again, the organization is divided into sort of 3 major pillars to support that effort. We work with funders and institutions to help develop new policies to sort of enhance the incentives and embrace the change that this would mean. We also have an arm that does our own research, meta research, and it was just mentioned the reproducibility project, Cancer Biology, is one that we in-house have been leading. And this is to gather the evidence of change. So as Emma was speaking, and she did a fabulous job talking about this project and what it is finding when we try to do replications of some of these studies, the real challenges that we face because of decisions made by researchers early in their process. So that's something that really helps us understand it and encourage the change through this evidence. And then lastly, it's the infrastructure. So the technology that enables all of us to kind of go and implement these practices in our daily workflows. The reason why we have these different pillars is because we take this entire thing as a culture change, this entire movement as a culture change, which I know everybody's talked about, but the reason that we've sort of stacked these pieces together as our theory of change is to make it possible with the technology to really provide UX design and UI design into making it easy in these workflows to work with communities. In fact, our roadmap is largely informed by community needs, stakeholder needs, to make these behaviors more normative. And as I said before, working with different stakeholders to make it rewarding and make it required. So when I talk about the infrastructure focus, which is the piece that I came to speak more about today, we're really narrowing in on making it possible and then making it easy, enabling these open behaviors to advance science. And so we think about 3 major pieces to that: openness, transparency, and rigor. And I just want to call that out just because those words are getting tossed around a lot, but to really kind of narrow down what they could imply. With openness, we talk about data sharing and we talk about data reuse, but really having that data that's not reproducible is a real risk for us as a scientific community, because what's the value if you can't reproduce those findings? And so what does that mean? There's a lot of transparency within that data that has to be there to enable that behavior. And that includes providing the null findings and the things that didn't pan out, because we can all learn from that. And not to see that as a negative result, negative meaning your name is now associated with something negative, but actually as a gain for all of us as a scientific community, and to really build in that rigor around the data and the findings that we have. So I guess one thing that I like to sort of pose as a question is, if we have infrastructure, we've heard from several folks already that are talking about the infrastructure being available to share and reuse data. And we have lots of data out there. And we have policies that are sort of starting to say, well, you have to share your data. We know funders are starting to recommend or strongly encourage or even require this. Why don't we have tons of open, reproducible data? It's because we're focused on the wrong end of the workflow. We're thinking about the very end when it's like all years of work, all of this has gone into it. And now it's time to share it out there. And we stumble at this stage because it really has to start from the very beginning. And so, you know, I think incentives are tied into a little bit of that, where our success is focused on getting it published, not necessarily on getting it right. And that's, again, focusing on the beginning stages of our research and all the work that really goes into the planning, not to be rushed, right? And to start with the end in mind. If you're thinking about sharing your work from the very beginning, then you might make different decisions. You might organize your files and your naming convention and your code books and all the aspects of your analysis plan and your protocol. You might spend the time to really polish that work and organize it in a fantastic way that helps your collaborators, but also helps your future self. So a lot of times I think it really is, you know, helping future Nikki or future Megan or whoever to try to build that into their daily workflows and daily habits. I know that sounds great, and I think we can all like nod, like, yeah, I sort of agree that that is something I should do, but I'm not going to be naive in thinking that that there aren't obstacles that we all face. And so I think thinking through those and trying to address those in the infrastructure design and the workflows that we implement is important. So not saying that these are separate things, they're actually very related. And the incentives being misaligned is a very important one. And I think there's a lot of effort going on right now within the stakeholder community for research to try to support that. One being the burden of time that this takes. And so building into your grant proposals and your budgets, really the time to share, you know, the time that it takes to package up and curate data and to share this at the end. Usually we run out of money, right? Like you get to the end of your project, this is the last check, and then you can move on, but this actually takes the most time. And so thinking about doing this from the very beginning and where the tools might be supporting parts of it, but have gaps. If I have interoperability between the tools in my workflow, then I don't have trouble when I try to move things across. But sometimes that's not the case. And so calling those things out to the tool providers and the creators of these tools to make sure that they're aware of it and they can come up with solutions, because sometimes it may just not be something anybody's spoken up about yet. And I think not knowing where to start is a challenge. And so hopefully today you're going to see some things that'll help you get started. But definitely reach out. I think we've all offered email addresses and support email addresses so that if there are questions, just know that you're not alone. This is something everybody is at some point in the spectrum trying to figure out and how to pick up these best practices. And that's sort of the other thing I wanted to really hone in on, is that it's not an all or nothing. There is a spectrum of implementing these best practices, and we're going to talk about lots of them today. I'm going to talk about one of the ones that I think is on the more rigorous end of pre-registration, but there's lots of steps that get you that kind of build on that and get you there. Thinking about just defining metadata, getting your ORCID, putting DOIs on the different pieces of your research as they come to be. Even if you're not ready to share them right away, you can still get DOIs or plan the metadata to support the DOI when it's time to get that registered. Thinking about using, the basic practices of open data, planning ahead with your data management plan, writing a data dictionary, putting it in a system that actually tracks your version history instead of in your inbox or on your local drive where, they just overwrite and it's really hard to pull that out after time. And thinking about all the open materials that are supporting your research. So yes, you have data, you have the final data set, but there's so much else that you're using in that workflow. And so as you saw, protocols.io is a great tool to publish a protocol and to store that in a way that at some point you can publish it and make it discoverable, and even linking the things together across these different systems. And pre-registration, which again, we'll get into, that's really what I was supposed to focus on, but I'm talking about lots of things around your pre-analysis plan, the study design that you're using, your hypothesis and variables. And at the crux, that's really all it is. It sounds like a big word, and I know sometimes it seems overwhelming, but it isn't all that difficult to sort of articulate these things at the very beginning. And then obviously we talked at the beginning with Joe talking about sharing open access versions of your paper, or even using a preprint to provide some early dissemination of your research and to open it up for some review among colleagues is a great way to build rigor into your study and even to how you sort of report out. So OSF is a tool for researchers, and it has a lot of functionality, and I'm going to talk quickly about it, but I certainly wanted to mention it as something that should be sort of in this toolkit for researchers in ways as an option for how you collaborate and manage files and sort of keep that transparency and openness at the forefront. And it helps you build in from the very beginning some of those things you'll need later that you may take for granted. And it just kind of does it for you with ease. A lot like Protocols.io was just sharing some of their steps. So at this point, I want to jump off of slides and move over to a more active, engaged sort of tour of OSF and looking at pre-registration. But I just want to call out a few key things to be thinking about if you don't take away much else, which is, support for the entire research life cycle is what the OSF is meant to support. It's not the only tool, and because we're sort of mission-driven nonprofit, all we care about is that people are doing these behaviors. And if you find a tool that works better, I think that's fantastic. We are open source, so not a tool that at some point will lock you in or your data, or is going to leverage that for some other sneaky thing down the road. It also has a public API, and we're constantly working towards how to leverage that for integrations with other tools. In fact, Protocols.io recently built an integration with OSF, meaning you can share your protocols into OSF as well. So I think these are fantastic ways to, again, just build that ease of use across the different tools that researchers need. So looking at how you can manage your project, manage your collaborations, put together a pre-registration and publish that, manage all the different files and outputs, share them when you're ready, and look at how it can support some of the requirements that you might have that you're trying to keep track of as you move through your project as well. So talking specifically about pre-registration, because I know this is a concept that not everyone is familiar with. Maybe you've heard about it loosely, but aren't really sure what it means or why you should do it or how it will fit into the things that you're working on in your research. So essentially, like I said, pre-registration is specifying your research plan in advance of your study and submitting it to a registry. It's really that simple. And what it does provide is it will increase the credibility of your research by sort of staking the claim to your ideas at the very beginning, what your plans are. You actually protect yourself from being scooped down the road. You have a time-stamped, immutable version of what your study is about that you can always point to if somebody else comes out later. No, I actually kind of claimed that early on. And if you are worried about some of those things, you can embargo your registration. You still get that original timestamp, but you can keep it private until it gets closer to the time that you're going to be publishing all the data and findings at once. And that's something that's offered. But it just helps you plan the more rigorous, reproducible research that I think we're all after. It asks you questions that you kind of may gloss over or think, oh yeah, I have that figured out. It's gonna sort of walk you through what those important pieces of your research are that you want to nail down so that it is reproducible for others or for yourself the next time you have a question or you want to go back and analyze that data. It also is important because it separates the exploratory from the confirmatory testing. You can't really mix the two together. The initial data that helps to find the research question can't be the same data that you're testing to answer the question. And so I think this is important. It's not to say that you can't do both. You can't have exploratory testing and confirmatory testing. In fact, I would expect everyone actually does that, but you can separate that out very clearly in your research process, and that way you don't have the risk of biasing your own thinking and your own study design, and therefore reduces, sort of bias in publication or anything else down the road. So these are all great things that really raise the bar as far as openness, transparency, rigor, and reproducibility. And that's why pre-registration is such a beneficial step to take in your research process. So I am going to jump off. Like I said, I've got this project. Whoops, that's not what I meant to do. And if I can, I will quickly grab that link and drop it in the chat in case anyone wants to take a look at it. Let's see, is this working? It is working. Okay. So here is a public project on OSF, which I just dropped the URL to. And so a few things that I just want to call out. I don't have enough time to go give you a full tour of the OSF, but some key features that you may be needing to sort of solve for in your workflows. The project that I created, I actually made from a fork because I gave this professional development talk last year, slightly different one with Mark at Cedar about metadata, but I just forked the same one. And so you can actually see that I forked that and it links back to the original. Similar to protocols.io, I think it is a great tool for being able to build on research and to be able to trace back to some different branches in that workflow. But the project is going to be private by default. And at the very beginning, I can choose who I want to add as my collaborators. And so I can add lots of members of my team. I can search for them if I know their last name. If I have their email address, I can also put that in. I can add them, and I can decide what level of permission or access to give them to the work. So, and it spells out for you what administrators can do versus read-write versus read, and if I want them to be bibliographic in the citation. And I can determine which of the components in this nested structure that I had already built to add them to. You may not have that from the beginning, and that's fine. But over time, when you create new components and add them into your project to organize your data, your analysis, as you do that, you can actually you can set different contributors on each of those components and give them different permissions. And I can also make each of my components private or public. There is no all of it or nothing. This is meant to support sort of those different transitions in the workflow and the different access levels that you'll need. Each of these has lots of metadata. You can create a DOI. You can put a description and license this so that it can be reusable if you do want to share files and content. The other thing about the OSF is that it's supported by many integrations, which means if I keep most of my lab work over in GitLab or OneDrive or whatever tool you're using, you can directly integrate with that tool and not have to move your files around. It will keep track of whatever folder you're pointing to, whatever root directory you say is the one that I want to include. Then that is the one that will be in the files of your project. And that also means that if I make this project public, I don't have to give everybody my Bitbucket login information. By adding it to this project, I make all those files accessible for people to read and download without having to work through all the sharing and credentialing that goes through that. You just do it one time and it's there in your project, which is a lot easier. The other functionality I just wanted to share quickly was around the wiki. So in this wiki, I actually used it to kind of give some overview of myself, to give you a very extensive overview of the different features within OSF and some links to different important details. And then to talk about pre-registration here as well. So some of the FAQs that we might come to at some point in this conversation, I've gone ahead and tried to provide them here. And this is a very useful tool for collaborating. The other thing too about files is that all of the files have version history, which means that over time, if I go and make updates to this, it's all captured here, so that if I ever have a question or somebody asks me about where this started and how I got here, I can help remind myself. And that's important, I think, when you look at having transparency. But for this, we're going to talk about creating pre-registrations in the OSF. And so one way to do that is from a project. The other way to do it is actually going to the registry's discovery sort of location and adding a new registration. And you can do this without an existing project. So what it will do is create that project space for you as you build the registration, so that down the road, when you do want to add additional files as you work through sort of analyze data and different steps in your workflow, you can add them to the public project or private project and keep continuing that work. But for this, I've gone ahead and started a new registration. So if you go in here, there are several different templates, which is a really important thing to call out. And we're continuously adding more. We actually just added 2 new ones in the last few months, one for qualitative study pre-registration and one for secondary data pre-registration, because we have a great standard for the typical pre-registration with OSF pre-registration. It's the most rigorous template that we offer, but doesn't always work for these other types of studies that people are conducting. And so another that we are working with communities to develop, for example, is longitudinal. Longitudinal study is slightly different, and there are certain things that you want to call out and mention as part of your study design before you begin to collect that data. And so that's something that we're working on, as well as systematic reviews. Those are slightly different steps in the process that we want to articulate. So for this, I'm just going to look at the basic OSF pre-registration, and when it starts, it immediately will copy in the metadata from your project, but you can also enter that the first time around, adding your collaborators. If you're affiliated with an institution, you can add that, you can license it, you can add certain subject areas, and even put tags to help for sort of discoverability. But the real meat is in the different questions that this template is going to ask you to respond to. So thinking about your hypothesis. And that's essentially what is the research question that you're going to look into and that you plan to study and investigate. And we even give you some examples, but I think just articulating what the question is really all that's needed, because further it's going to ask you more and more. So talking about your study design plan, figuring out, is this an experimental plan, observational, a meta-analysis, or something different? So just at a high level, it will sort of impact the way that you answer some of these questions. Will you have blinding within your study, or if there is additional blinding, explaining that. And then describing your study design. A lot of times that is something you can do in a few sentences, but it may be something that you actually have articulated with your whole lab and other collaborators. Maybe it's a multi-lab collaboration, and you've defined that very well somewhere else in another file. And so you can very easily upload that file to your project and select that as the response to the question. And then talking more about, will randomization be involved? These are all things you can specify. You can write them in the text fields, but you can also upload files that may have more articulation if it's very detailed. And then in the sampling, we like to know, do you have existing data, and at what where are you in that data collection process? So looking at answering any explanation of existing data, what your data collection procedures will be. Again, this may be something that you want to upload a file or protocol for. Your sample size reasoning, your stopping rule, what your variables will be. Again, this may be something that's very detailed and you have this huge striation that you need to put together and put that up as an upload. But it may also be something that you can define within this text space. Measured variables, indices, and then lastly, it'll ask you for your analysis plan and what statistical model you plan to do. Again, you may actually have that in code somewhere that you want to upload or connect to through an integration into the OSF, and that is certainly possible here, which makes this a little easier to start to pull out the different responses to these, and talking about the specific exploratory analysis like we were talking about, separating that out and calling it out and defining what that looks like. So moving through this process, answering all the questions, you can review it and then finally submit it when you're ready. This is asking if I want to include the other components that I have. Right now, I might have my exploratory data in here. Maybe I want to include that as part of what I'm registering. Maybe I don't, and that's up to you. You can also decide if you want to make this public immediately or enter an embargo, and it will create a DOI when it is registered. This is it takes some time for the system to archive, as well as it's going to email all of the other admins that you've added to this registration to make sure that they approve if they agree with what you've put out as a study. The other thing I didn't show, and I'm regretting it, but I can't go back now, is that I can actually save that registration as I'm working on it. I don't have to finish it all at once. Those are a lot of big questions that you may not be able to just type an answer in right away, that it may take some time to work through. And when it's in a draft state, multiple people can log in and access it. So if you add, Megan and I, we could work on this together simultaneously. And I might have sections that I'm answering and working through that maybe she has other parts that she's leading. But all of that to say that, the registration will show up. The drafts are accessible here. And then, back to sort of the goals of why you would take this on. I think overall, hopefully that sort of walked through the steps that you would take and how this would support you defining these important steps at the very beginning of the process for planning your research. So that's all I had on that really quick tour and deep dive into pre-registration. Certainly, if there are questions, happy to answer them now in the chat, but also happy to be followed up with. My email is nikki@cos.io. Happy to hear from you.
Awesome. Thank you, Nikki. This is great. Yeah, we've got one question, it looks like, in the Q&A related to this, but then as a reminder, everybody, if you have questions for Nikki later, if you continue playing with the demo page that she set up in OSF, and then you have questions later, then we can do that in the panel discussion. And I think that you've started answering Eduardo's question here, which is about how to reconcile the need to make science open from the very beginning, and also balance that with concerns about authorship, novelty at the time of publication. You did mention this idea that you can embargo it and those kinds of things, but maybe you could expand just a little bit in a couple minutes on how you're viewing those two almost warring, motivations.
Yeah, I know we talk about it at a high level, and so it's not something that it gets really detailedly thought through. But I do agree that it is a challenge to think about putting my work out there before I'm finished, right? Like, I'm still working on this. And so having it be open for everyone to see and to potentially, take some of the things that I'm working on and run off with them and do them as well in competition with me is troubling. But I do think the couple things to just reiterate here. One, you can embargo it. So if that is something, this is your first time and you aren't sure how that's going to go, I think doing an embargo might be the best way to feel secure in that. But I also think that if you move past that step and you do share it publicly, realize you do have your name and your ideas timestamped publicly in a way that, like, these don't just come down. If somebody says, I want to take down my registration, there's a process. You actually have to withdraw it, and it doesn't remove all the metadata, and it requires a justification. So I think that it's not going anywhere if you publish that. And, you know, something else that I didn't really dig into too much, but it is worth mentioning, is that all these things that get DOIs are part of that larger PID graph, right? Which means that you are able to kind of point to, at that final publication stage, the pre-registration step that you took and when you took it. So that may actually put your work at a higher level when compared to another published finding that didn't take this step. Seeing that you were transparent from the beginning, that you know, involved rigor, and even got feedback potentially in that workflow. I think that really makes it seem much more valuable in the grand scheme of things if you take those steps. So I don't know if that answers the question. It is a tough one, but I would say do it incrementally, open yourself up sort of as you feel comfortable and as you sort of build experience with working through these different steps in the planning process.
I think that's really helpful. Thanks. So I think we will move on to our last speaker now. Thank you so much, Nikki. This was really helpful. And so we'll move on to our last speaker now. And then at the end, we are going to have time for any lingering questions and kind of a panel discussion as well. So Lokia, are you here? Would you like to come on camera and share your screen? Shall I unmute you? I can do that. There you go.
Oh, okay. I look pink. Oh, that's brilliant. So it's 8 p.m. here. Hi from Cardiff.
Hi. Thank you for joining us.
Oh, I disappeared. How can I share my screen?
If you hover over yourself with your mouse, you get a little arrow button. There we go. Perfect. Okay.
Well, can I first make a comment about the previous talk, because I in my lab we include pre-registrations on the CV. So I think for early careers and PhD students, it can be quite useful to show that you have this research output. It's just not in that final stage, but you have something to work for it. So I think it's quite useful for showing your work, basically.
I think that's a really good point. Okay.
Hopefully not many people will be asleep at the end of this talk. I'll try to be not take too long. So my name is Lucia. I work in Kubrick at Cardiff University in Wales. I started in psychology and doing research on eating behaviors. But after doing a lot of open science myself, I found an interest in meta research. So that's where I'm moving to now. And today I'm going to give you a primer on register reports. For the 5 people who have done a register report, I apologize because you probably know already all of this. Okay, so let's start with the paradox. So the research process is quite lengthy and it requires a lot of effort and time. You start with a concept and you formulate a research question, and then you may spend weeks or months at a time designing a very rigorous study that can allow you to test those research questions. Then you're finally ready to collect the data, and that can also be weeks or months, or for some people, more than a year spent in an MRI room or in the lab collecting the data. Once you're done, you can analyze the data and see your results. Now, if you're a lucky one, if that's the way you see it, you'll get positive results and your hypothesis will be confirmed. But if not, you may have a little bit of a problem when it comes to publishing those results. So as part of advancing your career, you're encouraged or sometimes required to have a strong publication record. So all of those studies that you spent weeks and months developing, they all depend on which part of the process? The results. So the part of the research process that is actually, well, not in your control, something you can't really fix, that's what's going to determine if you get published or not in the traditional route of publications. So you write the manuscript and you submit to a journal, and you get the notification for the email that the decision is in, and you open it and you're not really sure because your results were not really positive. You had null findings. So some of these rejections may look like this. So study presents null findings. So why is P not lower than 0.05? You have no groundbreaking results. Results are not novel enough. They do not have enough impact. Reviewers may ask you to do more experiments because there was a flaw in your methods. They may request for alternative analysis that can show you something positive that can come out of this manuscript. They may request for more storytelling. It's not a very compelling story if you start out very, very optimistic and then you found nothing, because that's the way that people see it. Null findings is not anything to present. But for many, many people, null findings can actually be really useful. So this is the file drawer problem. So the studies that did not report positive results did not actually see the light of day. So we don't know what the real state of the literature and our evidence base looks like, because it's very, very skewed, it's biased. We don't see the results that we should be able to see to guide our decisions in future studies. So how did the registry reports tackle this problem? The Registry Reports initiative, as we know it today, started in 2013. The first publication format was launched at Cortex, a neuroscience/psychology journal, and it had 4 key components. The researchers would decide all their methods and hypotheses and analysis before the data collection can take place. And part of the peer review process would take place before the studies can be conducted. Now, if you pass this stage of peer review, then you're virtually guaranteed publication. What that means is that if you follow the protocol that you said you would follow at that first stage, then you get published irrespective of the results. In this initiative, replications are very, very welcome. So your study doesn't need to be too novel for you to get in principle acceptance and get the paper published. So all these factors that actually hurt a little bit when you get that email, they do not count when we talk about registry reports. So anything that has to do with the results will not affect the decision of the journal to publish your research. So going back to the research process is quite simple. At the stage where you can still make adjustments to your research design, this is when the peer review takes place. So before you start collecting the data, you get expert feedback on your study design, and you can still make alterations, and then you can get in principle acceptance, and your study will get published after the results are in. Now, this all sounds great, and why is it important for science? So one of the things is that when you submit a registry report, one of the criteria is that all your methods need to be very, very reproducible. So you need to be very detailed, and you need your methods to be repeatable. So this will increase reproducibility in all the outputs we get published. Then there is a greater need for transparency that comes with that. So most of the journals that adopt registry reports will tell authors that they need to share their data, the code, and the materials where that is possible. So if there are copyright issues or anonymity issues with sensitive neuroimaging data, that's fine. But when possible, you need to share your study data and your code. There's also a very clearly distinguished label, if you want, in the manuscript between the hypothesis and your analysis that were pre-registered during that first stage of peer review, and any exploratory analysis that you did after that you didn't say you were going to do in the beginning. So that can help with hindsight bias. So you're not going to present results from exploratory research as something that you predicted when you didn't, and it will not allow you to selectively report anything. So if you have 15 hypotheses and 2 of them are confirmed, you can't only present those 2, you have to present the results for all 15 hypotheses as part of the registry report. And of course, like we said before, this can help with publication bias, because irrespective of what you find, the study will get published. Now, there's quite a few advantages for authors. I think the most attractive one comes to the end, but if we really think about it, Peer review is supposed to help make the research better and check for problems. Now, when that happens at the end of the research process, there's not really much you can change unless you do more experiments or you redo an experiment and change some variables and some design parameters. So with registry reports, peer review takes place when it matters the most. You still have time to make adjustments, and reviewers don't have the chance to say, I reject this paper because you didn't control variable X, because they can tell you to control it, you do that, and then it gets published. That can also mean that's a higher acceptance rate for registry reports. So if you submit to a journal of your choice, so the journal that you would really like to get published in, you have more of a chance to get it accepted because there's still time to make these adjustments for the reviewers. So for example, at Cortex, these are data that's shared by Chris Chambers, who edits at Cortex. Over 90% of the regular articles were rejected, with only 10% of stage 1 registry reports being rejected. Why? Because there's still time to change the methods in the analysis. This can also mean, it's all connected, you spend less time resubmitting to journals. I know countless of authors that have spent unnecessary time reformatting their papers and trying to find another journal to resubmit to, only because the desk projection took 2 months to arrive. This will likely not happen for registry reports because desk projections are quite fast, and if it gets to peer review, it's in your hands to change the outcome. So if there are major revisions, you have time to do that, and then the article progresses through the peer review system. Now, the best possible advantage for authors is that the paper can be accepted irrespective of the results. So if you are a PhD student and 3 out of 4 of your studies had null findings, I'm not talking about myself, maybe I am, then you won't be worried if you have 1 or 2 studies submitted as registry reports about getting published and having this on your CV, because the results are not going to matter. So I'm not going to bore you about the history of registry reports, but basically the initiative has grown quite a lot from 2013. So the latest development was in 2020 with a rapid network of reviews for COVID-19 related registry reports. So there was a lot of interest and a lot of expert reviewers who provided peer review and then IPA for research for COVID-19, with over 275 journals adopting registry reports. And around 2016 and 2017, there was like a disciplinary expansion, because in most cases we think about registry reports in psychology and cognitive neuroscience. But now we have registry reports in many, many disciplines, and the adoption rate, it keeps going up. If you want to read more about the history of registry reports and how it started and where it's going, there's a preprint here. You could find more information from. Okay, with over 280 journals, like I said, adopting registry reports, there are many fields to choose from. I think for our viewers, psychology and neuroscience are the most relevant, but there's plenty of disciplines here. And there are over 500 published registry reports that can be found in a public Zotero database. I'm just going to quickly show you that. So this is the database. If you're looking for examples of registry reports for your area of research, or like a specific methodology or a neuroimaging technique, that may be a good place to start, just to get familiar with how the articles can look at the final stage. So this has registry reports that have been completed, but it also has, I think it has the stage 1 as well. I'll check back later. It also has published pre-registered studies. So going back to the previous talk, you might find some interesting examples in this database here. Okay.
Lucia, could you actually drop the link to that in the chat? I think there are a lot of people who might really want to go check that out, or maybe we can do it later, but just
I was going to ask if the slides were going to be available, because I have so many links.
Okay. Yeah, never mind. Sorry to interrupt.
It's all right. There it is. I wish I knew there was a database before I did my first registry report. I had no idea what it looked like. Okay, so the Center for Open Science has a page that is the best information hub on registry reports. So you can find all the information that you're going to need there. I'm gonna post that as well, just to. Okay, no, story of my life. There we are. Great. So how do they work in practice? How does writing a registry report actually work? So there's two main stages, stage 1 and stage 2. In stage 1, like in the normal research process cycle, like we said before, you come up with a research idea and you design your study. Now you have a protocol that you submit, so your proposed study goes under peer review at stage 1, and if you pass the major or minor revisions, or if you're extremely lucky, you might not get any revisions at all, you get in principle acceptance, or IPA. What that means is that the journal commits to publishing the research at stage 2, irrespective of the results. After you get IPA, you're ready to conduct your research as exactly you proposed during stage 1, and then you submit the full final manuscript for peer review. What happens at stage 2 for the peer review is that you can't change anything that was accepted at stage 1, but reviewers might have some comments about exploratory analysis or the discussion or your conclusions. So it's quite minor most of the time. And then your register report is published. Now, to break it down into a little bit more detail, what is the stage 1 register report manuscript? So your protocol includes a full introduction, exactly as it will be published, right? So you can't change it unless you have some linguistic edits, like changing a future tense to past tense, but the introduction will remain fixed. Then you present your research questions and your hypothesis, if it is hypothesis-driven research, that is. Then you have a full methods section, so as reproducible as possible, your proposed analysis, and if you have any pilot data to show, you can add them in your stage 1 manuscript. So introduction, methods, analysis, stage 1. Then the manuscript goes through peer review. You need to submit it to an eligible journal. So you need to check for a journal that has adopts registry reports and also is compatible to your research. We're going to go into more detail about that a bit later on. And reviewers assess the manuscript for specific RR criteria. So they're going to check if your analyses are appropriate, if there are concise links between all your hypotheses and your analysis and your research questions, and they may revise the methods as well. So if there are any flaws or things that could be improved in your methodology, that can also be accounted for during the stage 1 peer review. After you pass the peer review stage, the reviews are positive, then you get IPA, irrespective of the results, and your protocol is archived, for example, on OSF. So you can publish the registration on OSF and have it either public or embargoed. This is a good way to track for deviations at stage 2, to check what you said you would do and what you actually did. Now, I'm just going to stop here and see two things. So on the Center for Open Science page, there is a table. This is this link. I will post it as well. That has all the journals that are currently adopting registry reports and different features with regards to that, where you can check. For example, do they offer registry reports for novelties or for meta-analysis? There's a few different things here. So you can check the journal of your choice and the different types of registry report features that they have. Now, there are guidelines on there as well, so you can find generic guidelines, but yeah, I don't think you have to worry about that just yet, because if you do find a journal of your choice, they may have specific guidelines, not generic guidelines. So on the site where it says information for authors, they should have a section for registry reports specifically, where they tell you what they require of the authors when submitting a registry report at stage 1. Okay. Oh, stage 2. Fun. So you pass this stage and now you're ready to do your research. The only thing you need to worry about here is that you don't deviate like in a major way from your methods unless there is a flaw or something didn't work or there was an error in the manuscript. If that happens, changes can be permitted, but it depends on the editor. So you need to contact the editor in such a case. Any deviations will then be transparently reported at stage 2. So the readers will know if something didn't quite work out and it had to be corrected. So you write up your full manuscript. You have everything ready from stage 1. So a lot of the writing is done: introduction, methods, analysis. All you need to add is results and discussion and conclusion. The point here, and it's quite a myth, is that most authors, when they start off with register reports, think that they can't explore their data. So they can't report some interesting results that came up from the correlation or from something else they found. That's not true. So what happens at stage 2 is that you have 2 sections, if you do have exploratory analysis, where you clearly report registered analysis, these are the results, and then exploratory analysis, these are the results. So the reader will know what was confirmatory and what was exploratory, and that's absolutely fine. So the stage 2 manuscript goes through peer review, and then the reviewers are asked to assess whether you complied with the protocol, so whether you did what you said you would do, and whether certain quality checks were passed, or whether your conclusions were evidence-based. So if in your discussion you hide most of your results from pre-registered analysis and then instead report something from an exploratory analysis as like your main message, then that's going to be a problem. That's going to come back with a revision asking you to follow exactly the hypothesis in the discussion, in the way that you discuss your findings. So one of the things I didn't know about when I did my first register report was choosing the right journal. So that can be a bit tricky, but it's not just about finding a journal that has register reports. It's about finding a journal that has register reports that work for you. So especially in neuroscience, that can be a bit tricky. So how do we know it's a good fit? I'm gonna link that as well. So there is a Shiny app for that by Katie Drax, where you can check easily, I think more easily than the table, you can easily check the journal of your choice for the different features. So for example, you have a study that you don't have ethics approval for. Most of the journals ask for ethics approval to be in when you first submit a register report protocol at stage 1. But maybe you don't have time, or you have a clinical sample, or your ethics committee takes ages to reply. Then that's something you need to consider before you submit. Maybe you have a hard to reach population. Oh, what am I doing? And you can't, let's do this. Okay. You cannot recruit 200 people. So power requirements may be a problem for you. Many journals have strict power requirements. So let's look at frontiers in neuroscience. There's one example here. I'm gonna go to, my computer is not cooperating with me today. Okay. Okay, so Frontiers in Neuroscience doesn't have minimum statistical power requirements. So if I have a clinical sample and I can't get 90% power for my study, then I'm going to choose a journal that doesn't have that requirement. Does it require ethical approval straight away? No, it doesn't. Okay, so that may be a good choice. So there's many different features that you can check for using this Shiny app. Let me post it here. Oh, someone, did I do that? Thanks. Someone did that already. Okay. There's also going back to seeing for examples. So get an idea of what everything looks like for your research area or type of research before you start doing this. Just check if it's going to work for you. There are registrations on OSF you can also look for. So a register report protocol registration or completed register reports, you can check for things here. OSF registries. Okay. Maybe you want to check what the register reports for specific journals look like. Right. One other thing that I know I had a problem with, and other ECRs have a problem with, we don't really know how the publication system works, and it can be quite intimidating because the editors seem like these very scary senior people hiding behind the desk, making all the hard decisions, but they're actually, I think most of them, very approachable. So if you have any questions about how these guidelines can fit your research, for example, if they have very strict criteria for specific RR elements, like a sampling plan, but you really, really want to submit to that journal, like it's really important for you, you can send them an email and ask whether they're flexible with making necessary adjustments for a clinical study or for a very, very expensive MRI study. So things like that. You can send pre-submission inquiries to check before you make a full proposal, which takes time, and submit to that journal. I'm going to breathe because I've been talking quite fast. There is a few top tips for submissions that I'd like to share with you. So first, we have the planning stage. So thinking about doing a register report and planning everything. You have to have clear research questions, scientifically valid research questions, and if applicable, respective hypotheses for that. You have to consider the feasibility of your study as early as possible. For example, think about your sampling plan and your resources or time constraints. Is your sampling plan feasible, well justified, and does it meet the journal requirements? Like I said before, this is something to consider before you submit to a journal. You have to check your methods and analysis for feasibility as well. Is everything valid, and is it going to work in the lab? Or is it all good in theory, but maybe it's not going to work? So in these cases, it's helpful to pilot. You don't have to do extensive piloting, but just check that everything works. And maybe as a fly, use data simulations to check if your analysis are going to work as you thought they would. This can help with avoiding deviations at stage 2. The only reason you would have a problem with major revisions or just I don't think it's happened before, getting rejected at stage 2, but major deviations from the protocol can be a problem. So this can help avoid it. Pilot and try out your analysis on simulated data, or just play with your structure of the data and see that everything is working as supposed to when you try your analysis. Now we have the submission stage. As part of the RR criteria, so one of the things that the reviewers will look for is that there are very precise and exhaustive links between all your elements in your protocol. So your research questions, your hypothesis, your analysis, and your sampling plan. So if you have a specific hypothesis, they all have to be tied to a sampling plan. They all have to, even if it's the same, they all have to have analysis. You can't have analysis for 3 of them and then 2 of them don't really have any register analysis tied to them. So everything needs to be linked. This is a way of minimizing researchers' degrees of freedom. So if you manage to do that and be as precise as possible at stage 1, you may get IPA without going through major revisions. But that's one of the most common reasons for getting major revisions for a register report, that or method flaws. So if you can check for that, that can really help you get IPA faster. There is an example protocol that checks for that, let's say, cohesion between our elements, which is available here. I had to do that. It was a nightmare, but it actually helped. So it's very, very specific. I'm going to post it here. I think this is now at Psychonomic Bulletin and Review, but I don't know if it's being shared by other journals. But it's just an example to see things that reviewers look for. So, for example, you need to, if you have a directional hypothesis, you need to be very specific about the direction of that effect in the way that you describe it, so that there is no misinterpretation on exactly what direction that effect is in and between which variables or anything like that. You need to be very specific. You need to make sure that all your variables are clearly labeled. It's quite long. If you talk about procedures, you need to explain if you have randomization in any like a group or within subjects variable, you need to explain exactly how that happened. So you need to be very, very specific about parts of your methods. Now, if you use a protocol coding sheet just like this, it can really help you avoid that major revision coming back with all those questions. So you're prepared and you've already checked your methods for that. So that can really help. It's like doing part of the reviewer's job yourself, which I think hurts a little bit less than getting a 5-page review back. Okay, and then the next part is analysis. This is quite hard for many types of research because without the data, it's really not easy to know what exactly we're going to do. So this is why data simulation can help. One of the things that people don't think about when it comes to analysis is that as part of your registry report proposal, you need to really talk about how your data are going to be analyzed. So how you take the raw scores from something and achieve a t-test. What did you do? Did you pre-process the data? How did you handle missing data? How did you exclude participants based on performance, if you did? What type of aggregation methods did you use? Did you analyze means from trials or medians? So you need to be very, very specific about what type of analysis you're doing. For the main analysis, so not pre-processing, you need to think about contingent plans, if that applies to you. So if you're checking for assumptions of a statistical model, what are you going to do if these are violated? So do you have any contingent analysis plans like alternative tests, or are you going to transform the data? Things like that need to be in the stage 1 protocol. I'm going to pause here. I don't know what my time is. I thought it would be cool to introduce you to a new development for registry reports. The Peer Community in Registry Reports, or PCI-RRs, is a non-commercial platform for reviewing and recommended registry reports, independent of journals in the form of preprints. So how that works is that you submit your proposal to that platform, and it's peer-reviewed by the community, and the reviewers can recommend it to proceed to stage 2. When you get APA, you post the manuscript on the preprint server. Now you complete the study and the preprint gets reviewed, so you update with a stage 2 version. And what can happen after is if a journal has signed up to PCI, so if a journal is PCI-RR friendly, they can publish that registry report without further review. That can mean that you take some of the power back in choosing a journal. And once you've done all the work, you choose which journal to publish in without going through further peer review. It's quite neat, and it's quite new. So I think more journals are signing up now for that. If you wanted to have a look, because it's just going to go through here. So there are not many journals just yet. So it looks like this is very new, like I said. But for neuroscience, I think neuroimage could be quite interesting. Maybe BMJ Open Science, Journal of Cognition, Reproducibility in Neuroscience. So there's a few journals and RSOS that have joined this initiative. And there's other journals that have joined, but they require a little bit of say in what happens in stage 2. So they might be willing to do some further peer review. If you want to have a look at the PCI website, that's here. I'll post that later. Okay. One of the things that I talked about today is how everything needs to be linked together for the registry report to be really, really tight in what we considered minimizing researcher degrees of freedom. So the PCI website has a study design template that requires authors to complete. So you have all your research questions, your hypothesis, and your sampling plan, etc., all linked in the table. So you can clearly check that everything is in there. As part of my work now, we're developing an app for this. So instead of having to come up with all this yourself, you answer a few questions and the app guides you through the process, and you get a report for a stage 1 proposal with all these elements linked with each other. I'm not going to say more about that because I'm still working on it, but it's I think it will be very, very helpful. Now, future improvements for registry reports are quite a lot. So we have, they're great, but there's also many things to improve. And that's absolutely fine, because the more they're getting adopted by journals, the more things to improve might actually come up. So one of the major things for us is monitoring how the journals are actually implementing registry reports. So there's no registry reports police. There's nobody to tell us if editors actually abide by the guidelines or if they reject manuscripts at stage 2. I don't know if that has happened, but there may be a need for community-driven monitoring of these things. So a PhD student in my lab is actually working on this, just a website where people can post different types of reviews and feedback for how journals handle registry reports. We may need more meta science projects to check whether they are working as intended and whether bias is reduced. We may also need some alternative approaches for faster peer review at stage 1. Some of these things are discussed in the preprint I talked about in the beginning. I'm going to post it later in the chat. And also, there's a few variants of registry reports, like exploratory reports, that are not adopted widely at the moment. So they may need to be a little bit more promoted. And the guidelines are also expanding. So for more types of research, rather than just quantitative hypothesis-driven research. So if you're doing mixed methods and you have qualitative data, there's going to be guidelines for that. So there's an expansion of how registry reports can be used. Thank you. I don't know if I'm over my time. I hope that was enough.
That's
I can talk
perfect.
about registry reports, just give you the questions. It's quite a lot.
No, that was great. Thank you so much for sharing all of that fantastic information with us. So I think we've got a couple questions here that are related. There was one that was answered in the chat, so that's great. Thank you, Nikki, for jumping in. Lucia, we've got your video is frozen for us. I don't know if it's frozen for other people, so I'm just going to flip your video back off and then back on again just to see if that fixes it. Well, it has not fixed it, but that's okay. So we have, I had a couple questions actually. One of them, you've provided a lot of really fantastic resources for which journals have registered reports. And actually, this is the first time I'm seeing this. So thank you so much for sharing that, instead of having to hunt through a bunch of different websites to see if a journal actually has a registered report option. Okay, good. I'm glad that it's not frozen for other people. Instead of having to hunt through all the journals, you can just go to this website. But some journals still don't have registered reports, right?
Yes.
So in your experience, or would you be able to comment on whether there are cases where a journal might be willing to consider it, even if it's not officially on their slate of article types?
Yes, there is an initiative for that. It's called the Registry Reports Now. I'm going to post that. It's actually on OSF.
Okay.
Where you can write letters to journals for this. Okay. Yes, so you can actually reach out to the journals and request that, oh, there's interest, give us registry reports. So you can actually do that.
Great. Well, that's fantastic. So hopefully what will happen is that table will grow and more journals will be interested in doing this. So I'm going to run a quick poll here to see whether we are convinced to do registered reports or not as a group. So we can see if people want to join in on that poll. But in the last few minutes here for the panel discussion, we can actually bring up everybody on screen who is here, and we can also go and grab, let's see if we can get Joe and Dawid back as well, if they are interested in joining us. So I'll grab them and see if they can come up. And, okay, so it seems Joe is no longer available, but that's fine. So Dawid, please join us on the screen, and we can pull on other folks who, if you want to turn on your cameras. Great. So this was a fantastic panel. Thank you all so much. I learned so much from this. I feel like, Nikki, we met last year. We talked about OSF, and I started to wade in a little bit, but it really feels like there's a lot that I didn't know, and a lot that I hope that our audience either knew about, at least tangentially, but that they'll actually go out and do this now. So yeah, does anyone first to get just a few minutes at the end here? We've got about 15 minutes before the end of the session. I know it's been quite long, but does anybody have any comments for each other that you would like to say?
I have one comment. I love how enthusiastic everybody is, because when I talk to people at my department about open science, I don't get the same reaction back. So that's quite neat. Yeah.
I'll just quickly note too, this was really informative for me as well, though I have looked a lot at register reports too in the past. But it's also just what really strikes me is how complementary all of these approaches are and how interlinked they already are too. So there's kind of a lot of people maybe think we're not that far along with open science, but when you see people come together like this, you really can see that all the links are starting to happen, and what we need is more researcher uptake, but it's looking really positive, I think.
Yeah, I agree. I always learn a lot from these as well. So thanks, especially Lucia. I haven't heard that much about registered reports before, even though I have been to some of these talks. But it's like new information comes out every time, so it's great to keep up to date. Something here's like, I think we don't just learn about the subject matter anymore, and I think the fact that everybody here, even the presenters, have learned new things shows that the way that we do science is constantly improving. So it's just really nice. It's like a nice time to be doing science.
Yeah, one of the things that struck me about all of these tools, not only in how complementary they are to each other, we have tools that are every little tiny detail of your protocol, and then you can embed that in pre-registration, and then that can become a registered report, and all of these, and then you can share those papers so that they can become available without paywalls. Each of these pieces is really important, but I think that if there's one big takeaway that I have from this session too, it's that open science isn't just kind of the moral good thing to do. It's actually a really beneficial thing for us all to engage in for ourselves. So if you are needing to be selfish and have a reason to convince someone that this is a good thing to do, not just for like science in general is a good thing, but for yourself, that you can get your name out there, you can highlight and brag about your research productivity and output and capacity on your CV this way. This is a way of actually showing that you're working on stuff, even if halfway through data collection, COVID-19 happened and you weren't able to finish the study or something. So I feel like there's a lot of really strong utility that's been shared with us today about these approaches that is beneficial not just to the greater good, but also to us as individual scientists and individual research groups. Yeah.
I was just going to really quickly add to that. Nikki talked a few times about, like future Nikki and things like this. And so there's that whole beneficial piece that you've just mentioned, Megan. But it's one of the things we often say is your most likely future collaborator in 6 months' time is yourself. If you can't remember in 6 months' time what it is that you did and how you made something work, then, what are people in your lab going to do? And so all of this sharing, you're your primary audience. Everyone else benefits too. But actually, if you're feeling selfish about it, you already are your own primary audience for just keeping better records and tracking everything.
It's the same argument for using GitHub for your code, is you don't need to have like version 8, really definitely use this one for sure. Like don't use this one or whatever. Like, I don't know how many times I went back to my code when I was not using GitHub, and you have, 15 versions, and it's like really, really, really use this one final at the end of, your analysis file, and GitHub avoids all of that And now we have tools for avoiding all of that and all of the rest of the science that we do. And then sharing what worked and what didn't work with the world and with our future selves. We have a question actually that I would like to pose to the whole panel, if I may. This comes from Miriam, and she's asking, given that there's a time cost in practicing open science and the benefits that we have just talked about as well, of course, this is my addition, do you have any specific suggestions for how to convince one's PI that this is a good thing to do?
I mean, I would refute that there's a time cost in the long run. I would actually argue that there's a massive time saving if you're looking in the long term and embedding these processes into your daily work cycle.
Yeah, I would also ask that PI if how they'd feel if they started a study that had already been done. If only they could have had the null finding or other aspects of that research to lean on that would have said that's a dead end or that's a path not worth pursuing for these reasons, and to actually look beyond that, to be innovative on top of that research and to build off of what's been done. And that only happens if we do this work that we're all talking about. So I would actually pose it back to say, how would you feel then? And I think you'd say, I wish I had done these steps every single time. So.
Can I disagree with something? I think it does take time. If you start on your own in a department when nobody's doing this and they're actually, you know, like not very supportive. That wasn't my case because my PI was Chris Chambers. But you don't have any training or resources on how to do proper coding or how to share your data or how to anonymize. Like these things take time to learn. I think it's absolutely fine to say that you can start little by little. So it's not like, I think someone mentioned this already, it's not you have to do everything open science. You can start with one small thing. And if your PI is one of those senior people who care about impact and publications, you can tell them that the REF or the funding bodies do care about data sharing and things like that now. So higher impact means more dissemination of the research and greater accessibility. So maybe they will care about that if they don't care about good science. I don't know. Give them a selfish reason to do it, you know?
Well, there's another selfish reason, I think, which is that the time is worth it because aside from your most likely future collaborator being yourself, it's going to be someone else in your lab. And so for a project that's worked on by a trainee or someone who's in the lab who then leaves the lab, they might leave their lab notebook or computer behind, or they might have uploaded all their files to, you know, the same shared hard drive. But like, how do you then take that project and say, here, incoming new graduate student, pick up where they left off? You spend 8 months just figuring out where all the files are. Whereas if you have this, then ideally they could pick up right where they left off. This is me convincing myself to actually do this because while all of this was going on, I was sending messages to my lab. It's like, this is a new lab policy. Now we are doing all of this.
Yeah, I think it may well be that there's an initial time investment, which is kind of expensive, but it is an investment for the long run. So if you look at something like that Ebola protocol, that's massively long. The fact that you can very quickly then make the new version and adapt it because you invested time the first time around, like if you apply that to the different processes and things that you're doing in your lab, that quickly pays back. So I think it's kind of, the activation energy is high, but it is worth it in the long run if you can actually get this stuff set up right.
I really like that, like what was said earlier about little by little, something that we try to do on a slightly different scale is to create what we call a proof of concept, like a proof of concept product or activity or something. So you do something that's just big enough that it can show its own value. So obviously you need to decide for yourself what that is, but it might be a single pre-registration, it might be just starting to put protocols on protocols or something like that. But yeah, the idea of doing it in manageable bite-sized chunks until you can demonstrate the value is probably a smart thing to do.
Absolutely. As with anything, you don't need to jump in, you know, all the way to the deep end. Great. So we've got a couple other questions. One is a comment. Thank you, Miriam. Have definitely done final. Okay, we have another question here that's related to the registered report specifically, but the question is about the estimated timeline for stage 1, and specifically whether the timeline is feasible within the constraints of a master's or PhD thesis. And the second part of the question is the work for registered reports seems reversed over that for a traditional thesis preparation, which is analyses and then selection of the journal kind of coming first. Is this a correct interpretation?
Okay, I'm going to answer based on my personal experience. I did a register report for my thesis, but it took me 2 years. The reason for that was that it was a lot of piloting, and I didn't know what I know now about register reports. So the things I shared with you today, things to take into consideration before you submit, these are things I didn't do, and they added time. So not choosing the right journal, not having all your hypotheses and your analysis linked in the way that register reports are made for. After that, it was around 3 to 4 months. So it was the best chapter I wrote because it was all written. So when it came to the write-up stage, I had done everything already. So it's not that you waste the time to do all these things just to get a publication. This is things that you will do anyway for your thesis. So it's flipped, exactly like you said. But it was the only chapter that I had a publication for in my CV after I finished. So that's pretty useful. So if you can do it, I would say go for it. And my approach to PhD students from now on is we can try if it's a suitable study, if it's not too novel, if it's confirmatory, hypothesis-based, let's try it. If it is rejected and you don't get there, we pre-register and go ahead. So the formula is register reports, if not pre-registration, and then go ahead. And you also have at least half of your research already written down for your thesis. So it's quite helpful. You will remember me in, what was it, 3 years. You have most of the writing done, so it's good.
It probably feels pretty good when you're 2 months out from your defense and you're going, oh my God, I need to write how many words per day? Yeah. We are coming up on the end of this session, which has been extremely engaging and informative. So thank you all so much for being here with us. And in the last final minutes, I will say if there's anything that anybody wants to share as a last minute words of wisdom for our audience and for our group. No pressure, you don't have to, but if there's something that we haven't touched on that you would like to say, now is the time. I guess we have covered everything. Well, again, I want to thank everybody for being here. What an amazing session. What an informative session. Tons of links in the chat. We can also, if some of the speakers have mentioned they are willing to share slides. And then for those of you who have joined from the Comp Neuro course, we will also have a channel set up for the professional development, under the professional development section, where we can pass messages on to the the speakers here. And if any of the speakers would like to join, you are welcome to do so. If you're here from the deep learning course, you will get that channel. Just wait until you get onboarded to the Discord server, which will happen in the next few days. And so once again, thank you all for being here. Thanks also to the Templeton World Charity Foundation for sponsoring this event and for sponsoring Neuromatch Academy in general. We're very grateful. And I look forward to seeing all the fantastic registered reports and protocols and pre-registrations that come out of the fun group projects and everything that's happening in Neuromatch Academy. So thanks again, everybody, for being here, for sharing everything with us. And we'll see you next time, I guess.
Thanks so much, everyone. Take care.
you.
Thank you.
Thanks. Bye.
Who are going to talk about logical fallacies in fMRI, EEG, and big data, and how to avoid them. So I'm really excited to have you both here. Thank you so much for spending time talking to this group. We really appreciate it. And we're also grateful to the Templeton World Charity Foundation for sponsoring this event and for sponsoring Neuromatch Academy. So all of you students out there, thank you to the Templeton World Charity Foundation. They are helping support our teaching assistants. So without any further ado, I will let you guys just take it away. And I'll ask if you have any, when you want me to run the polls and stuff, just let me know. And I'll remind the audience also that we have a nice big ask a question button at the bottom. So if you have any questions, please put them there, and also check in on those questions periodically. Even if you're not asking them, then you can upvote the ones that you think are good. So yeah, please take it away, Walter. Thank you so much.
Cool. Well, thank you very much for having me. Can you see my screen now?
Not quite yet. If you hit the button again.
No, what I've got to do is I've got to do the share screen feature. I don't even get to do that. It goes straight to my slideshow. So where's the share screen feature? Oh, it's over. You hand over here. Share screen. Allow.
Beautiful. Okay.
There you go. Now you can see it.
All right. So
Yeah.
we're mute, but we'll be hanging out here.
Good. So thank you very much, Megan, for arranging all this and for inviting us. I'm actually going to be in 2 sessions today and on Thursday. Both of them are really background about how philosophy can be useful to neuroscientists. It's also true the other way around, that neuroscience can, of course, be very useful to philosophers. But we're going to talk today, Felipe and I are going to talk about logical fallacies. I'm going to focus in on kind of the micro scale. He's going to talk about big data. Then Scott Hutel and I are going to talk on Thursday about applications of neuroscience and some of the ethical problems that they raise. But I'm going to start real small. I'm going to start off by telling you about human nature. So human nature is something that a lot of people don't even believe in. They think it's all about personality, individual personality traits, or culture and society. But I want to say there's some things that should count as human nature simply because they're universal. They apply to all humans. Oh my gosh, what is that going to be? How are you going to know what's universal? Well, here's one. All humans make mistakes. I mean, that's, come on. I've never met anybody who doesn't make mistakes. Megan, you make mistakes, don't you? Felipe, you make mistakes. All humans make mistakes. That's all I'm going to assume about human nature. Oh, well, there's a second premise to this argument. All scientists are humans. Look, my cats can figure out how to catch a mouse, but they don't have a theory. They don't have a scientific theory about how to catch a mouse. They have learned traits that enable them to achieve their goals in particular situations. But in terms of generating scientific theories, humans are the ones who do it. So it follows that all scientists make mistakes. I wanted to start off with an example of a good argument. Notice this is valid. If the premises are true, the conclusion has to be true, but you probably already knew the conclusion to begin with. So you haven't learned much yet. Neuroscientists are no exception. That's the next claim. Neuroscientists are scientists, so they make mistakes too. That shouldn't be surprising. Mistakes, what kind of mistakes do they make? Well, they make all kinds of mistakes. We're going to be talking about mistakes in arguments, which are fallacies. You could get technical about what counts as a fallacy or not. I'm just going to be talking about the kinds of mistakes that neuroscientists make. And I'm going to go through 5 main fallacies in the next 45 minutes, and then Felipe will take over and do the last 45 minutes. All 5 fallacies I'm going to try to illustrate in the same way, namely with reference to a classic neuroscience experiment by Benjamin Libet. Libet was a physiologist at the University of California at San Francisco. He studied consciousness and action. He was particularly interested in questions of timing. And little known, he was awarded the first virtual Nobel Prize in psychology in 2003. I think most people don't even know that was a virtual Nobel Prize. I don't think they award it anymore, but he won it for the first time. And what he did in order to warrant that award was a series of experiments that we're going to focus on. So the instructions of these experiments were very simple. Sit in your chair, put your hand on the table in front of you, and flex your wrist whenever you feel like it. You decide when to do it, and you just flex your wrist whenever you want to. Sometimes it was move your finger, sometimes it's squeeze a ball, but we're going to talk about flexing your wrist. And there was an EMG electrode on his hand to see when the wrist muscles began to contract in order to flex it. And there were EEG electrodes on the scalp in order to measure the brain activity that was going on when this very simple physical motion was performed. Now, Korndecker and Huber had discovered in the 60s, 20 years earlier, that the signal in the EEG would be an RP, or readiness potential. We're going to call it an RP, Bereitschaft potential. They called it, but we're going to call it an RP. So that was all pretty well known. What Libet really contributed was the clock that the participant is looking at in the cartoon. So the clock looks something like this. They had a dot that went around the clock every 2.5 seconds approximately. I think it was 2506 milliseconds. And imagine that you're a participant. You're sitting in the chair with your hand on a desk, and you're told flex your wrist when you feel like it, and you flex it. And then he says, but what I want you to do is to tell me when you feel the urge to move your hand. So the instructions are flex your wrist when you feel the urge. And when you feel the urge, tell me where the dot was as it's going around. So we could get a timing of when the person became conscious of feeling that urge. Now, other experiments did it and make a decision to move your wrist or exert the effort to move your wrist. It's been replicated many, many times, but we're going to talk about his original experiment, and there the instructions were to report when you felt the urge. What he got from individual subjects, so this is 4 subjects, SS, GL, SB, BD, are individual subjects, and these are the waveforms that are averaged over 40 trials. RP1 is when somebody was looking at that clock and said, oh, I'm going to flex my wrist when the dot hits 30. So it was pre-planned when it would happen. RP2 was not pre-planned. So flex your wrist whenever you want. And we got somewhat different waveforms from the 2 of those. One thing that's important for our purposes is the S there, the column S, which is often neglected, but he wanted a control, and he wanted something where you would report your inner subjective feeling and the timing of it that wasn't an urge to move. So S means sensation. He would prick your hand, and this participant's hand, and say, okay, when did you feel that? And you would have to report the spot on the clock when you felt it. So you got the timing of the sensation and the timing of the urge to move. And notice that in the urge to move, in the RP, it goes up, then it goes down again and comes back to the norm. You don't get that shape with the sensation alone. If you average 9 participants, that's all he had in 1983. As I said, it's been replicated many times, but if you average his original 9, you get the type 1 RP pre-planned, starting at about 1,000 milliseconds before the motion. The no pre-planning, about 500, 550 milliseconds before the motion. W there is the average time at which people reported becoming conscious of willing to act. So W stands for conscious will, and M stands for the motion occurring. So you can see that The will occurs after the brain activity, the RP, begins, but before the motion. Okay, so this is a very simple experiment. I'm sure it's known to many of you, but I thought I would just stop and see if there are any questions so far about the experiment and the setup. So Megan, you tell me, because I can't see anything on my screen.
Well, we don't have any questions now. We can wait
Okay.
just a couple seconds. If you guys have questions, then we can also put them in the ask question and address them later. You can drop them in the chat. Right now, there are no questions, so you are being perfectly clear.
Okay, good. I just wanted to make sure before I move on, because we're going to be referring back to this experiment for the rest of the discussion, my part of the discussion.
So
Okay.
I'll let you know if there are any questions that come in that need to be immediately dealt with.
Cool. My questions, okay, you've got the Libet results, you know the experimental setup. What does any of that show? Well, a lot of Libet's followers drew some pretty extreme conclusions from this. Wegner, for example, said, well, Libet's experiment shows, along with other experiments, but among others, it shows that conscious will is an illusion. It's not clear what he meant by an illusion. So I'm going to refer back to Huxley and Nietzsche in the 19th century. They said that conscious will does not cause our actions. So it's like the smoke coming out of a train. It's a result of the train, but it doesn't drive the train. The smoke does not have any effect on the motion of the train itself. And that's what Huxley and Nietzsche thought the conscious will was like. It's a sign that action is occurring, but it's not what's causing the action. And as a result, we never act from free will. We never act from will at all, regardless of whether it's free. And so that's a pretty surprising conclusion to most people. What I want to look at is, how do they get there? How can they argue from Libet's findings to conclusions like that? Okay? And we're going to look at a number of arguments that are in the various commentaries on Libet. First argument. Well, look, these conclusions that Wegner and Nietzsche and Huxley drew are about all actions, but Libet only studied some actions. So at some point, they have to reason something like this. An RP preceded all actions in Libet's experiments, right? All of those physical movements of the wrist. Therefore, an RP precedes all actions, right? The RP is not going to tell you anything about all actions if it's only occurring in some. So you wouldn't be able to draw the conclusion if you didn't reason something like that. So the first question is, does this conclusion follow? So here's the first poll. I invite you to go to the poll site. I'm not sure where it is on your screen. Megan might be able to help you with that. But does the conclusion follow logically from the premise? Yes or no, or I don't know. You tell me.
So we've got the poll on the bottom of your screen here. It'll show up as an orange button. So please go and answer and see, and I'll read out the results in just a second. And while we're waiting for those results to come in, let's ask a very quick question, which is just to clarify why the time of sensation was important to be measured in the Libet experiment.
Because a lot of people thought that by looking at the clock, you're going to get distracted and not be good at timing what's going on in your mind, your internal sensations. And so he wanted to show that you could time the sensation and you could also time the urge to move. So he wanted to show that his clock was actually useful at figuring out when people had internal sensations, either of a prick or of an urge to move. So it was part of showing that the apparatus really worked at getting at the question he wanted to get at.
Great, thanks. So Eduardo, my response to you was partially correct, and Walter's response is the one that you should pay attention to. All right, so we have the results of our polls are coming in. Of the folks who participated, all of them said no. The conclusion did not follow the premise.
Good. So one thing about logic is that the answers are often supposed to be obvious, but then the trick is to figure out why. So let me say a little bit about why it's a bad thing. And an example, one way to show it's a fallacy is to find another argument that's similar to it, that's obviously bad. So you say, this person lied to me, so everybody like that person is dishonest, will also lie to me. So it doesn't matter whether the X in this is, African American or Chinese or male or female or rich or poor. It's a bad argument anyway, but it's the basis of stereotypes which people make all the time. And so you can see that generalizing to a whole group of people from one member is no good. But wait a minute. If this is a bad argument, doesn't all science generalize? When you do an experiment, you always test a certain small set of actions, and then you generalize to others, or you test a certain sample of something, and then you generalize to others. So wait a minute, if you can't generalize, then how can you do science at all? Well, it's okay to generalize if you already know a few background facts. So for example, if I take a penny and throw it in the lake and it goes under, it doesn't float, then I can conclude that pennies don't float. I don't have to check every coin in my pocket, in order to see they don't float, because I know that if one of them doesn't float, the others aren't going to float either. If I take an apple off a tree and put it in the water and it floats, I don't have to take every apple off the tree to make sure that all the apples will, because I know all the apples are basically the same in the ways that are relevant to whether it floats in water or not. Okay. The other way you can generalize is when you've tested enough of a sample that it varies in ways that represent the overall population. Of course, you have to know how they vary in the sample and in the population and make sure they match, but you can do that. The problem with generalizing in Libet's experiments is that we don't know that all actions are the same in relevant ways, and the actions that he studied are actually atypical. I mean, think about just moving your hand for no reason when it makes no difference when you move it. It's not the kind of thing that people do very often. There are many other actions that we make that are much more consequential. So let's study some of those consequential decisions. This is Uri Mautz and Gideon Yaffe and Christoph Koch and Liad Mudrik did a study to distinguish an arbitrary choice, like there is in the Libet experiment, from a deliberate choice, to see whether you can generalize from a small sample to the larger sample. What they did is they took a $1,000 donation, and they said, okay, you can either press the right button or the left button, and if you press the right button, then this charity will get $1,000. If you press the left button, then this other charity will get $1,000. They didn't really give $1,000 on every trial, but it went into a pool where they would pick one. Great experiment. If you can figure out a way to design an experiment that enables you to give money to charities, that's great. So this charity on the right, that charity on the left, that's the deliberate one, because you got to think about which charity you want. But what if it says, if you press the right button, this charity gets $1,000, and if you press the left button, the very same charity gets $1,000. Now it's just arbitrary. You can press either one. And the question is, do you get an RP on both of those? Well, what they found was that there are actually different neural mechanisms that you can differentiate in a number of different ways. It's a great paper. But what we're interested in is they got an RP. Here it's plotted downward because the voltage is negative. And so you can see the blue line is showing an RP for the arbitrary, but not for the deliberate. So what that shows you is it's not just you can't get all from some, but in this case, there are differences among the actions that make a big difference and so you can't generalize from arbitrary actions to deliberate. Now we know that there's a difference in the sample. So as a result, you're all right. No, it does not follow. Any questions about that? I'm going to stop every once in a while, just see if there are any questions. And if there aren't, that's fine. But
No questions so far, but this should indicate to all of you that we are going to have lots of opportunity to answer your questions. So don't be shy if you do have questions. There's no silly question, nothing too small. If something is unclear, speak up.
Okay, second argument. Okay, let's assume that there is an RP before arbitrary physical acts, okay? There was not an RP in trials where the subjects felt sensations. We talked about that before. That's when they just felt the sensation without physically moving, without deciding to move any part of their body. They might have, had some kind of reaction to getting pricked, but they did not intentionally move. So it looks like it's only physical actions that are preceded by an RP, right? It's not just sensations like that. So does that conclusion follow? There's an RP before arbitrary physical actions, but there's no RP when the subjects feel sensations without physically moving. Therefore, only physical actions are preceded by an RP. Does that conclusion, C, follow from the two premises, P1 and P2? Yes, no, or I don't know.
All right, the poll has been launched, and while you guys are responding, click the buttons, please. And if there are any other questions, please do put them also in that Ask a Question button. We've got a couple votes. Come on, I know there's like many of you in this room and only a few of you participating. This is an interactive session.
We can wait and check the poll later too, but then people get the answer. That's not fair.
That's true. The answer comes in a few slides, so maybe we can, here we go. Now they're starting to come in. Okay, good. And just so you guys know also, once you've answered the poll, you should be able to see everybody else's answers too. So that's kind of an interesting way to check your understanding. And we've got a question, if we've got time for it.
Sure.
Did contemporary scientists point out the generalization mistake others made immediately? Just a little surprised that some scientists made that mistake. And this question comes from Sean.
It does. That's why I started with my slide about human nature. Scientists are humans. Humans make mistakes. It might be surprising that scientists would make such simple mistakes, but they're all over the place in the literature on Libet and many other topics.
And that's
so
why
people, for example, say things about humans by only studying, college students at elite universities in the Western world. Like, come on, you can't generalize from that to the, to all other people. And yet scientists have been doing that for a long time.
And this is why all of you in the room are not going to make these mistakes now, because you're learning about them. All right, so the answer to this poll is as follows. We have 1 vote for yes, 13 votes for no, and 5 votes for I don't know.
Okay, so let me give you my analogy. Remember the first fallacy was called hasty generalization. I like to call this one hasty exclusion. And the way you show a fallacy is by getting a parallel argument that has the same form but is obviously no good. Here's mine. Birds can fly, but snakes, lobsters, and pigs can't fly, so only birds can fly. Obviously wrong. They're flying fish, they're flying insects, they're flying squirrels on the right of the screen. So that argument's no good, but it's the same form as the other argument. It's saying this happens in this case, not in this other case. Therefore, that's the first one is the only one it happens. You can't get there, okay? But are there flying fish and flying squirrels and the equivalent in the world of actions? Libet tested physical acts and sensations. And he said, you don't get an RP for sensations, you do for physical acts. Okay, so what's different from performing a physical act, but also different from feeling a sensation? Okay, so it's not a physical act, and it's not just feeling a sensation. Well, it's what philosophers call a mental act. I can sit here and plan what I'm going to have for dinner when I finish giving this talk without moving at all. I can calculate 17 plus 21 in my head. Just did it without moving at all. And so you can have mental actions like picking something, planning, and calculating, and they don't involve physical motion. So what we did, Alexander and Prescott Alexander, Scotty, so we had people look at the waiting phase, look at that. They're waiting and they see letters appear in the 4 quadrants inside the Libet clock circle, okay? With the cursor moving around just like in a Libet clock. And at a certain point, we say, now decide, pick one of those numbers. So let's suppose I pick J in the upper left of the decision phase. In some trials, we had you press a button to indicate which one you had picked or when you had picked. In other trials, we said, just stay perfectly still and just pick it in your mind and you'll press a button later. And if, in fact, you only get an RP before physical motions, you should not get an RP when you're merely picking and not moving any part of your body. But look in the upper right, we got the same waveform of RP when it was merely a decision with no physical motion. So it's not only physical actions that are preceded by an RP, it's also physical actions. So the conclusion does not follow. And the experiment with mental actions shows that. Let me know if there are any questions. Otherwise, I'll move on.
We've actually got a couple questions here.
Cool. Good.
Can we please go over briefly again why the clock was used in the experiment? Does it have anything to do with the time perspective of the participants? This question is from Romeo.
So you mean the original Libet experiment, it was used in order to time conscious will, because there was at that time no external indicator of the presence of conscious will or the timing of conscious will. So you needed a visible clock in order to time it. We used it in our experiment here with mental actions in order to be as close to Libet as possible, because we wanted to see whether a mental action would have an RP when there was no physical motion. So we tried to mimic Libet as closely as we could.
Great, thanks. And one other, well, a couple other questions, but we won't take too long here. Anna is asking, is this really a case of making mistakes? Or alternatively, you won't publish unless you make your claim sound important. I feel like we are forced to generalize if we want to publish.
I think that is absolutely right. I think you're completely on target there. I think a lot of times people overgeneralize and make it sound a lot more important and a lot bigger conclusion than it is because they want to get published. And there's pressure to do that. And I think that's bad for science and it ought to stop, right? People ought to admit that they've shown this much, but not that much. That's, you know, I love the fact that science papers have limitations and future directions at the end of the paper. Philosophers don't do that. Philosophers should do it as well. It's not just scientists. But you're right, there's pressure to publish, and overgeneralization can sometimes get you published if the reviewers don't notice it.
Great. And there's one more question that's about correlation and causation. And
we're going to come back to that. So
I figured we'd get there in a little bit. So Yiti, just hang on to that thought and we'll come back.
Good. Third argument. Let's assume there's an RP before all arbitrary physical acts. It might also be between mental acts, but at least before all arbitrary physical acts. So what? What does that have to do with free will? Okay. Well, some of Libet's followers seem to argue like this. They say, well, look, what is it that causes your hand to move? Either it's the brain activity, the RP, or it's the conscious will, the W. So either RP causes the motion or W causes the motion. And RP causes the motion because, you know, let's just take that for granted for now. We will come back to the causation question in a few minutes. So W does not cause the motion, okay? Does that conclusion follow? So here's another quick poll. Let's see if we can get some answers to this one. Either the readiness potential causes the hand motion or the conscious will causes the hand motion. the readiness potential causes the hand motion. Let's take that for granted for now. Does it follow that the conscious will does not cause the hand motion?
All right, the results are rolling in. Click the orange button at the bottom of your screen, folks, and participate in this poll. All right. Come on, there are about 5 times as many of you in the audience as are participating in the polls. I know you guys can do better. All right. So, well, it looks like the results are overwhelming, even with the few people who are participating. The answer is no.
No, you guys are getting the hang of this. I'm telling you about fallacy. So the answer is no. Good. But again, we want to understand why. So what's going on here? It's called affirming a disjunct. A disjunct is when you have an either or sentence, that's called a disjunction. And the either part is one disjunct and the or part is another disjunct. So let's do something analogous to the RP argument that we saw before. Either smoking causes death or lung cancer causes death. Smoking causes death, therefore lung cancer does not cause death. No, like that's not right. Why? Because they both cause death. Because smoking causes lung cancer, which then causes death. Well, analogously, maybe the readiness potential, the brain activity, might cause the conscious will, the W, and then that causes the motions. It could be a chain of causes. Now, notice that the conclusion would follow, but only if the or meant one or the other and not both. But then the argument would need to exclude both. And that's exactly what the issue is at stake. So the argument ends up begging the question and doesn't really show the conclusion because it just assumes the conclusion. So the answer to this poll question is no. Want to give another chance for questions if there are any. Don't have to
So we
be.
do the correlation causation one now, or do we want to hang on to that one for a little bit later?
That's the 5th argument.
Okay, all right.
I'll do the 4th one quickly and we'll get to that.
All right,
Okay?
just hang tight, ET. We'll get there.
Let's step back and look at the big picture now. What kind of interpretation can we make of the libet data? Some people are libertarians. This is a traditional view among some philosophers, not me, but some philosophers, that the conscious will is what causes your hand to move, and your conscious will is not caused by anything. It's completely free. Then there's backwards causation is another view. Well, the conscious will causes the brain activity, the RP, the readiness potential, and the readiness potential causes the motion. But that's backward causation because the W occurs later than the RP at negative 150 milliseconds instead of negative 500 milliseconds. And then there's epiphenomenalism, which was the view of Wegener and Huxley and Nietzsche at the beginning, that the RP causes the motion. All the causal action is going on at the physical level, but the W, the conscious will, doesn't cause anything. So it's tempting, and many people who argue for epiphenomenalism, like Wegener and Huxley and Nietzsche, say, well, look, one of these is true, either L or B or E, but L can't be true because you can't have a physical event that has no cause whatsoever. And B can't be true because there can't be causation backwards in time. So E must be true. So what about that argument? Either L, the libertarian view that the will causes the motion, but it's not caused by anything, or backwards causation, that the conscious will causes the RP, or epiphenomenalism, the conscious will doesn't cause anything. Okay, well, L doesn't seem right, B doesn't seem right, therefore E. Does that conclusion follow?
All right, guys, the poll's up.
Is it possible for the premises to be true when the conclusion is false?
Hit that button. And while we're waiting for the results to come in, we've got a quick clarification from Sean asking, what is the definition of cause, and is it always the same for all of the slides here?
I take cause to be, I'm an interventionist about causation. So it's what is a difference maker in the result that makes a difference to whether or not the event, the result occurs. That's the way I'm understanding causation. If you want a good Making Things Happen by Jim Woodward is a great book on causation if you want to look at it. But, Judea Pearl in his work on causation, such as his recent book, Why, would take a similar view.
We had a whole day or half day at least on causality in the comp neuro course as well. So
wow, if I'd known that, I would have thrown it.
For those of you who came from comp neuro, go back and look at those. And for those of you who are coming into deep learning soon, maybe have a look at the comp neuro materials. Okay, so the results of the poll are in. Oh, this one's a little bit more evenly distributed. So we've got yes for 5, we've got 7 no's, and we've got 3 I don't knows. 8
okay.
no's. Yeah, a little bit more distributed.
The answer is yes.
Aha.
Because it's not possible see, I tricked you. The first 3 were no. You got in this habit. If the premises are true, there's no way for the conclusion to be false. The problem with this argument is not that the conclusion doesn't follow from the premises, it's that the premise is false. It's not true that either L or B or E. Compare this argument. Either my shirt is blue or green or yellow. It's not blue, it's not green. Therefore my shirt is yellow. I assume you can see it. It's pink, right? What the problem is not that it doesn't follow. If it were true that my shirt was one of those colors, but it wasn't blue or green, it would have to be yellow. But the first premise is just false. So the mistake is overlooking another option. What's the other option that Libet's argument overlooks? It's the common sense view that RP causes W and then W causes M. Notice that if we go back a few slides, that was left off. Libertarianism, backwards causation, epiphenomenalism. Oh, forgot about the one that's just kind of the common sense view. The RP is at -500, W is at -150, M is at 0. So what's the problem? No backwards causation, no epiphenomenalism, and no uncaused causes. Just solved all the problems. Let me go out. Instead of taking a question here, let me go on to this question of causation because that one's been pressing people, okay? And I want to make sure I leave Felipe enough time. Because the question is fine, that argument didn't work, but is it really true that the readiness potential causes the will and the will causes the motion? Let's assume, it's not really true, but let's assume that an RP always precedes a W. Every time you have a conscious will, there's an RP that occurs before. And a W always precedes an M, that is an arbitrary physical movement of your hand, right? Then one might be tempted to conclude that RP causes W and then W causes M. But does this conclusion follow? Another poll, but I'm pretty sure what people are going to say about this one, it doesn't follow, right? I'm not sure we need to wait for the poll on this one because people have been saying it doesn't follow. Causation's not going to follow from the mere fact that it always comes before, okay? So I want to say, no, that conclusion doesn't follow. Just like ice cream sales are correlated with shark attacks, but that doesn't mean that ice cream sales cause shark attacks. Why? Because there's a third factor, namely the heat. When it's really hot, people buy more ice cream. They also go swimming, and then there are going to be more shark attacks. But how can we tell? Well, manipulate the ice cream sales. You close down a bunch of ice cream shops. Is that going to drive the shark attacks down? No, it might drive them up. Because after all, if you don't have ice cream to cool you down, you might go swimming more often. So you need to manipulate the cause and see if the effect follows. So in Libet style experiments, what we need to do to test whether RP causes W is to vary the RP, the brain activity, And to test whether the W, the conscious will, causes the motion, we need to manipulate the conscious will. So let's first talk about varying RP. You can't really manipulate it. You can't make it occur earlier or later. But remember that those results where RP and W were averaged over 40 trials. So what you can do is split the early RPs, the earliest 20, from the later RPs, the later 20. And the same for the Ws, the early Ws from the late Ws. And if RP, if that brain activity causes conscious will, then the early RPs should be associated with early Ws and the late RPs should be associated with late Ws. Right? So if it doesn't happen that way, then the RP does not cause the conscious will. That is, the brain activity does not cause the conscious will. Okay? So Alex Schlegel there, we did an experiment following up on Haggard and Eimer and found that if you look at the diagram, there's no difference between the early Ws and the late Ws with regard to RP. The brain activity is no earlier for the early Ws than for the late Ws. There's just no significant difference. So it looks like the RP, can't be what's causing the W, because if it were, you'd have an association between the early RPs and the early Ws. Let's go to the next one, manipulating W, which is conscious will. If the brain activity causes the conscious will, and the conscious will causes the physical motion, now let's get rid of the conscious will, but try to keep the brain activity. Then, since the W is what causes the physical motion, there shouldn't be any physical motion if there's no W. So if you get RP and M, that is the brain activity and the hand motion, to occur, even when we've gotten rid of W, the conscious will, then the common sense interpretation C can't be true. It can't be true that RP causes W and W causes M. The trick is, how can we get rid of W but keep the RP and the M? That is, how can we keep the brain activity and the hand motion but not have a conscious will? Why is your hand going to start moving when you're not consciously willing it? The answer is hypnosis, but not one of these weird magical trick hypnotists that are all over the place these days. Instead, Talia Wheatley, my friend, who is a great hypnotist. She can hypnotize an entire classroom of students. It's amazing what she can do. So what we did in our experiment was we hypnotized people, and then when they came out of the hypnotic trance, we asked them, do you remember it? If they remembered it, we threw them out. And then we had a post-hypnotic suggestion that they would squeeze a ball. But we told them this story. We said, actually, that electrode on your hand, which was really an EMG, we said, it's messing up, and sometimes it sends electricity into the muscles and makes your hand squeeze. So you got to tell us whether when you squeeze that ball, you did it consciously and chose to do it, or whether it was the electrical impulse that caused you to do it. And some of the trials, these people said, oh, it caused me to do it. I didn't do that. It was the electrode. The electrode didn't have any chance of doing it. It was totally a post-hypnotic suggestion. But what that gave us was motion without conscious will. And sure enough, we found an RP in these subjects. It's quite over the place. It's not as clean as the others, but it's basically the same shape, which means that we had RP. We got rid of W, but we still had RP and M. But if that's true, then the common sense interpretation has to be false. Because if RP causes W and W causes M, then the W is missing. You're not going to get the M. You're not going to get the result. But we did get the result. But wait a minute. The other options also had problems. Libertarianism, you got uncaused causes. How's that going to work? Backwards causation in time. How's that going to work? Epiphenomenalism. That means I can't make my hand move by choosing to move it. That seems weird. That doesn't seem right. So do we know what causes what here? how can we solve this mystery? I have no idea. I don't know. I'm not going to tell you the answer to this. Instead, I'm just going to summarize. We've seen hasty generalization, hasty exclusion, affirming a disjunct, false dichotomy, and correlation to causation. We've seen a bunch of these fallacies. They all seem so clear you wouldn't think scientists would make them, but they do, believe me. I can show you videotapes if we had more time of people doing it in lab meetings, for example. So what conclusions do we draw? Well, my conclusion is we need to learn to spot fallacies in order to avoid mistakes like this, even simple mistakes that people think they don't make. And that requires training in logic and in philosophy. We don't yet know what, if any, conclusion to draw from Libet's experiments in particular. Okay. And more generally, I hope that some of this shows that we don't really know nearly as much as we think we know. So we've got to keep, looking into these issues with a careful, critical mind. So I'll take a few questions and then we'll turn it over to Felipe.
I will applaud, even though you can't hear the rest of the crowd applauding. I'm sure they are. Thank you very much. This was fascinating, and I think it was also fun and engaging for everybody here, including me. I love when the talk ends with "I have no idea, but we should."
Yeah.
While we wait for Felipe to get his slides going, we've got a couple questions that we can answer, and we can kind of do this during the transition if we want.
Okay.
So the questions, we've got one question here. Do philosophers seriously consider the notion of retrocausality?
So the answer to that, interestingly, is no, but neuroscientists do. If you look at the writings of Stuart Hameroff at the University of Arizona, he's actually an anesthesiologist and a neuroscientist. He has argued that that is the solution to the Libet puzzle. I do not know a single philosopher who argues that, but Stuart is one neuroscientist who does argue for that claim. Very interesting.
Very interesting indeed. Okay, I'll have to go look that up too. I know Hameroff's work. I didn't know that he had he'd argued for retrocausality. Okay.
It all has to do with quantum mechanical things happening in the microtubules.
That's right.
But how he knows that's what's making it happen, I don't know.
That's right. That's his theory of consciousness. Yep. Is the quantum microtubule vibration theory. All right. So we've got a couple other questions. Felipe, how are we doing with tech? Do we want to give it a go?
Do you want to? Yeah, let's give it a go.
All right. So I think we'll close this, and we'll see if Felipe's slides can share. We were having trouble with this earlier. Aha! Hooray! Okay. So maybe we've got a couple other questions here, but maybe we can do the big question, which has the most upvotes. And then we'll move on to Felipe's.
And I'll answer quickly, Felipe. I don't want to take up too much.
Okay. So I think that this one also will probably be something that Felipe can speak to as well. So Romeo's asking, what kinds of training do you suggest to spot fallacies in logical thinking?
Oh my gosh. Well, I would have to say, what would I recommend? I would recommend that you take my MOOC called Think Again, that you buy and read my book called Think Again, that you work through my textbook called Understanding Arguments, and that you take my pod course. I forget the name of my pod course, I'm sorry, but it's with Himalaya and Lyceum. There are a lot of materials out there. I mean, I've got my own way of doing it, but other people have theirs. But when it comes to fallacies, I think, the message is pretty similar, and any one of those textbooks or MOOCs or pod courses will do the trick.
Can I chime in also and answer that I think that a course, even if it's just one in philosophy of science and or logic, should be required training for everybody who wants to be a scientist.
Yes.
It's really,
Here.
and it, like taking a philosophy of science course when I was an undergrad, changed the way I viewed science, and I was a cognitive science major. So strongly encourage everyone, seek out, if you've got philosophy departments at your home universities, go find a course and see if you can audit it, sit in, even take it for credit. Cool. So we've got other questions too. And Walter, you can feel free to type comments if you'd like to, or maybe we can have a little bit of a discussion at the end if people want to stick around.
I'll be happy to stick around.
Okay. But for now, we will move on to Felipe. So thank you very much for being with us. And we will talk now about how to avoid logical fallacies in big data.
All right, awesome. Can you see my slides now?
Yes, it looks beautiful.
Great, and you can hear me well.
Yes.
All right, so Walter, you should feel free to answer to all those questions in the chat because you have seen this talk before. All right, so many of you are probably like me and like Walter and many here are interested in trying to understand how the mind correlates with the brain. Actually, this is a very venerable tradition. You can trace it as back as the ventricular theory that started with Galen, according to which the ventricles of the brain were related to our brain functions. This was a very long-standing theory that later on got replaced by a few others. Perhaps one of the most famous ones is phrenology. Many of you probably have seen these kinds of images of the phrenological maps. But perhaps what you don't know is that phrenology was really considered as a science, and there were several journals, including the American Phrenological Journal, in which there were special issues just dedicated to argue whether one particular map was a better map rather than another, which is not that different from the kinds of journals that we have now, where we look at brain correlates of brain functions, right? Moreover, just as it happens now, where we have practical recommendations on the basis of cognitive neuroscience, back then there were practical recommendations on the basis of phrenology. So for instance, this is a book from 1902 called Vaughan's Practical Character Reader, in which they use phrenology to advise people how to find husbands. So for instance, they say to look at husbands, men who have head shapes like this, those are genuines. But this kind of head shape is actually unreliable. And part of a reason, for instance, is that these individuals tend to be polygamous. Polygamy, they say, is an amalgamation of amativeness, spirituality, and veneration, which were supposed to be parts of how the mind related to brain. And according to them, this was strange but perfectly true. So people employ the science of the day of how to correlate mind functions with brain structures, if you want, to even produce advice. And we see that even today. Now, when cognitive neuroscience started in the 1980s, it started off with a rather naive assumption that there was going to be something like a smooth transition from psychological terms to neural terms. Here, the P's are the psychological terms and the B's are the brain terms, right? This should have been a P. And in fact, if you sort of look in Google Scholar for papers, the titles of which start with "the neural correlates of," you get, and this was the search that I did about 3 months ago, with over 2,300 results, in which the complement is some sort of folk psychological or folk scientific or psychological term, like verbal working memory, maternal and romantic love, subjective value during intertemporal choice, sensory awareness, spider phobia, and so on and so forth, right? But when you start looking into the actual picture, it actually starts to look like almost every brain region actually appears to be engaged in all sorts of different cognitive processes during all sorts of different experimental tasks. So what I am trying to depict here is that the correlations are really sort of many to many, and the different widths of these lines are supposed to indicate probabilities of there's being correlation. So far from being a one-to-one relation or even a one-to-many relation, the picture is rather a many-to-many relation. So for some, this is just becoming an impossible task. This is something that we're just not going to be able to do. Now, recently, there was this paper published a couple of years ago by Acob's group, first author Sarah Gannon, in which they tried to tell us that we shouldn't give up, that actually there is a possibility for us to characterize the function of brain regions. And in fact, this paper, I single out this paper, not so much because it's just unique, but rather because it is the most concrete example of a new trend in philosophy, in cognitive neuroscience, to the effect that big data is going to be sort of the solution. So the suggestion is, look, the reason why it is so difficult for us so far to identify it or have these correlations between cognitive functions and brain structures is that we have been following what I call here the top-down approach, according to which the starting point is some sort of a priori cognitive, or what they call a behavioral function, such as episodic memory, working memory, visual attention, so on and so forth. And the unknowns that we're trying to identify are the brain regions that are associated with such cognitive functions. Instead, what Gannon and collaborators suggest is for a critical change in viewpoint. That's how they call it. And they say, look, we have to go with a bottom-up approach. What we have to do is to start with an a priori defined construct of a brain region, which is the physical stuff, according to them, that we have. And then the unknowns are actually the behavioral functions that are associated with such brain region. And we can do that now because they say we can use big data. Now, what I want to do in what follows is just basically first give you a little bit of a sort of an overview about, first of Gannon and colleagues' arguments against the top-down approach. Why is it that they don't like it? And as you will see, I agree with many of those arguments. And then I'm going to reconstruct Gannon et al.'s bottom-up proposal. But then I am going to offer three, I think, very serious challenges to this bottom-up approach. And at the end, I am going to end up with something, and by the way, these bottom-up approaches, these challenges to the bottom-up approach are not unique to Gannon et al.'s proposals, but rather they should generalize to proposals in which the proposal basically suggests that big data is going to be sort of the solution to all our problems. And I'm going to end up with some sort of a final assessment in which I argue that neither a top-down nor a bottom-up approach are sufficient. And instead, I argue for, I recommend what I call a piecemeal approach. So here we go. First, a little bit of terminology. So they use the term behavioral function in reference to, quote, psychology-related constructs such as episodic memory, working memory, motor preparation, visual attention, memory consolidation, and so forth. By contrast, they use the term operation function to refer to a computational operation performed by a given region, which contributes to the observed behavioral output. So these two terms are going to become more clear in just a second. So the main suggestion at the beginning of this proposal for a bottom-up approach is that we should think of every brain region as being associated to many behavioral functions via a latent operant function that unifies them. And here's in their words: our current knowledge of the functional specialization of a given brain region can be conceptualized as a polyhedron with its many sides, that is, its many behavioral functions, the sum of which can only be appreciated by investigation from many different perspectives, but whose core center remains intangible. So to make this idea clear, let me give you an example of what allegedly is probably the best area of the brain, or at least my favorite one, the hippocampus. I am a memory researcher, so I'm pretty much in love with the hippocampus. So here we have a couple of slides showing the hippocampus, and then the idea is to try to understand what the function of the hippocampus is. We can think of it as a polyhedron, right? So there's a bunch of stuff that traditionally has been associated with hippocampal functions, such as declarative memory, associative learning, consolidation, novelty detection, binding. Sometimes they call it as a comparator, spatial discrimination, prospection, and so on and so forth. And the idea is that in the center, or at the center of all these different behavioral functions, there is a core latent center that it is unobservable. And that center corresponds to what they call the operation function of the hippocampus. Now, how is this polyhedron created? Well, traditionally, we have employed this top-down approach. So, for instance, we use lesion deficits, right? So HM and so forth. The problem with lesion deficits is that it is very difficult to identify and isolate pre-lesion factors. We don't know to what extent an individual's deficit is because of the accident or as a result of stuff that happened prior to the accident or even after the accident. Additionally, lesions are not terribly specific. And then there is an additional concern, which is the fact that neuropsychological profiles are only as good as the tests we employ to characterize them. And I am extremely critical of a number of different neuropsychological profiles that give you an idea that, say, working memory is impaired or it is intact in one individual, but it turns out that the tests for testing for working memory are very skewed, biased, or incomplete. One example is the example of HM's working memory. I have written extensively about why it is unclear whether HM's working memory was actually intact, and that there is actually some reason to believe that HM's working memory was actually compromised. Here's another top-down approach: stimulation, like intracranial stimulation. It could be extracranial stimulation, like magnetic transcranial or single or repetitive stimulation, and so forth. There are a variety of them. Typically, stimulation is messy. It's also very limited. If it is extracranial, it's limited to surface cortical areas. If it is intracranial, is usually limited to wherever the electrodes are actually located. Lacks some sort of ecological validity. And another concern, which is often not mentioned, is the fact that when you have intracranial electrodes, is usually because these individuals are undergoing some sort of surgery, and as a result, these individuals are usually neurologically compromised. So it is also all these pre-lesion factors come into play as well. Another approach, another top-down approach that is very typical is, of course, activation approaches, being fMRI being one of the most typical ones, but of course, PET scans, EEG, and so on and so forth. All sorts of problems with fMRI. I'm completely sure that you have seen lots of talks in these sessions about difficulties and the perils of fMRI. Here are some examples: the problem of pure insertion, which is the inconvenience of having to assume when a process starts and when a process ends when you are making experimental designs that have some sort of condition comparison. There are also all sorts of concerns with that specific effect. And despite the fact that we have other, and of course, pure insertion is not an exclusive issue of neuroimaging. We see it with reaction times, for instance. And despite the fact that we have all sorts of strategies, such as multivariate and machine learning approaches and so on and so forth, it is unclear whether they can actually completely eliminate concerns with activation approaches. And finally, there is another top-down approach, which is structure behavioral correlations, basically like looking, for instance, volumetric measures in some cortical structure, subcortical structure, and then comparing that with behaviors of cohorts. It's complicated. There's usually a lack of experimental controls. Often there are alternative explanations, and there is an enormous amount of individual variability when it comes to differences in the structures. As someone who has seen probably thousands of T1s and cortical images, I can tell you that no brain is identical to another brain, and you get to see a lot of differences. Of course, there are one can, for instance, conduct between group studies or large involve large data sets, but usually the conclusions that you can draw from structure behavioral correlations are rather limited. So all of these are well-known concerns from what it is called the top-down approach. And again, and colleagues' conclusion is that this top-down approach is actually inadequate and rather incomplete. This is something that I am to agree with, okay? As a result, they said assessing the relative functional specialization brain region requires a change in viewpoint, where the a priori defined construct is the brain region and the unknowns are the behavioral functions associated with it. All right, so what is this famous bottom-up approach according to Gannon and colleagues? Well, it is easy. It involves just 4 steps. The first step is simply to identify a region of interest, and they gives us 2 examples, the hippocampus and the anterior insula. Then the idea is that you use big data, so brain map, neurosynth, and whatever repository of fMRI data you prefer. And then you do functional behavioral profiling, which is similar to the polyhedra that I showed you before, but now is data-driven. So this is like sort of data-driven polyhedra creation. And then at the end, you infer the underlying operational function. So how does this work in practice? Now, if you're like me, you probably have seen this website several times in your life. This is neurosynth. So what I did is that I just basically looked for anterior insula, and I found, you know, however many papers that just report activity in anterior insula I could find. This is just a quick selection, which I found 681 studies. And then what you do, I didn't even have to read all of them, but basically what you do is that you look at sort of the abstracts of the first few that came out to identify the kinds of behavioral functions that the anterior insula had been associated with in the literature. So you don't have to read all of this stuff because I summarized it in this made-up polyhedra that I just created. So this is just the list of the first few that I found in neurosynth. So you find papers associating the anterior insula with sustained attention, pro-social behavior, social cognition, inhibition, and so on and so forth. Now, their suggestion, Gannon et al. Suggestion, is that once we have all all these behavioral functions and a list of all these behavioral functions, then the latent operation function is going to become absolutely clear. And they suggest that this latent operation function is task engagement maintenance. Now, how did they get to that? It is a very big question, which I am going to get to in a second. So they argued, sure, look, this strategy still is in its infancy, but their suggestion is as you aggregate more and more big data, behavioral profiling is going to become more and more precise. And in turn, the identification of the latent operation function is going to become more accurate. And this strategy, it says, should help with what cognitive neuroscientists are already doing. So here they give us the example of the hippocampus, right? So look, the hippocampus, beautiful structure. People have associated with episodic memory, good encoding, with mental simulation, with spatial navigation. But let's be honest, just like Walter was saying before, we are humans and we made mistakes. We're also humans and we have a finite capacity to reading all these marvelous papers that are being published every single day about the function of the hippocampus. So what big data is going to do for us is that it is going to read those papers for us, and then is going to complete all this list of all the behavioral functions that are associated with the hippocampus. And then we're going to see that it is going to be also associated with consolidation of motor sequences, self-referential counterfactual thinking, this is my stuff, statistical learning, higher order priorities, perceptual discrimination, and so forth. And once we have this whole list of behavioral functions, then we're going to identify what the operation function of the hippocampus actually is. And according to them, this very daunting picture that I painted at the beginning is going to become way more manageable, because all these psychological functions are going to be narrowed down into just a single computational operation that is going to be associated with a brain function. That is the picture that Gannon and many of the people that like big data are going to be pushing toward. And what I want to do now is I want to offer 3 challenges to this bottom-up sort of big data approach. And the first challenge comes with a very simple question. What's a brain region? Well, Eichhoff himself said a couple of years ago that there are many ways to demarcate the basic neurological atoms that are associated with a latent operation function. So what I want to do is to just delve a little bit into the neuroanatomy and the neurophysiology of the one example of what they consider a basic brain region is. This is the anterior insula. So many of you probably know, here's the anterior, here's the insula. The anterior, macroscopically, as you know, the insula is a portion of the neocortex that is folded right inside the lateral sulcus and is covered between 2 and 4% of the total of the cortical surface. And it is enclosed dorsally by the frontoparietal operculum and is ventrally by the temporal operculum right here. So this is an image. Don't do this at home. If you remove your right operculum, then you're going to be able to reveal the insula. Since broadband, we know that it is separated by the sulcus circularis, so it's anterior. The anterior insula is whatever, according to Brodmann, is whatever is rostral to that sulcus circularis, whereas the posterior circular is whatever is going to be dorsal to that sulcus circularis. Now, this was the original cytoarchitectonic representation of the insula by Brodmann in 1909. As you might remember, the different kind of sort of little shapes here indicate different granularities in the cortical layering. So you can see here the sulcus separating the anterior insula. This is the joint of the posterior insula, which is supposed to be connected with the rest of it. Now, that was 1909. I could give you, like I'm literally writing a paper right now about the history of the parcellation of the insula, because I'm pretty interested in that stuff. But just to give you an example, there were several different parcellations between 1909 and 1940. But this is a very different parcellation from Brockhaus in 1940. So here Brockhaus makes the claim that we shouldn't, this is the sulcus right here, the sulcus circularis. Brockhaus makes the claim that we should not distinguish between anterior and posterior insula, because it turns out that Brodmann was pretty wrong about the kinds of granularities that we find here. And here what Brockhaus shows is that there is different granularities the more superior versus the more anterior you find. This portion of the insula has almost no granularity. That is to say, there are very few neurons in layers 4 and 5, as you can see. There is a little bit of more granularity in this midsection, and there is definitely way more granularity in that posterior section. So what corresponded to the anterior singular here is actually divided into 3 different sections for Brockhaus. Now, just to give you even more complication of these views, so this is a meta-analysis of the insula that came by Dean et al. It is supposed to be a functional parcellation. Data-driven functional parcellation, and here what we identify is somewhat 3 different regions, and this one region is sort of overlapping with Brodmann's anterior insula. But just to give you an idea, this is one. In reality, this region that it is about. Oh, sorry, I have the mapping here. This region, I just superimposed that fMRI overlapping here on Brockhaus' original cortical mapping. So this region is about 2 centimeters by 2 centimeters, like it's a volumetric 2 centimeters in each side. Suppose it is an isometric measure. Suppose this is a voxel. You have you have something close to a couple hundred of isotropic functional voxels. Now, in addition to that, each one of these voxels has something of the order of 630,000 neurons. So when you look at just half of the anterior insula, you're really looking at thousands and thousands of neurons that are actually cytoarchitectonically very, very different. So the question is, what reason do we have to believe that all the fine-grained neuroanatomical operations of the anterior insula happen to converge in the single operation function task engagement maintenance? And during the Q&A, you can ask me more about the anterior insula, because it turns out that many of those subregions in the lower agranular portion of the anterior insula are actually functionally connected with different areas of the brain. So there is an even further question as to whether these regions are actually projecting onto the same areas. But here are more problems. Cytoarchitectonic borders are purely delineated. V1 and V2 very nicely columnar organization, one next to one another. But as you go more anterior in the brain, and as you go more ventral in the brain, just cytoarchitectonic borders just really are not nicely delineated between one and another. Additionally, when you use different algorithms with neuroimaging, there is a lot of disagreement among algorithms. And this is known in the literature as the concordance problem. And it is actually pretty complicated at times, because one algorithm and anyone who has ever worked with different kinds of atlases would know that different algorithms sometimes give you a particular coordinate as belonging to one or another region. Moreover, even when that happens, and what usually the typical solution for that is to call a researcher who is an expert in their area, it turns out that there are disagreement among researchers. One of my favorite papers of illustrating this point came a few years ago from Pascal Tremblay's group. What they were showing here is that they ask experts, neurobiologists who are experts in language, to identify Broca's and Wernicke's area, which are supposed to be two of the most important areas for language production and understanding. Briefly, what I show you here is that just the different proportions of how the books in the history of neuroscience have identified these regions. As you can see, there is quite a bit of disagreement. Some of this disagreement overlaps, some disagreement do not overlap. But even if you ask current contemporary experts, you see, for instance, here in Broca's area, this is how I learned it, that it included both the pars triangularis and the pars opercularis. But you have people, for instance, that even include this gyra here toward the inferior frontal gyrus. But more interestingly, you have people, for instance, for whom Broca's area is only the pars opercularis, whereas some people include only the pars triangularis. So depending on which expert you have nearby your lab, you're going to have entirely different responses as to whether one particular region was or not active in Broca's area. And here's even more problems for what a brain region is, because it is possible, for instance, that for many operation functions, the brain regions involved might not even be topographically unified. They might be instead discontinuous bits of neural tissue working together. And this is a point that Fox and Friston made a few years ago in neuroimage. So now this alleged solution of one computational function associated with one brain region actually is going to be more complicated, because this brain region might not be a contiguous, like a unified bit of brain tissue. And if that is the case, then this one-to-one mapping between brain region and operation function might actually break down. So why did Gannon and colleagues pick the anterior insula or the hippocampus as basic brain regions? Well, for two reasons. For one, because of the resolution of our instruments. And second, because our theories, because we have neurophysiological, neuroanatomical, and all sorts of theories that tells us that the hippocampus is somehow a unified structure and presumably has some sort of unified function to it. But then if that is the case, then the selection of this relevant brain atom is both instrument and theory dependent, and not a priori, as they suggest that the bottom-up approach does. So there is, and in fact, and this is, Edvard is a neurophysiologist, a monkey neurophysiologist and neuroanatomist whose work I really like. And he argues that there is actually no such a thing as a theory or a hypothesis-free way of determining what a brain region is. All right. Now, the second challenge, and the first challenge is the longest. The second challenge is relatively straightforward to point out. The problem is that the operation functions or the computational functions are always going to be model dependent. There is no such a thing as a computational function that is model free. So Gannon et al. Mischaracterized the way in which we, cognitive neuroscientists, normally attribute operation functions to brain regions. So consider again the hippocampus, right? So the received view is that the hippocampus was for memory. But as we explore the hippocampus a little bit more, it turns out that not all memory, but only declarative memory was involved. And then it turns out that not all memory processes, but definitely encoding was involved with the hippocampal function. And more recently, there's been all sorts of amendments, right? Oh, look, the hippocampus is not engaged only in memory. It turns out it's also engaged in certain kinds of imaginations, like episodic future thinking and episodic counterfactual thinking. Oh, lo and behold, not only some kinds of imagination, also perception. There's some interesting work by Lucida Saxida and Morgan Barense and a bunch of other people showing that certain perceptual tasks, particularly tasks in perception that involve discrimination of highly overlapping properties, also require an intact hippocampus. And then later on, we have even more refinements. Certain kinds of imagination, such as self-centered, future hypothetical thinking, spatially coherent, and so forth, and only certain kinds of perceptual processes. So what you end up doing, I'm going to skip this next, is that you end up sort of psychologically gerrymandering the function of the hippocampus. And you end up with something that is really difficult to characterize. Well, it is so difficult that there have been entire volumes like this special issue in 2013 in which they ask experts in the hippocampus to determine what the computational function of the hippocampus is. And what I'm showing you here are just basically the abstracts. And what we see in the abstracts is that everyone disagrees as to what the function the computational function of the hippocampus is on the basis of the evidence that is available to us. So Eleanor Maguire and Sinead Mulally say that the computational function has to do with the construction of spatially coherent scenes. Daphne Hammy and Nick Turk-Brown argued that actually there are two functions, one having to do with memory modulation, and the other is some sort of adaptive function on top of that memory modulation. Mark Howard and late Howard Eichenbaum suggest that actually is involved in the relation of spatial temporal information. James McClelland, of course, argue for his dual model system, according to which you have something like pattern completion and pattern separation and so forth. So look, just in 2013, with an enormous amount of data and all experts in the hippocampus, and what we find is an issue that has a bunch of papers in which everyone disagrees about what the computational function of the hippocampus is. So why is there no agreement in the operation function of the hippocampus? Well, because even if there was such a thing as the theory-free notion of a basic brain region, the computational description of its putative associated operation function will likely depend upon the particular computation framework that you, the researcher, like or prefer, right? And we all disagree about the right computational framework. Gannon and colleagues assume that there is something like a Bayesian, that the brain is something like a Bayesian machine. I won't get into detail here, but I'm happy to talk about this at the Q&A. But you probably have seen that there are many ways of cashing out this notion that the brain is Bayesian, and a lot of people disagree about the claim that the brain is Bayesian. So there are disagreements about computational frameworks, and thus there's going to be clearly obvious disagreements as to what the computational function that underlies the behavioral function should be. And the final challenge is pretty obvious when you think about it, is the fact that, look, we never really measure behavioral functions or cognitive functions directly. We measure behavioral functions via tasks. Every time that we put a person in the scanner, we're not asking them to remember something. We're asking them to do a task that we hope indexes memory. But oftentimes, there are disagreements as to whether a certain task measures one or another behavioral function. And I'm just going to give you a couple of examples of such disagreements and why they become really consequential. So one example involves a task to measure skill learning, and another is an example of a task that is allegedly supposed to measure working memory. So I'm going to give you, this is an example of a historical example that I really, really like. So as you might know, HM allegedly was able to learn new skills. That's what the textbooks tells us, right? Well, in 1965, the now 103-year-old Brenda Milner tested HM on a number of different kinds of allegedly skilled tasks. This one was a visually guided maze. It was very simple. These were all metal pegs on a board, and the participant would have a metal stylus. And then the task was to start with this metal pole, and then sort of follow an invisible path that they didn't know it existed until they will end up in the finish pole. Every time that they made a good move, there will be silence. But if they made a bad move, then there will be a noise. So this will be like silence, silence, meh, silence, 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 meh, that sort of thing. And then the idea is that they will learn how to move in the right path until they've had the finish. Now, controls took about 17 trials, and they made on average 92 errors. HM, after 215 trials and 2,877 errors, they stopped the testing. They never learned this task, right? So in 1968, 3 years later, they said, okay, let's do a simplified 5 by 4, just this size. And in that, controls took, in 20 trials, that was the upper limit. All of them took less than 20 trials, and by the end of those trials, they have 0 errors. HM took, on average, 155 trials. He had to be brought back to criterion every day after that for 14 days until the error curve was comparable to controls. And in fact, HM showed no transfer learning, even with a path that was just slightly longer than the one that just learned. discussed these issues in this article a couple of years ago. Now, why do I talk about this task? Well, because this task is extraordinarily similar to this task that is called the zero reaction time task, that is the paradigmatic task for what? Skill learning. And I won't get too much into this task, but this task is essentially identical to the smaller version of that maze task that HM failed to do. And in fact, nowadays, they're like in this paper on motor learning that came out last year, John Krakauer's group, they argue that actually the zero reaction task might be a really bad task for measuring motor and skill learning. Another example with working memory is the N-back task. There is a lot of discussion as to whether the N-back task is actually a working memory task or actually a cognitive control task or an attention task. Now, the problem with this, of course, is that if you have big data and you get a bunch of results that tells you, all these areas are associated with skill learning, but the task that they use is a task that conceptually now we think is not really a good task for measuring skill learning or working memory, you're not going to be able to identify from that data which tasks really are tasks that are measuring working memory or skill learning and which ones aren't, because this big data is supposed to be reading that off from you, right? So the problem is that when we reconceive what a task measure, right, and it's not because there is something changed about the brain or because something changed about the task, but rather because we, researchers, reconceived the nature of the cognitive or the behavioral function that this task was supposed to measure. There's a beautiful work by my colleague Tobias Engler about why working memory must actually be conceived as internal attention. And as a result, if there is such a merging of memory and internal attention, then it's going to be just wrong that certain tasks are giving you information about attention as opposed to working memory, right? So the problem is that as a result, we might mischaracterize the operation function because we wrongly categorized the behavioral function that it was supposedly indexing. Now, big data might reduce this concern, but it's just not going to eliminate it. I can talk about the unique task problem, which is related to this in the Q&A. So the problem with this is that this conceptual confusions about the right sort of computational description of a putative operation function that just simply won't get averaged out from aggregating massive amounts of data. And the reason is because they are not noise. They are just simply the wrong kind of signal. So in sum, there are three challenges to the bottom-up approach and in general to any bottom-up approach that capitalizes on big data. We have questions about what a brain region is. We're also questions about how to identify the operation functions, because as I try to argue, they're always going to be model dependent. And finally, there are really serious conceptual challenges about mapping tasks to behavioral functions, because we might actually reconceive what the task is actually measuring. So to end, let's ask what to do. Well, I think that both the top-down and the bottom-up approaches are incomplete, but probably they're needed. Having more data is way better than having less data, but it is also the case that it is important to use single patient data, and it is fine to continue using top-down approaches and bottom-up approaches all the same. But we have to refine our methods, and presumably I think it is important to standardize, to use certain things like standardization and cross-validation of neuropsychological testing, stuff that we know from machine learning. I think it is very good to include other lessons from machine learning, like regularization via penalization and things of that sort that you probably have been discussing in these sessions. But I also think that it's extremely important that we accept the validity of what I call piecemeal approaches. Basically, theoretical and conceptual pieces about how to resolve local discussions about what a brain region is, what are the computational frameworks that are valuable, and what are different tasks that are relevant to measure different kinds of cognitive functions. In other words, I think that we really have to dedicate a lot of time to conceptual and theoretical clarifications. And to anticipate the questions, how can we do that? Well, my suggestion has been for quite some time that scientists need to hire a philosopher, because we philosophers transit in conceptual clarifications. That's what we are trained to do. So with that, I end. I would like to thank the two co-authors, my former student, Dr. Brian Gessel, who's a philosopher now, a professor in Virginia, and Dr. Matt Stanley, a psychology and neuroscience student, and now a postdoc at the Fuqua School of Business here at Duke. And thanks to you.
I will clap on behalf of everybody in the audience. Thank you so much for this brilliant talk. And from the chat, I think we have all become soberly disillusioned about how much we actually know about the brain, but in a good way. Because if we don't know there are problems, we can't even go about solving them. You have to first identify that there is even a problem to begin with.
So
I
apologize. I couldn't read any of the questions while I was talking.
No, that's fine. That's what we'll do now. So we've actually got a couple. The chat was busy, and part of that is my fault. I like to kind of jump in and comment on how some of what you guys are talking about. This is the beauty of Neuromatch Academy too, is that you guys didn't see all the materials ahead of time. You don't know exactly what every single day at NMA went through, and yet you've touched upon some very core topics to what we're trying to come up with. So for everybody in the audience, turns out we all are kind of converging on the same skepticism and ideas, even if we don't explicitly communicate behind the scenes. But we've got a couple questions that have come in. Some of them are still from Walter's talk, and I know that Walter, you answered some of those in the chat and in the Q&A, which is great. We did have one specific question from Yiti Lee about plasticity rules and shaping hippocampal activity. That's very, very specific. So we can go with that one if you'd like to. Or we can go first to maybe some more general questions and comments. Actually, you want to do it really quick?
I was just going to say a couple of things. So beautiful. So yes. So heavy on learning is definitely involved in likely in hippocampal computations, but also there is heavy on learning everywhere. So one misconception that we have about the hippocampal function is that we think that hippocampus is somehow special because, for instance, it has long-term potentiation. It turns out that there is long-term potentiation everywhere. One of the main reasons why we think it is mainly in the hippocampus is because it has been most widely studied in the hippocampus. But there is long-term potentiation in several other areas. For instance, in the granular region in the anterior insula, there is long-term potentiation. You see long-term potentiations in structures in the anterior cingulate cortex and so on and so forth. So, and likewise with heavy on learning. A lot of the same plasticity that occurs in the hippocampus, you can see elsewhere in other pyramidal neurons. One thing that I do highlight, though, and this is a computational neuroscience crowd, so you might appreciate this. I think that in addition to looking at the particular kinds of computations that we think neurons have, it is important to look at the structure. And this is an insight that you get from Marr. Many of you know Marr from the Vision book, but Marr was really a pioneer in computational architecture of the hippocampus and in the neocortex. And in particular in the hippocampus, he was very interested on reentrant activity in CA3. And he thought that the reason why the neurons were sort of circled around themselves must have to do with the timing. So one thing that I would say is, like, look at the structure of the neuron. I know that we are accustomed, we as computational neuroscientists are accustomed to thinking that function and structure might be somehow independent, but there might be a reason why certain neurons are structured the way in which they are. So that's kind of like my intriguing, to be developed message here.
So what you're saying is we can't just assume that all cows are spheres, as physicists do, and just kind of go with the oversimplified way that morphology is really important, and that depending on the questions you're asking, you need to pay attention to that. There may be some questions that don't depend on morphology, but we shouldn't assume that the abstract representation of a network or system is sufficient.
Absolutely. I think that so you might remember from in the early discussions in the 1980s when Rumelhart and McLellan were trying to be concerned about backpropagation algorithms because things were happening too fast. So you needed to slow down the flow of information in certain layers of the network so they wouldn't like reach outer layers first. Well, this insight came from Marv 15 years earlier, in which he thought, how do you do it? Well, you don't insert more layers in the middle. You make those layers longer. If you take a detour that is much longer than going straight, yeah, that is a way of guaranteeing that you both are going at the same time. He thought that certain regions in the hippocampus, because the neurons that were sort of circled around themselves, might have to do with timing. So I think that we should not simplify a structure where we're looking at neurons, even at the computational level. Structure might be telling us something about the kinds of computations that they're doing.
Perfect. So Walter, I've just unmuted you as well. Yeah, we can have a little bit of time for discussion here. If people have other questions, we've got a couple that have come in as well. But also, I want to give you two a chance to kind of respond to each other if there's something in particular that you'd like to highlight from each other's talks and bring up in the last few minutes here before we close up.
I don't know. Well, yeah, go ahead.
So I'll ask. Oh, go ahead.
I was just gonna say, this is a little bit of an anecdote, which speaks just directly to what Walter was mentioning. So when I was in grad school, in my first or second year in grad school, people might not know, but I was doing sort of a dual PhD in philosophy and in cognitive neuroscience. And my advisor in cognitive neuro came to me one day and said, like, look, Felipe, there is something weird about this paragraph in this paper that I am reviewing. And I read the paper and said, you know what is weird? What's weird is that this is a logical fallacy. They are making, they are literal, like that. They're trying to make a conclusion. You know, it was like the affirmation of the consequent. I cannot recall exactly what it was. And I just went like first order logic. I just formalized it. If the P and P, that's like a fallacy, and did exactly the technique that Walter was suggesting today. And it was like extraordinarily insightful and just simple. It's a very simple explanation. So look at the way in which discussions are written, because often they hide fallacies or arguments that are invalid.
And it's not only neuroscientists. I want to say it's philosophers too. One of my favorite stories is Kurt Beyer, who actually taught logic, but he wrote a book on ethics. And in the end of one of his books on ethics, he had this argument, and somebody said, well, that's just a fallacy. And then one of his students said, no, it's not. And they fought back and forth for like 10 articles until Kurt Beyer wrote a response that goes, yes, it was a fallacy. It was a bad argument. Yes. I admit it. So the trick is, number one, to be able to locate the fallacies. But number two is admit it when you made one too, because you're human like everybody else. And don't get stuck and spend your life trying to defend bad arguments that you gave in the past.
And that's another good lesson that you can learn from philosophy, I think. So one thing that I find interesting is that, look, I have friends in philosophy that hold crazy views, like crazy banana views. I mean, I'm going to say it here. I don't know if this is recorded or not, but I love Dave Chalmers. Is it recorded? Okay. I love Dave Chalmers that's fine. I love Dave Chalmers to pieces, but I think that his view about consciousness is crazy bananas. And we can go to a talk and he will give it and I will argue against him, and it will be a back and forth, and it could get heated if you want. And at the end of that, we will go for a beer and we're like good friends. There is like nothing happens, right? Because we as philosophers, at least I feel as a philosopher, I'm trained the way in which Plato suggests that we are mouthpieces of reasons. We are trying to do our best in what is a very complicated topic, right? And it is fine to err. We're doing our best, right? I think that science could learn a little bit from that. I feel that a lot of the concerns about replication crisis and stuff like that is because somehow we scientists feel that we own the result. And if there is, it must be our fault. No, it's the world's fault. It's fine. It turns out that the generalization didn't hold, like there were some boundary conditions that we didn't control for, and so on and so forth. It is fine. It is fine to make mistakes. You don't own, like, if it is not true, it's not your work. It's not your fault. You did your best. It's the world's fault. So just go have a beer and be friends again.
I think that's very sound advice.
I don't know if it's valid, but it's,
So I have a question for you, Felipe.
yeah, go ahead.
So this problem of structure function and mapping the computational models onto the, the actual structures of the neurons, it's very complex. Is it specific to the brain, or is the same thing apply to the kidney?
That's a very good question, because in fact we actually do discuss nephrons. And this is very interesting when you look at neuroanatomical books. People normally think of nephrons as the functional unit of the kidney, right? Or the liver, yeah, the kidney. Sorry, yes, kidney. And then the idea is that they are relatively homogeneous, and whatever happened to one is going to happen to another, and so forth. And I gave a talk once in front of a bunch of medical doctors, and I was trying to make a point to the effect that neurons were not like nephrons, because nephrons are completely identical to one another, and what it goes for one neuron, what nephron gets to another nephron. But neurons, they are very difficult, they're complicated, and in addition to that, we have oligodendrocytes and astrocytes and so on. And boy, the rhinologist got mad at me. It's like, no. Nephrons are extraordinarily complicated. No nephron is identical to another. And then they went on, and so, and it turns out that there is quite a bit of discussion as to exactly how to identify. Like, that's what nephrology is like, right? Like the people interested in, and then they're trying to do. Now, I think that there is one difference, however, which is the fact that most of the functions, the biological functions of things like kidney and the liver or what have you, even though there is some penumbra as to exactly how to characterize them, usually what you have is some relatively easily quantifiable behavioral stuff, and then there is no further questions. Now, I think that if our kidneys were self-aware, or if our kidneys were doing some stuff other than the stuff that they biologically do, then we will have like a kidney function to the kidney structure problem. But they don't. Typically, the range of operation or behavioral functions of those organs, it is much more limited, and the discussions are less overwhelmingly difficult. The brain is not in that sense like that, because it's vastly more complicated what this a range of behavioral functions might be.
I just wonder whether a kidney specialist would agree.
I don't know, but I gave it, and oh boy, did they get mad. It's like, no. Nephrons are very complicated too. It's like.
Well, on that note, which is that brains are probably more complicated than kidneys, we think. Yeah. We probably should close this session up here. It's getting a little bit late. It's late for you guys on the East Coast. So yes. I think before we go, though, I will ask, I'll put you both on the spot and say, do you have any final words of wisdom that you have not already imparted to our audience that you would like to share?
Oh my gosh.
I know it's a big question. It can be as simple. Here's mine. Take philosophy. Yeah. I already said it. I'll say it again. Take philosophy classes. If this session did not convince you, please listen to these words. Take philosophy classes. They will help you be a better scientist. So that's mine.
I think I would quote my old high school buddy, Joe Sanders, who wrote a song that said, always go with your heart. Don't pick a topic to study simply because you think you'll be able to get papers published on it, and it'll help you get tenure and all that. Pick the topics that you think are really important and work on those. I think too often people, pick the topics for the wrong reasons. Find out what's important to you and work on that. That'd be my word of wisdom.
Yeah, I will just echo both of those. Take philosophy and or hire a philosopher. Do what you like and what you love. You're going to spend an enormous amount of time doing this sort of thing, so you better like it. And the last thing is fine to be wrong.
Yeah. And it's fine to admit you're wrong.
Yeah.
That's true.
People will think better of you if you admit you're wrong instead of if you dig your heels in.
put in a word for the Templeton Foundation and the Templeton World Charity Foundation, because one of their big, projects is on humility. And I think it's extremely important, and there's not enough of it in academia.
Yes, agreed.
Well, I think we all got a pretty hefty splash of cold water and humility with these talks, which is good for us to show us how far we've got to go. But it also, to me, is very exciting that this is a super complex system, and it is hard to study. And for me, that's kind of what makes it so exciting for me to do this. Like, I don't want to work on simple problems. I want to work on hard problems. And this is maybe one of the hardest problems that we can solve, maybe aside from, I don't know, like astrophysics or something like that. But brains are
harder
hard.
than astrophysics. Come on.
Brains are hard. Brains are hard, and that's what makes them fun. So we do have a couple other questions in the Q&A, but in the interest of not keeping people too long, I think we will close here. I want to thank you both again so much for being here and for sharing all of this with us. I learned a lot. This was great fun. And we'll also thank the Templeton World Charity Foundation for sponsoring Neuromatch Academy again and for sponsoring this session. And if you like this, and if you like talking about philosophy and logical fallacies and that kind of thing, we'll be back on Thursday at 9:30 a.m. Pacific, 12:30 p.m. Eastern Time, and whatever that translates to in Europe and Asia. But put it on your NMA calendars. We'll be back to talk about consequences of neuroscience and law and medicine and business. And
if I can make one last plug. So if you are interested in philosophy and in neuroscience, you should consider applying to another project that is funded by the Templeton World Charity Foundation, which is directed by Walter and myself, which is called the Summer Seminars in Neuroscience and Philosophy. Is housed at Duke or SNAP.
Yeah.
So you should check it out. And if you're interested, you should apply.
I'll second that. Great initiative, great program. So thanks again, guys. This was really fun.
Thank you for having us.
Yeah.
Yes, thanks so much.
We'll see you next time.
Thank you.